Hi everyone, I hope I'm audible and visible. Hi everyone, I hope I'm audible and visible. Hi guys, so welcome to PW NEET English. I'm your biology educator Ambika Sharma, and today's session is very important. You know what are we going to discuss today? You can see here as well, Plant Physiology NCERT plus PYQs. This is our topic for today. So today it is not just going to be the NCERT reading in detail, in depth. We are going to revise all the 3 chapters from the Plant Physiology. I have seen students complaining this thing again and again. We do not understand plant physiology. We do not understand the concepts there. We do not know what is ETC, what is glycolysis. So guys, I am here and I'll simplify everything for you. But there is one condition, guys. There is one condition. And what is that condition? That condition is you have to be with me till the end. You just have to grab a pen. You have to keep your NCERT with you all, and you have to keep underlining it. And to understand plant physiology, we will go a little beyond NCERT as well. So here on this particular channel, when it comes to my lectures, beta, I'm not going to compromise with the quality of the content, right? It is going to be top notch, and the most important units we are going to discuss in detail, isn't it? Isn't it a good option? Yes, everyone. Yes? Yes? Tell me quickly. Isn't it? Ma'am, please finish genetics first. Now, beta, we'll complete the genetics later. Right? Let's finish the most important topics from the class 11th, and then let's start the genetics. Your request is accepted. So before going forward, make sure you hit the like button. Make sure you subscribe our channel, right? So that you will get the update regarding the quality content, right? Regarding everything. So everything whatsoever is required for you, right, students? This will be provided to you from this particular channel. You are going to get everything, be it the content, any important update, everything will be provided to you. So this is what we are going to start today. So do not worry, bacha. Genetics will also be covered. Ma'am, when will the Yaqeen NEET English batch start? Beta, it will also start soon. Any other query? Any other query? Yes? So make sure you hit the like button. Make sure you subscribe our channel. So let's get started, guys. Let's get started. So let's start with the very first chapter. Let's start with the very first chapter, that is photosynthesis. And if you guys are having any other right request that ma'am start respiration first, so whatever you people will say, I'll move accordingly. So do let me know in the chat section. Do let me know in the chat section. So there is a condition for today's lecture. Nobody is going to ask me the duration. Nobody is going to ask me the duration. It's our kind of Sunday, right? It's weekend for you, it's weekend for me, and here we are going to utilize it, okay? And in a good way, in a good way. Yes? Ma'am, what is hepatic portal system? It comes in PYQ many times. Beta, we will, Neha, through a shot, you will get the update regarding hepatic portal system as well, a very basic concept. And the frog part also, I will discuss it. Any other thing? NEET, Yaqeen NEET English will start soon, right? Anything else? Beta Faisal, I think this question is not relevant. Okay, so should I start? Obviously, be it the Yaqeen NEET English batch, Arjuna NEET English batch, or the Lakshya NEET English batch, we always start from the basics. You can see my YouTube lectures as well. I always start from the scratch, right? So I'm going to start. So if you want me to start with the photosynthesis, so everybody write some fire emojis in the chat section that yes ma'am, go for the photosynthesis. Let's discuss the photosynthesis. The photosynthesis which we have been studying from last so many years, but still we are not clear what exactly the photosynthesis is. So ready? Yes, everybody ready? Quick everyone, ready? So should I start the photosynthesis? Should I start the photosynthesis? So would like to see some fire emojis here in the chat section that yes ma'am, quickly start it. Yes. Teju, before 10th of June, the complete biology or the complete physics and chemistry will be done and dusted. Anything else? Sure. Some more fire emojis, and make sure you share the session link with your friends as well. Let them know that in today's this particular session, right, the depth of plant physiology you all are going to understand. Quick, you all are going to understand. Quick. Quick. Okay, so let's start with the very first, right? Let's start with the very first topic, that is photosynthesis in higher plants. Right, students? What is the name of the topic? The name of the topic is photosynthesis in higher plants. This is our topic for today. Okay, this is our topic for today. And trust me, you are simply going to love it. Okay, you are simply going to love it. So we are going to study photosynthesis in higher plants. So first of all, let's break down the things and let's understand the basics here, and then let's go ahead. So the word here is synthesis. So what is the meaning of synthesis? Synthesis means formation. What is the meaning of synthesis? Synthesis means formation. And when I'm saying the word photo here, I'm talking about the light. So in the presence of light, there is the formation. There is the formation of what? There is the formation of food by plants. We know it very well. Okay. So now students, you know it very well. When we talk about the plants, right? They have the pigments. Firstly, I'm giving you the overview here, right? Plants, what do they have? Plants, they have the pigments, right? They have the pigments. And what are these pigments? They are again the proteins, right? Protein complexes. So they are the one that are going to trap the sunlight, right? Like we discuss about the chlorophyll, chlorophyll A, chlorophyll B, etc., different pigments and different plants. So firstly, they have the pigments, right? Mainly, we talk about the chlorophyll here. So what are they going to do? They are going to trap the solar energy, right? They are going to trap the solar energy, and then they are going to convert that solar energy into the chemical energy. They will convert that. They are going to trap the solar energy. They are going to convert, right, the solar energy into the chemical energy. Isn't it? Isn't it? And here in photosynthesis, you will see carbon dioxide and the water. It is required. Right, carbon dioxide and the water is required, right? By trapping the light energy by pigments, they'll be converted basically. So what is going to happen? There is the reduction of the carbon dioxide in this particular process. There is the oxidation of the water here, right, students? And ultimately, carbon dioxide will get reduced. It is going to give us what? It is going to give us the sugars. It is going to give us what? It is going to give us the monomers. Is that clear? These are carbohydrates. Is that clear? Is that clear? And obviously, so this is what we have to understand here. This is what we need to know here. Is that clear? Right? And here in this particular process, we will also see the oxygen will also release. So this is what basically we have to discuss here in this particular process, in which carbon dioxide is getting reduced, water is getting oxidized, right? So reduced carbon dioxide is going to give you the sugars, it is going to give you the carbohydrates. So this is what we have to discuss here. Is that clear? So if there is one reaction, in which oxidation and reduction both are occurring simultaneously, can I not say that it is redox? Can I not say that it is a redox? It is the redox reaction in which, right? So photosynthesis is what? It is the redox reaction. Number one. As I told you, we will start from the basics. We'll start from the scratch. So we have the redox reaction here. Plus, here there is the synthesis. Build up is there. So what should be the word? The word is anabolic. Anabolic is the word because build up is there. Right? Something is getting synthesized here. Okay? And when you synthesize something, then what's going to happen, students? This is going to be a process in which energy will be used. So it is going to be endergonic. It is going to be endergonic. So these are the details that we have to understand here in this particular topic, that is photosynthesis. In depth, we are going to study what is light reaction, what is dark reaction, what do you study in light reaction, what do we study in dark reaction. Everything in depth we are going to complete. And yes, this is a point that we will start from the scratch. And especially the chapter photosynthesis, right? We will start it in detail so that you guys can have a better idea of that. But one thing that I can assure you, after attending this particular lecture, you do not have to go or you do not have to study anything else. Just solve the PYQs, read the NCERT, everything will be on your fingertips. So that's what you have to keep in your mind. Is that clear? That's what you have to keep in your mind. This is what photosynthesis is. Now, moreover, plants, they are converting one form of energy into another. They are converting one form of energy into another. Is that clear? Is that clear? Yes or no? So what do you call them? You call them as transducers. Why do we call them as transducers? Because they are converting one form of energy into another. So that's what you have to keep in your mind, students. So let's get started. Let's discuss it. The notes will be shared in my own Telegram group as well, in our WhatsApp channel as well, which you guys can join from the link which is given in the description box. You can be the part of our WhatsApp channel as well. And moreover, students, we have started a free batch on PW. Basically, if there is a free batch, you can check our app, right? You are going to find a free batch, free RE-NEET test series also. So do join that as well. Okay, now come back here. So what are we saying? That when it comes to the photosynthesis, it's a physicochemical or photobiochemical process. It's a physicochemical or photobiochemical process. Okay, so what is happening here? The organic compounds are synthesized. What will be synthesized here, guys? The organic compounds, they will be synthesized. Can you see that? So organic compound means I am talking about the carbohydrates. So they will be synthesized from inorganic raw materials. How will they be synthesized? They'll be synthesized from inorganic raw materials, guys, that is water and CO2 in the presence of light and the pigments. So that's what you need to keep in your mind. So organic compounds will synthesize from inorganic compounds in the presence of light and pigments, and the oxygen is evolved here. Oxygen is evolved here. So this is the balanced reaction that you people need to know. So yes, the 6CO2, 12H2O, sunlight and chlorophyll is required. You will have the sugars right here, and the water will release, and the oxygen will is also the byproduct. But with the time, we are going to understand, right, that oxygen will be the byproduct if you are using the water here. If you are not going to use the water here, if at the place of water, if there is H2S, then obviously oxygen will not release. So you cannot say that all the organisms that do photosynthesis, they all release the oxygen. No. There are organisms which show N-oxygenic photosynthesis as well, where there is no, right, N-oxygenic photosynthesis as well, where there is, right, no oxygen release. So this is also what we are going to discuss here, right? So photosynthesis overall, guys, what is it? It is a redox reaction, right? So oxidation of water, it will occur during the light reaction, right? So there is the oxidation of water as well, which will take place during the light reaction, students, and reduction of CO2 will be there, right, which will be there in biosynthetic phase. So in light reaction, which are two parts of the photosynthesis, light reaction and the dark reaction. So in light reaction, you people will see the oxidation of water is there. Water will get oxidized, right? It will provide H positive, that is proton and electrons. And in the dark phase, in the dark reaction, in the biosynthetic pathway, we will see that there is a reduction of CO2 as well, right? The carbon dioxide will be reduced. So yes, as I told you, it is going to be anabolic and endergonic, that is energy absorbing process. So I hope up to this part everything is clear. Now we will talk about the experiment that are going to say that why there is a requirement of chlorophyll, there is a requirement of light, CO2 for the photosynthesis. So these are the experiments which we are also going to discuss in detail here. Is that clear? Yes, everybody, is that clear? So let's start with these experiment as well, where the very first is variegated leaf. So do you understand what is the meaning of variegated leaf here? Right? So we have variegated leaf experiment. What do we have here? We have the variegated leaf experiment. Is that clear? Is that clear? So when it comes to the variegated leaf experiment, what does it mean? See beta, so here you have the diagram as well. You all have seen such leaves. There are some leaves in which you will not see that they are only green in color. They have different colors as well, something like this, something like this. Okay, so now in this particular experiment, right, what will be done? Just say, let's suppose there is a variegated leaf. Okay, please pay attention. There is a variegated leaf or a leaf which is partially covered with the black paper, right? Let's suppose you are having a leaf. This is the leaf that I have. So either it is variegated, like some part is green, some is having some other colors. Now let's suppose even if it is green, or you are basically covering, right, you are covering a part of it with the black paper, right? Either variegated leaf, or you can take any leaf or green leaf, and you are covering it with what? You are covering a part of it with the black paper. Is that clear? Now what you have to check? You have to check the starch synthesis in that. So what will you find? You will find that the starch will only be there in the green part. Why? Because that green part is basically having the chlorophyll. Is that clear? When it is exposed to light. So these are some basic experiments that they conducted. They observed, they checked the starch synthesis. So that is why, right, they came to the conclusion, right, that the photosynthesis occurs only in green parts of the leaf, and that too in the presence of light. Right, that too in the presence of light. Because when that leaf is covered with black paper, right, when that leaf is covered with black paper, because it is covered with black paper as well, it is covered with black paper as well. So obviously here you will see that firstly the green part, it has to be, light should be there. So they both are required for the starch synthesis. So if anybody is having doubt, madam, what is starch? Guys, I hope you know that. So your glucose, right, it is the monomer, monosaccharide. Right, so glucose together, many subunits of glucose, right, they'll form amylose, amylopectin, and then finally you have the starch. So starch is basically a polysaccharide, right? Its monomer or its single unit is basically the glucose. So starch, right, starch is the word here, starch, right? So plant, they store food in the form of starch. So now we will discuss about Moll's half-leaf experiment. So diagrams, right, they are shuffled beta, they are shuffled, right? So don't worry about that. So this diagram, it was supposed to be there on the previous slide, so do not get confused. Okay, this is not the diagram that we need here. So this is basically for that variegated leaf experiment. So I'll write it here, and before sharing the notes even, I will edit this portion for you. Okay, I'm going to edit this portion for you as well, don't worry about that. Okay, so here you can see next experiment is Moll's half-leaf experiment. It is going to be very interesting for all of you. Right, just say in Moll's half-leaf experiment, what they did, let's take the example of a beaker, a vessel. Just say, let's suppose this is a kind of beaker or a vessel. Now what's the point? In this particular beaker or a vessel, in this particular beaker or a vessel, a part of leaf is present here and rest of the part is present outside. A part of leaf is present here and rest of the part is present outside. Now here you are having some cotton balls which are soaked with what? They have KOH. What do they have? They have KOH. What do we call this KOH? What do we call this KOH? Quick everybody. Quick everybody. What do we call KOH? Yes. What is it? What is it? Quick everyone. KOH, what's that? It is beta, obviously K, potassium. It is potassium hydroxide. It is potassium hydroxide. So this is the experimental setup. So basically what they did, right, there is a leaf, a part of this leaf is in this beaker, and this particular beaker is having the cotton balls which are soaked with what? Which are soaked with this potassium hydroxide. So now what's going to happen here? Please pay attention. A part of leaf is in the test tube with KOH. This KOH, it absorbs CO2. I'll explain the logic behind as well. Don't worry about that. I will also explain the logic behind. So it is going to absorb KOH. It is going to do what? It is going to absorb KOH. Why? We will discuss that. But keep it in your mind, potassium hydroxide present here is going to absorb the CO2 here. So while the other half is exposed to here, so that's why I'm telling you, just just try to imagine this, that you are taking a leaf, a part of it is in the in that beaker where KOH is there, and rest of the part is outside. Now what is going to happen here? What is the observation? So obviously the experiment, it is in the light. So exposed part is showing the starch presence, but the part inside the KOH is negative. The part inside that beaker is not showing the starch synthesis. Now, why is it so? What is the case here? Because carbon dioxide is not available. Carbon dioxide is not available, students. This KOH, if it is going to, right, this KOH, if it is going to absorb CO2, right, then it means the CO2 is not available, right? If it is going to absorb CO2, it means the carbon dioxide is not available for this particular process. Initially, I told you that this is the reaction 6CO2, 12H2O in the presence of light and the pigments. You are going to get the glucose there. Water will release, the oxygen will release. Now here, CO2 is not available. Why is it not available? Because KOH has absorbed the potassium hydroxide. Isn't it? Isn't it? Right? So the leaf, so this will be tested. So the leaf is spread out, right? And iodine solution will be poured on it. So, okay, fine. Got it, ma'am. The part which is present inside the beaker, that it doesn't show the starch synthesis, rest of it will show. But how and why? Because this how and why is not written in NCERT. So this is what I'm going to tell you, and this is my job, obviously, as a teacher. So what is happening here, bacha? So first of all, you will take that leaf, right? You are going to take that leaf. So now when you will take that leaf, on that leaf, when you are going to pour the iodine, because it is going to tell whether the starch is there or not. If bluish coloration is there, starch is present, otherwise it is not. It is not the basic test. So if starch is present, that part will turn blue-black. If starch is absent, that part of leaf will be brown-yellow. Right? So this is telling us that CO2 is important. And why? Why do we take this potassium hydroxide, which is also known as caustic potash? It's simple. It is basically going to absorb CO2. This reaction is going to take place. This reaction is going to take place because your KOH is a strong alkali. It's a strong base. And your CO2 is what? It is an acidic oxide. It is an acidic oxide, a normal chemical reaction, right? This is the chemical reaction which is taking place here. This is the chemical reaction which you have to keep in your mind, that why is it taking place? So when the part of leaf is inside the beaker, because there is KOH, which is basically potassium hydroxide or caustic potash, it is basically the alkali, right? And CO2 is the acidic oxide. So this strong alkali is going to combine with it, right, students? Right? So acidic oxide is coming in contact with this strong alkali. It is getting converted into the potassium carbonate and water. So you do not get CO2 free here. That is why, right, you will not find the CO2 free here. So that is why beta. So we can see that CO2 is important for this photosynthesis. That's what you have to keep in your mind. Is that clear? Is that clear? So why KOH is preferred here in this particular experiment, beta? It is highly soluble in water. These are basic points, right? Because it can dissolve easily, and it is even in schools and colleges easy to deal with, right? Moreover, here when this reaction occur, you are not going to find anything whitish kind of liquid there, right? Properly, because they are highly soluble, uniformly dissolve it will, and it is, right, it will make it more efficient at absorbing the CO2 completely. And it's a very strong base. That's why it is preferred here. Immediately it will combine with the CO2. It is not going to leave a trace of CO2 free there, and there will be no unwanted byproduct. That's why KOH is preferred. Okay, right? So magnitude of, so this is the Moll's half-leaf experiment. So next point is, what is the magnitude of photosynthesis? So when you talk about the magnitude of photosynthesis, it produces more than 170 billion tons of dry matter annually. It produces more than 170 millions of dry matter annually. So annually about 7.0 into 10 raised to power 13 kg of CO2 is basically fixed through photosynthesis. Basics there. Now, next experiments that we have to discuss. Here, you have to remember a trick, that is PIS T Cartoon Network RHK. These are basically the initials that you have to remember. What you have to remember? You have to remember PIS T, Cartoon Network, RHK. PIS T, Cartoon Network, that is CN, RHK. So repeat that. PIS T, Cartoon Network, RHK. That's what you have to remember. PIS T, Cartoon Network, right, RHK. Now, what does it mean? What does it mean? Ma'am, what do you want to say? Basically, how are we going to remember? PIS, J is common here. So, first, Joseph Priestley. Then, Jane Ingenhousz. Then, Julius von Sachs. So, beta, these are not the Indian names, so might be, right, the pronunciation is little different. So, just remember this. So, Joseph Priestley, Jane Ingenhousz, Julius von Sachs, then T, right, that is T. W. Engelmann. Then you have Cornelius van Niel, very important. And then we'll talk about Ruben, Hasid and Kamen. So this is basically the sequence of experiments which we have to remember. What's that? This is basically the sequence of experiments that we have to keep in our mind. So Joseph Priestley, Jane Ingenhousz, Julius von Sachs, T.W. Engelmann, Cornelius van Niel, Ruben, Hasid and Kamen. Right. So once again, let's repeat it. So we are talking about the Joseph Priestley, Jane Ingenhousz, Julius von Sachs. So J. So it is J. J is the common PIS, P-I-S, PIS, P-I-S. You can repeat it again and again. Try to remember everything here in the class. PIS. Right. So first of all, Joseph Priestley. Joseph Priestley, because it is going to be JP, JI, JS. Then Jane Ingenhousz, Julius von Sachs, T.W. Engelmann, Cornelius van Niel, Ruben, Hasid and Kamen. Is that clear? Ruben, Hasid and Kamen. Is that clear? Is that clear? Is that clear? Done? Sure? So, scientist name should be clear. So, try to remember everything here in the class. So, JP, JI, JS, right, PIS, T, T.W. Engelmann, Cornelius van Niel, Ruben, Hasid and Kamen. So, you have Joseph Priestley, Jane Ingenhousz, Julian von Sachs, T.W. Engelmann, Cornelius van Niel, Ruben, Hasid and Kamen. So, let's start with the first experiment here. Let's talk about the Joseph Priestley's experiment. Ready, beta? So, make sure you open your NCERT and you underline the portion there as well, okay? Right, so 1733 to 1804. Okay, so in 1770, he performed a series of experiment, right? In 1770, he performed a series of experiment. Done? So this is what you have to keep in your mind. So I'll tell you. So Joseph Priestley, 1733 to 1804. So in 1770, he performed a series of experiment, right? He performed the experiment on green plants. So I think you all are well aware of it. So I forgot to add the diagram there. If you remember the bell jar experiment. Here what we have to discuss, bacha. Here what we have to discuss. Here we need to talk about the bell jar experiment. Do you remember that? Do you remember that? So there is bell jar experiment. In one bell, right, you are having the candle, and then what else do you have? Having a mouse there. My mouse is not looking like a mouse, but it is actually a mouse. Okay, so done. So it is a mouse. And here now we'll talk about another plant as well. So do you remember this diagram from the NCERT? Do you remember this diagram from the NCERT? So whose diagram is better, mine or NCERT? Which one is better, my diagram or NCERT's diagram? I think my diagram is better, hai na? So basically a bell jar experiment, if you remember. So that is what we have to discuss first. what we have to discuss first? We have to talk about the bell jar experiment. Which experiment do we need to discuss? Bell jar experiment. So let's talk about it. So when you people talk about the bell jar experiment, right? When you people talk about the bell jar experiment, what they did? See, first of all, let's have a look, right? Let's have a look. So you can see that. So what they did, right? Basically, they revealed the, right? Joseph Priestley, he revealed the essential role of air in the growth of green plants. Okay? Essential role of air in the growth of green plant. He observed that a candle burning in a closed space, right, soon gets extinguished. This is what he observed, that a candle burning in a closed space soon gets extinguished, right beta? So in 1770 he performed the series of experiment and he revealed the essential role of air in the growth of basically green plants, right, and discovered oxygen in 1774. Years are also very important, right? So yes, in 1770 he performed a series of experiment that explained the role of, that explained the essential role of air in the growth of green plants. And in 1774, when bacha? In 1774, he discovered oxygen. He discovered oxygen. Is that clear? Is that clear? So now let's discuss that. So a candle burning in a closed space, that is in a bell jar, soon gets extinguished. This is what he observed. Similarly, a mouse in a closed jar will soon collapse. So he concluded that burning candle and breathing animals, they damage the air. Like here in this bell jar, what is going to happen after some time? After some time, you people are going to see, right, that this candle will soon get extinguished and the mouse will also collapse. So this is my collapsed mouse, which is looking like a mosquito, but trust me, it is a mouse. Okay. Now, when he conducted the same experiment, but now what has changed? He placed a plant there. He placed a plant there. So you will read mint plant is mentioned. If you will read this topic, so what will you find? That the mint plant is present. So some students, they ask me that, ma'am, why mint plant? Why mint plant? So beta, mint plant is a skyophyte. I am sure you do not know the meaning of skyophyte. It's a skyophyte. What's the meaning of skyophyte? It's a shade plant. What sort of plant it is? It's a shade plant, students. It's a shade plant. Right. So basically we are conducting this experiment in a bell jar. Where are we conducting this? Or where did that Joseph Priestley conducted the experiment? In a bell jar. So he kept a shade plant there. He kept a shade plant there. So it is a shade plant in that bell jar. Obviously, right, it is not demanding that I need this much of sunlight, this and that. In a shade, it will be able to grow there. So that's why mint plant was preferred. But here, right, this is not what we are going to observe. Now here, right, what is going to happen? Here, what is going to happen, bacha? Right, the mouse stayed alive and the candle continued to burn. Right, what happened here is that the mouse stayed alive. Mouse stayed alive and candle continued. Mouse stayed alive and the candle continued to burn. This is how this happened here, right? Right. Now, ma'am, what's that? So this is little extra. He placed a mint plant, right, which is a skyophyte. So what's the meaning of skyophyte? It's a shade plant, right? You don't have to be like, it needs proper sunlight, this and that. In shade, it can easily grow, right? So candle extinguished after some time, but literally he relit the candle inside the jar with the mint plant, candle burned again. So mouse also survived. So in NCERT, they have explained it very simply that when he kept a plant with it, right, the mouse stayed alive and the candle continued to burn. So what they concluded, this is what they concluded, right, bacha, that the plants, they restore the air that animals and burning candles remove. So this is basically the conclusion from this particular experiment. this is basically the conclusion from this particular experiment, and it is important that the plant restore the air that animals and burning candles remove. Is that clear, guys? So later, Lavoisier identified this as oxygen. So in 1774, I told you already. So this is as per NCERT. Obviously, we have to remember in 1774, he discovered oxygen, right? But basically, he discovered oxygen. So later, like he discovered this thing, na, he conducted this experiment. Bacha, right? So in 1774, he discovered the oxygen, but it was basically identified as oxygen later by Lavoisier. Now, the next experiment is Engelhousz. So JP done. Now let's talk about the JI. Is that clear? Now let's talk about the JI. Ready? So 1730 to 1799. So what he did, right? Let's discuss that. So first experiment done, second experiment. He basically repeated Priestley's experiment, but with sunlight versus darkness. Like what Priestley did, he kept this in bell jar, right? He kept that bell jar in the light, and then he observed. He placed the plant there. Now, when it comes to the Engelhousz, they basically did what? They repeated the experiment, right? He repeated the experiment, right? Engelhousz repeated the experiment, right, of this Joseph Priestley, but now he did what? Sunlight versus darkness. So, he showed sunlight is important for the plants to purify the air. This is important. Sunlight is essential. So in bright sunlight, small bubbles they formed around the green parts of the aquatic plant, and later which were identified as the oxygen. Is that clear? Later they were identified as oxygen. Is that clear? Yes. So Engelhousz showed that the sunlight is important for plant process, and that is going to purify the air fouled by burning candle or the breathing animal. So he took an aquatic plant and he showed that in bright sunlight, right, some bubbles will form, and that bubbles were nothing but that bubbles are oxygen. It was identified later, but in darkness no bubbles were there. So that is why, right, he proved that it is the sunlight, right? He proved that it is the sunlight which is important. So only the green parts release oxygen and light is important. So this is about his experiment, right? Done, beta? Okay. So, next experiment. Just 1 minute, beta. So, next experiment is of Julius von Sachs. So, anybody in the class who can tell me that experiment? Anybody here in the class who can tell me that experiment, that what he did? Yes? Done? Okay, so Julius von Sachs experiment, this is what we have to discuss. So, beta, in 1854, right? Basically, what's the part in 1854? Basically, what is there in 1854, right? What he just said, that obviously glucose, right? Glucose is made by green parts, by growing green plants. Yes or no? Yes, bache. And you know that glucose will be stored as starch, and this starch is stored in special structures, right? Special pigments are present, special structures are present, and today you know that we call that structures as chlorophyll, and that chloroplast, and that pigment as chlorophyll, if you remember, right? So he just provided the evidence that when plants grow, they make glucose, which is stored as starch, and green substance in plant, right, which is there in special bodies. Okay, they are present. Is that clear? So green part, right, it is a green part where the glucose will be formed. So his experiment was very simple, that is it. So glucose, it is made by growing green plants. Glucose is stored in the form of starch, and we know that this starch is stored in special structure, which we, right now we call it as chloroplast. So that special structures, chloroplast, they are having a green pigment, bacha, and today we call it as the chlorophyll. So this is what he mentioned. So it is the green part. It is the green part which is having this, you know, glucose, or where the this starch will be stored there. So this is it. That's what we have to remember. Okay, this is it. That's what we have to remember. So any doubts? Any doubts here? Chali, next experiment is of what? Next experiment is of T.W. Engelmann. Next experiment is of T.W. Engelmann. This is in 1882 and it is one of the most important experiments. Right, bacha? It is one of the most important experiments. Is that clear? Is that clear? Yes or no? So as per NCERT, it is 1843 to 1909. Right? If I go, because NCERT part we have to also cover. So this is what given in NCERT. So you can write it down, and even in the notes you people are going to get it. So T.W. Engelmann, so experiment is in 1882, but in NCERT this is this is what they have mentioned. So we need to know that. Now this experiment is very important experiment, beta, very important experiment. Okay, so this experiment is conducted, right? This experiment is conducted to find out which wavelength of light are most effective in photosynthesis, right? Which wavelength of light is more effective for photosynthesis, right, bacha? So this particular experiment has given us the first action spectrum as well, right? First action spectrum. From this particular experiment we got the first action spectrum. Don't worry, I will explain you what exactly is that action spectrum also, right? So keep it in your mind. Okay, keep it in your mind. So what is the material that they have used, beta? What is the material that they have used? Green algae, which is cladophora. Cladophora, green algae. So if I have to frame a question from this part, bache, so I can also ask you, I can also frame a question like that, that in which photosynthesis experiment, right, cladophora was used. In one way or another way, I can just make the question from this particular part. This is what I can directly ask you, right? So cladophora it is. So cladophora is which algae? Is it red, brown? So it is basically green algae. So yes, these are also the details, right? If you are if you genuinely want to score 330 or 340 marks in biology, you have to focus on such depth, such points as well, right? You have to know the content in detail. You cannot be like, I will attend somebody's, 1 hour ka session where they have committed that we have completed the photosynthesis and then we will be able to manage. No, beta, if you have completed your syllabus, if you just use a particular session for the quick revision, then this 1 hour session, 1.5 hour session or 2 hour session, 2.5 hour sessions can help you, right? Because you are already aware of that topic and you know that topic in detail. Right now, you just to brush up your concepts, you are using that. But if you don't know, right, if you don't know that topic in depth, you really need the proper sessions where NCERT in detail is covered. Otherwise, bache, if you think that by just attending a particular session of 1.5 hours, I will be able to solve at least 2 to 3 questions, you might be. But the point is, if there will be if paper will be, right, tricky, you won't be able to. You won't be able to. So you still have time. If you will go strategically, right, see, if you score 340 marks in biology, I'll give you very simple maths, 100 in physics, 100 in chemistry. You'll easily get what? You'll easily get what? 540 marks. 540 marks. So that though you can very easily get, na? That too you can very easily get. So this thing you have to maintain. This thing you have to maintain. It doesn't matter. It doesn't matter. The paper is going to be, see, ultimately paper will come from NCERT. So it doesn't matter whether it's difficult or it is easy. Ultimately, it will be from your syllabus. It will be from the NCERT. So biology, in biology, there are high chances that you can score maximum marks. Are you getting my point? In biology, there are high chances that you people can score maximum marks. So that is my suggestion, everybody, right? Please do not take it lightly, right? Try to target full house here so that at least you get 350 marks. So directly 550, which is a decent score. Some students, they ask, is it possible to take a jump? Yeah, if you are getting 300 marks in biology, 300 marks in your previous attempt means it is majorly from biology, then physics and chemistry. So try to boost now. Try to get 350 in the biology itself. Try to get 100 and 100 in the physics and chemistry. You will definitely get good marks. So it is all about the planning. It is all about your planning, right? So please start studying sincerely, okay? Start studying sincerely. So once I'll complete the most important units, right? Once I'll complete the most important units for you. Then I will take the full syllabus marathons as well. You don't worry about that. The full syllabus marathons then will also be there. Is that clear? Okay, so here in this experiment, green algae they have used, then aerobic bacteria, yeah, aerotectic bacteria, it needs oxygen. Aerobic bacteria or aerotectic. So it is attracted to oxygen. It is oxygen seeking bacteria. So say in some books they also use the word aerotectic bacteria, right? So basically it is aerobic bacteria, okay, attracted to oxygen. Then prism is required here, which is going to split the white light into its components. Is that clear? Into its components. So what is going to happen? What is going to happen? So Engelmann placed a filament of cladophora in a suspension of aerobic bacteria. Just for an example, let me show you. Just let's suppose here you have cladophora. You have cladophora. Here you are also having your aerobic bacteria. You are having, let's suppose, prism, okay? So when you will pass the white light through it, so you know that it will split into its component, right? Just trying to show you. It is going to split into what? Its components. Right now what they observed, just say, let's suppose if this is your cladophora, this is a filamentous green algae. So they observed that wherever red and blue light fall, right? This is what they observed, that wherever the red and the blue light fall. So this aerobic bacteria, it was basically around that region of this algae. Around that region of that algae, right? Just say, let's suppose this is a prism. They'll pass the white light through it. Now here in this experiment, they are having cladophora, they are having the aerobic bacteria. So what they observed, that wherever from this light, it will break into its components. That is what? That is your, one second. That is basically Vibgyor. I hope you all are well aware. Violet, indigo, blue, green, yellow, orange and red. So they observed that wherever it is the blue light or the red light, right, the aerobic bacteria, it started accumulating there, right? It was more there, right? This is what they observed. So Engelmann placed a filament of cladophora in a suspension. Cladophora here should be in italics because it is the scientific name, but yeah, you can do it. So in a suspension of aerobic bacteria, he then passed white light through a prism. He then passed white light through a prism, which will split it into different colors. That is your Vibgyor. So now the algal, right? Now the algal filament, it will be illuminated with the spectrum of light. Yes or no? So the algae carried out photosynthesis, obviously. It will release oxygen. So aerobic bacteria moved and clustered around the regions of algae that released more oxygen, because it is aerobic bacteria, it needs more oxygen, right? So that is the common sense. So what they observed, so they observed that they accumulated bacteria, they accumulated mostly in blue and red region, very few bacteria gathered in the green region. This is what they observed. Very important point. Very important point, bacha. This is what they observed. It's very, very important. Is that clear? It's very important. So what was the conclusion? Conclusion was the rate of photosynthesis is maximum in blue and red light. So that's why I told you that it is the first action spectrum, because it tells us that which light is more effective for photosynthesis. We will also discuss these terms in detail as per the NCERT sequence. So action spectrum means we will study two spectrums, absorption and action. Absorption spectrum will tell us which light will be absorbed maximum, and action spectrum will tell us in which light the photosynthesis is going to be maximum. So here, this is what you people will see here. So the rate of photosynthesis is maximum in blue as well as in the red light. The rate of photosynthesis is maximum in blue as well as in the red light. That's what you have to keep in your mind. And it is photosynthesis is least effective in the green light, right? And that is obvious, right? Plants, leaves, they are green in color. means they are not absorbing green color, they are reflecting green color. That's why in green color, right, it is least effective for photosynthesis. Just say for an example, if a structure is blue in color, it means it has absorbed all other colors, it is reflecting blue light, है ना? Blue light. So green means it has absorbed other lights, right? But it is reflecting what? Green. So that is why green is least effective for the photosynthesis. Is that clear? Is that clear? Because green light is mostly reflected by chlorophyll. So this is the first action spectrum of photosynthesis. If there is no doubt, then please let me know so that I can move to the next experiment. Sure. So this is the simple experiment. So always remember blue and red light, because these pointers are highly important, right beta? So in a bubble here also I am mentioning it. So blue and red light, most effective for photosynthesis, and out of blue and red it is going to be red. So this is going to be most effective for photosynthesis, and green light, it is least effective for photosynthesis. It is least effective for photosynthesis. Done, bacha? So that's what you have to remember. So next is before Van Niel, right? Next experiment, Cornelius van Niel, a very important experiment, because from this particular experiment, right, people will start focusing on this thing, that it is not always, right, that oxygen will release. If water is going to be used, then there will be oxygen. So we will discuss this particular experiment. So guys, please pay attention. So before Van Niel, people believe that oxygen released during photosynthesis is coming out from CO2. A very important experiment, a very important pointer, a very, very important pointer, that it is the oxygen, right? It is the oxygen. People used to believe this thing, that oxygen is coming from CO2, right? In the starting, I told you that it is the carbon dioxide which will be reduced, right? Reduced CO2 will give you the glucose, and I told you that it is the water which is going to be oxidized. It is the oxidizable compound, right? So if water is not used, there will be no oxygen. This is what I told you initially. But how we got to know that? Because people initially used to believe that it is the CO2 from where the oxygen is coming out. Is that clear? So Cornelius van Niel, a microbiologist, worked on photosynthetic bacteria and gave a revolutionary idea. It's a very, very, very important experiment. Ready for that? It's a very important experiment. Ready for that? Sure? Done? Okay. Right? So you can even write the scientist name, which is 1897 to 1985. So this is as per NCERT when you talk about the Cornelius van Niel, right? This is as per the NCERT. Okay, this is as per the NCERT. And before that, it was simple, that carbon dioxide and water is used for photosynthesis. So even the empirical empirical equation, it was also different, right, beta? So Cornelius van Niel, here you people can see also, right, microbiologist who studied purple and green sulfur bacteria. Again, it is a previous year question. Again, it is a previous year question. Is that clear? So Cornelius van Niel, right, studied the purple and green sulfur bacteria. That's a PYQ. So he observed, see, photosynthesis is a light dependent reaction. So hydrogen will come from a suitable donor which will reduce CO2 to carbohydrate. This is the point here. This is the point here. Is that clear? Is that clear? Yes or no? Is that clear? Yes or no? Sure? Yes? Is that clear? Okay, chaliye, let's discuss that. So basically, general equation 2H2A, CO2, CH2O, H2O plus 2A. Can you see that? Can you see that? जैसे even I told you that after his experiment, right, after his experiment, things they changed. After his experiment, the things they changed. Normally, what was the equation? Normally, this was the equation. CO2 plus water. Then you are having what? Right? You are having what? This is what we were having there. Right? CH2O. CH2O plus oxygen. So this is the general equation. General equation, because it was believed that it is from the CO2 from where the oxygen is coming out. It is the CO2 from where the oxygen is coming out. Is that clear? Is that clear? But now, this is general equation. Beta, it is 2H2A. It is 2H2A. A can be anything here, right? A can be anything here. So here in this equation, right, you will have the CH2 is there. The CH2 is showing what? It is showing the carbohydrate there also. It is showing the carbohydrate. Are you getting my point? It is showing what? It is showing the carbohydrate. So water is released and it is 2A. Why 2A? Because here we have 2H2A. If let's suppose here if I'm going to have oxygen, oxygen will release. If here I'm going to have this S, the S will release. That is basically we got to know from this particular, right, experiment. So in green plant, water is the hydrogen donor. So it will be oxidized to oxygen. So it depends which one is the oxidizable compound there. If it is water, oxygen will come out. If it is hydrogen sulfide, then sulfur will come out. It is like that. Is that clear? Is that clear? So in green plants, water is the oxidizable compound. It is the water which is the hydrogen donor. So it will be oxidized to oxygen, my dear students. And H2S is the donor in the sulfur bacteria. So it will produce sulfur or sulfate, not O2. So this is something, it is a milestone, beta. Basically, this is something which is very important. So before his experiment, this was the general equation. Carbon dioxide, water. The carbohydrates will be there, but they have simply mentioned oxygen. Why? Because at that time it was believed that from CO2 oxygen is coming out. But later what we saw, later after his experiment, so because he also studied what? He also studied the purple and green sulfur bacteria. So that's a PYQ. Right? That's a PYQ. That's a previous year question that you have to remember. So in green plants, it is the water, which is the hydrogen donor. Right? In sulfur bacteria, it is H2S, which is the hydrogen donor. So what came out? Sulfur or sulfate came out. So that's what we have to keep in our mind. So if there is any doubt, do let me know. Quick. If there is any doubt here, do let me know. Done? So photosynthesis is a light dependent redox reaction. That's the conclusion. So oxygen is released in photosynthesis. It will come out from water, not from CO2 beta. So here these are basically from my handwritten notes. Right? From my handwritten notes in a typed format, I'm presenting this in front of you so that you can have a better idea of the topic. Okay, so I believe this is clear. So different organisms, they use different hydrogen donors, right? So accordingly, they are going to release that. So basically, when we say the N-oxygenic photosynthesis, so this is what you have to remember. You cannot always say that photosynthesis is going to release oxygen only. So it is N-oxygenic photosynthesis also, right? N-oxygenic photosynthesis where there is no release of O2. No release of O2. So I'll give you one more example so that you can understand it in a better way. Like we talk about, when you talk about the bacteria, you remember we talk about the blue-green algae, which is also known as cyanobacteria. Blue-green algae, which is also known as cyanobacteria, if you people remember. Blue-green algae, cyanobacteria. So it is the first bacteria which basically releases oxygen. So it shows oxygenic photosynthesis. It shows oxygenic photosynthesis. Are you getting my point? It shows oxygenic photosynthesis. Blue-green algae or cyanobacteria, it shows oxygenic photosynthesis, right? This is the bacteria in which oxygen is getting released. Is that clear? Is that clear? Right? So this experiment is not given in the NCERT, but it is actually true that this idea of this Cornelius van Niel was later proved experimentally by Ruben, Hasid and Kamen. Ruben, Hasid and Kamen. That's why I said RHK. Ruben, Hasid and Kamen using, right, so what they did, they used the radioactive isotope O18 for their experiment. Otherwise, it is naturally, it is O16. But they use radioactive isotope O18. So when Niel's work laid the foundation of modern equation of photosynthesis, told you already. So what Ruben, Hasid and Kamen they did, they used chlorella, which is green algae. You know it. It is the unicellular green algae. What is chlorella? It is unicellular green algae. Is that clear? So they use this green algae for their experiment. They placed it in a water. That water was having radioactive heavy. It is radioactive as well as heavy isotope of oxygen. It is not just radioactive, it is heavy also. So radioactive heavy isotope of oxygen. So when bacha it was illuminated, oxygen released and it was tested with mass spectrometer. It was tested with mass spectrometer. So oxygen released contain isotope, right? Oxygen released from this because they placed the chlorella in that water where the radioactive isotope, right, where the radioactive isotope of oxygen was there. So obviously if chlorella is going to grow inside that water, basically chlorella is using that water, chlorella is basically showing oxygenic photosynthesis. So the oxygen that was released was having the radioactive, right, radioactive oxygen. It means it has used the water there, right? So with the mass spectrometer, this is what they shown. So oxygen released during photosynthesis comes from splitting of water, not from CO2. A very important point. A very important point. A very important point. Is that clear? Is that clear? So guys, quickly hit the like button and do subscribe our channel as well. Quick. Done? Sure? Sure? So next structure that we have to discuss is chloroplast, right? And then we will move to our next topic, right? So first let's study the chloroplast. But before that, do let me know if you guys are having any doubts. If you people are having any doubt, then do let me know. Sure? 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 So let's go ahead, let's discuss it further. So let's talk about the chloroplast, which is a semi-autonomous organelle. What sort of organelle it is? It's the double membrane bound. It's double membrane bound, semi-autonomous organelle. Who? Your chloroplast. So the chloroplast structure is very important to further understand this particular topic in detail. Is that clear? Is that clear? So see. So beta, when I'm saying semi-autonomous organelle, can you give me another example? Can you give me another example? Yes. I am saying we are going to discuss the structure of chloroplast. So chloroplast is what? Chloroplast is an example of plastid. Yes or no? Yes or no? So you know that, right? In the cell chapter also we discuss plastid. What do we discuss? We discuss plastid. So we have chloroplast, chromoplast, and we have leucoplast. Okay, and we have leucoplast. Let me write it. So plastid, where do you find it? You find it in plants or in euglenoids, right? In some euglenoids. So you have chloroplast, you have chromoplast, and you have leucoplast. So bacha, as the name is suggesting, chloroplast, chlorophyll it contains. It is having the green pigment chlorophyll. Then you have chromoplast. Chroma means color. So a plant is not just green in color, right? It is having different colors. Just fruits, right? Unripe fruits, they are green, and then they turn. When they ripe, right? When they get ripened, their color changes. So basically, one pigment, one plastid can get transformed into another as well. That is also something we discuss in the cell chapter. circular, and then leucoplast, which is colorless, and it is meant for what? So leucoplast, which is colorless, so it is meant for what? It is meant for the storage. It is meant for what? It is meant for the storage. So this is what you have to remember. So bache, initially there is a proplastid, which is the precursor, which is the beginner, right? Which is the precursor, which is the beginner, right? There is a proplastid, which is the precursor, which is the beginner. So this proplastid, this precursor, right, this proplastid, this precursor will form plastid, and these are the three different types of plastids here. These are the three different types of plastids here. These are the three different types of plastids here. Is that clear? Is that clear? So chloroplast, chromoplast, leucoplast is there, and this leucoplast is colorless, right? So leucoplast will have amyloplast, alluroplast, or elioplast. Is that clear? So this is having three types, bacha. Amyloplast is the one that will store the carbohydrates. You have alluroplast that is going to store the proteins, and then you have elioplast that is going to store the oil, right? Oil and fat. So this is what you need to know. This is important. So now our main discussion is for this chloroplast, which is a double membrane bound organelle. So here you people are going to see, you will have an outer membrane. There will be inner membrane. In between these two, bacha, we will have intermembrane space, which is also known as periplastidal space, right? What can we call it? Periplastidal space. Now, here in this particular chloroplast, this matrix is known as what? We call it as stroma. This matrix is known as what? It is known as stroma. Here what you people will see, you people are going to see circular DNA. You people are going to see circular DNA. You people are going to see the ribosome here. Which ribosome will be there? 70S ribosome will be there. Other than that, you are going to see the enzymes here required for dark reaction. Other essential, proteins, minerals, everything, other essential proteins, vitamins, everything will be there, and enzymes for dark reaction are also going to be there. Right, we'll discuss that photosynthesis is having two parts. Now, when you look at this particular structure, what this structure is, this is granum. This is granum, right? So, granum word. Granum is what? Granum is singular. And when you use the word grana, that is basically plural, right? So, here in between this, what do you have? You have intergranule thylakoid. You have intergranule thylakoid. So this intergranule thylakoid is also known as pretlamella. For thylakoid, you also use that word. You can also call it as stroma thylakoid. These are the names for this. These are the names for this, right? And here, moreover, starch grains are also there, right? And here you will see plastoglobules inside the stroma here. So I'll discuss what are these plastoglobules. We'll talk about it. Is that clear? We'll talk about it. Is that clear? So any doubts? Any doubts? Sure? Done, beta? Done? So plastoglobules are also there, which are lipid containing bodies, or you can say that fat globules present in plastid, especially in chloroplast. So starch grains and all, this is what we are also going to see. So the diagram part is also important. Let's discuss it. Okay. So starch grains are also there. Okay. Starch grains are also there. Is that clear? So that's what you need to know. So granum we have, right? Plastoglobules we have, enzymes we have, intergranule thylakoid. Now we have to discuss this particular portion. But before that, let me write it. The chloroplast, you know that semi-autonomous organelle. Why is it a semi-autonomous organelle? Because bache, it is having its own circular DNA. Circular DNA. It is having its own protein factory. It is having the 70S ribosome. Are you getting my point? So we also discuss the same thing in mitochondria. How many of you remember that? We have also discussed that in the mitochondria. So I believe you people remember this. So mitochondria and these plastidia, these chloroplasts are the semi-autonomous organelle, right? Because they are not totally dependent on nucleus for everything. They have their own circular DNA, right, which is having the information, and they have the protein synthesizing, that is ribosomes. They have protein synthesizing factory as well, and this is the 70S ribosome. So due to the presence of this circular DNA and circular DNA without histones and the 70S ribosomes, we see that in mitochondria as well as in chloroplast beta. So it is similar to what we study in the prokaryotes. So there is a endosymbiotic theory about the origin of this chloroplast and mitochondria. Right, endosymbiosis, yeah, endosymbiotic theory, that there was a cell, you know, a large size cell. This cell engulfed this small bacteria. This bacteria was able to, you know, do the photosynthesis. So it started living within it, right, as in a symbiotic relationship. So this bacteria or this has become the chloroplast in future, because this is having right, this circular DNA and the 70S ribosome. So that is also the case. Do read about it. So this is what we have. Now, when you talk about this particular structure here, right, please pay attention that this is what we made. This is what we made, which we called as granum. Which we called as granum. Which we call as granum. So this is granum. How that granum will form? How that granum will form? Bacha, this membrane bound structure here, right? Which one? Which one? This membrane bound structure here, this one that I'm talking, this is known as thylakoid. What is it? It is the thylakoid. This membrane bound structure, it is the thylakoid. So thylakoid, they'll pile over, right? They'll pile up, right? They are going to form the stacks. Just say, let's suppose you have the books, one book, second book, third book, fourth book. So you are just piling it up, right? So there is a stack of that books now. So same way here, you have thylakoids. What do you have? You have thylakoids here. So these thylakoids, right? These thylakoids are membrane bound structure. They'll pile over each other and they are forming the structure known as granum. They are forming the structure known as granum. So this is what you have to understand. Now here I would like to specify one more thing. Algal chloroplast means in algae, when you people talk about algae, algal chloroplast are agranal. Agranal means that is without grana. Yeah, without granum. So what does it mean, ma'am? See, in some, just say algae is a lower plant. So in algae, you will have thylakoids, but that thylakoids will not be able to pile up to form the grana. So yes, in case of algae, you people will also see, you people will also write that they are what? The chloroplast is agranal. It doesn't have the grana, right? Mostly this is what you see. So now here, I'm telling you clearly that this is thylakoid. What's that? This is thylakoid. So for thylakoid, you can also use the word lamella. For thylakoid, you can also use the word lamella. So it's a membrane bound structure, and in it, you will have the lumen. In it, you will have the lumen. What do you have? You will have the lumen here, right? So number of thylakoid, number of grana, it varies, students, right? Number of thylakoid as well as number of grana, both of them, it vary. Okay, so this is what you have. Now look at this. This is also a membrane bound structure, right? This is also a membrane bound structure, right? So here, right, just like plasma membrane, we discussed the plasma membrane also. Yes, people, we discussed the plasma membrane also. Right, we discussed the plasma membrane also, yes or no? You can see that. So here, right, it's a membrane bound structure, right? Please understand the diagram part. If diagram is clear, the topic is going to be very easy for you. So you people can see that here. You people can see that here. So this is the chloroplast. So chloroplast, it is having, right, imagine the diagram, right? See this. Let's suppose this is your chloroplast. What's that? This is your chloroplast, double membrane bound structure. That's what I told you. Yes, people, that's what I told you. So it's a chloroplast. It is a double membrane bound structure. So obviously this thylakoid is present in between. So you know that in the surrounding, the stroma is going to be there. So now I'm explaining the diagram first, because if diagram part is clear, right, then most of the things you are going to understand in the light reaction. So please pay attention. So obviously it's a membrane bound structure. So if it is something membrane bound inside that hollow place, so let's suppose this is your membrane bound structure. This is membrane. Inside this hollow, this empty space, you are calling it as lumen. What do you call it? You are calling it as lumen. So now when you look at this, this is your thylakoid membrane. This is your thylakoid membrane. This side is lumen, obviously. So surrounding me, definitely there is going to be stroma. So in this thylakoid membrane, you people are going to see there will be many protein complexes, and that's what we are going to read when we will talk about the light reaction. So let me specify here, thylakoid membrane is the, this is a very important point, people, please pay attention. Thylakoid membrane is the site of light reaction, which is also known as Hill reaction. Important previous year question. That's an important previous year question. So it's thylakoid membrane, which is, right, it is the thylakoid membrane, which is the site of light reaction. It is the thylakoid membrane, which is the site of light reaction. Okay. So here you have protein complexes in it, right? This is what we have to read. Now look at this part here. Now let's move to the next diagram. Look at this. This is what your stroma is. Thylakoids, they'll stack over each other, forming granum. Lumen is there. So this part I have already discussed with you all, right? Now I would like to specify some other diagrams here. Just allow me 1 minute, and that diagram again is very important to understand the topic. Just say, let's suppose this is your granum, okay? So in that granum, here you have the thylakoid. So this portion which is stacked, right? It is appressed membrane, appressed. Or can I not say that it is non-exposed part? It is not exposed. This part, end membrane, this is basically the non-appressed part. So if it is non-appressed, it is basically exposed. So that's also we need to know. Why? Because this part will help you to understand the position of different photosystems and everything. Jaise ye dekho, PS, like here, this is the non-appressed part. Non-appressed part means exposed part. This in between is the appressed region. Appressed region means this is basically non-exposed. It's not exposed here. So we'll tell you the importance of it as well. So that's it. This is the diagram. This is your NCERT diagram. So you people can see the starch granule here. Which ribosome do we have? 70S. What this S means? It is basically the Swedberg constant. It is basically the Swedberg constant. And here you can see stroma lamella, which is also known as fret lamella, which is also known as stroma thylakoid, Is that clear? Is that clear? So this is starch granule, and here this lipid droplet, for that you also use the word plastoglobules. So the diagram part need to be very clear. Okay, it should be very clear to you, and then we know we have stroma here. Understood? Understood? So this is important. Done? Beta, don't mind me saying that, right? First of all, you people start working on your spellings. Doubt means D-O-U-B-T. Why do you write D-O-U-G-H-T? Right? We will discuss everything related to PS1, PS2. Have patience. But doubt, the spelling for doubt is D-O-U-B-T. It is not D-O-U-G-H-T. Stop using the grammar part in your mobile phones, right? Stop that thing. Type the words. right? Try not to use the short forms. Try to write, try to type all the spellings. Okay. Chaliye. So chloroplast, in the chloroplast, how do we discuss? Hanji. So stroma, light reaction site, you are well aware of it. So it is the stroma in which dark reaction occurs. It is the stroma in which dark reaction occurs. In which dark reaction occurs. So this is the part that we have here. This is the part that we have here. So make sure you don't make the mistake. So we have membranous system, grana plus stroma lamella. Grana plus stroma lamella. This is the membranous system, obviously, beta, the one which is going to trap light energy, in which ATP and ADPH will form. Oxygen will also release in light reaction. We'll study that. And light driven, it's light reaction, which is also known as photochemical reaction. It is also known as Hill reaction. Now, the another part is fluid stroma. So it is basically enzymatic reaction in which your carbon dioxide will get reduced, right? In which sugar or starch will form, right? And it is not directly light driven, but dependent on products of light reaction. So this is a dark reaction. This is the carbon reaction. This is the biochemical phase. So this is what you have to reaction. Remember, light reaction is in grana, and to be more precise, in thylakoid membrane. It is the thylakoid membrane in which your light reaction will occur. It is the stroma in which your dark reaction will occur. This is important. Is that clear? So light reaction and dark reaction, these are the two parts which we have to discuss. But before that, let's talk about the alignment of the chloroplast. So alignment of chloroplast is also given in NCERT. It is in NCERT. They have just said, right? It is in NCERT. So do read the NCERT part. Do read the NCERT part. So in NCERT, they have mentioned this very clearly regarding what? Regarding the chloroplast beta, the alignment of chloroplast. So it's not like that your chloroplast is going to be having a fixed position there. No, it depends on the light intensity as well, right? Just like chloroplast, they can align itself inside the mesophyll cells for optimal light absorption, for the best suited light absorption. So what is mesophyll cells? Bacha, when you take the section of a leaf, right, you know that in the case of leaf, the ground substance is, ground tissue is what we call as mesophyll. The ground tissue is what we call as mesophyll. Is that clear? It's the ground tissue, what we call as mesophyll. That's what you have to keep in your mind. Is that clear? That's what you have to keep in your mind. Is that clear? So we have, see, if there is high light intensity, if light intensity is high, so you can see this is the incident light, this is the alignment of chloroplast. Can I not say that it is during high light intensity, chloroplasts are parallelly arranged to the incident light? So this is peristrop. This is the word, you can remember it, and even if you don't want to, totally up to you. Then you have low light intensity. When you have low light intensity, you have epistrophe. So here, right, the chloroplasts are aligned perpendicular to incident light. And here, when the light is moderate, so random distribution will be there. What will be there? Random distribution of chloroplast. It is going to be a random distribution of chloroplast. Is that clear? Is that clear? Sure? So it is written as well. Chali. Now let's talk about the pigments. Ready? Let's talk about the pigments. Ready? Another important question. So what is the meaning of photosynthetic pigments? I told you initially, they are the special proteins which can trap the light in light. So special molecule that can absorb, that can even transmit, that can even reflect the different colors or the wavelength of light in the visible spectrum. So what is the meaning of? Yes, what is the meaning of photosynthetic pigments, bacha? So photosynthetic pigments, right? Photosynthetic pigments are the special molecules. They are the special molecules that absorb, that transmit. They are the special molecules that absorb, that transmit, and that reflect. Once again, I'm repeating, that absorb, that transmit, and that reflect different colors of light in the visible spectrum. So pigments will appear in the color they reflect and use the color they absorb. So as I told you, chlorophyll, green in color, because it is reflecting the green color, it is absorbing rest of it. Right? It is reflecting the green color, it is absorbing rest of it. So you have to remember PAR as well, which is photosynthetically active radiation, right? Available for photosynthesis, 400 to 700 nanometer. This is important. So in visible spectrum, these pigments, they can absorb, transmit, and reflect the different wavelengths of light. That's what you have to understand. So we will talk about the chlorophylls here, carotenoids here, and the phycobilins here, right? So chlorophyll, chlorophyll, this is soluble in organic solvent, soluble in lipid, right? Water soluble pigments are there, right? So this is what we have to discuss. So types of photosynthetic pigment. Let's start with the chlorophylls, right? Let's start with the chlorophylls, right? So first of all, look at this particular structure. Look at this particular structure. If you can remember the formula, that is good. Otherwise, right, like I'm not expecting you to remember the structure and the formula, but yeah, there are certain basic points which need to be clear, okay? Okay? Sure? That's what you have to remember, okay? That's what you have to remember. This is what you have to remember for chlorophyll A and for chlorophyll B. What you have to remember, again, we'll discuss that. Done? This is basically having CH3, this is having CHO, 1 less, 2 less hydrogen, so 70, 106, and 4 this. That's what you have to keep in your mind. Is that clear? That's what you have to keep in your mind. The formula, if you can remember, it's good. Baki beta rest, it's all up to you. Okay? Chalo. Look at the structure of this pigment, that is chlorophyll. Where are we going to see that? In the thylakoid membrane. So please pay attention. So what is happening? This part, right, this part is actually the porphyrin head. This part is actually the phytol tail. Like when you look at the structure of the chlorophyll, right, it is also compared with the tadpole, right, with the tadpole. So this is going to be what? Once again, I'm repeating, this is going to be the head part. This is going to be the tail part. So for head, we are also using the word porphyrin head. Porphyrin head. And here, you people are having phytol tail as well. Porphyrin head is there and phytol tail is also there. Porphyrin head is there, phytol tail is also there. So you will see which one is the central metal ion. It is the magnesium, right? So this is another important PYQ. Magnesium is the central metal ion. What is magnesium? Magnesium is the central metal ion, an important PYQ. But when you people talk about the synthesis of chlorophyll, for synthesis of chlorophyll, iron is required. Right? For synthesis, for biosynthesis of chlorophyll, iron is required. Is that clear? For biosynthesis of chlorophyll, iron is required. That is what you have to keep in your mind. Okay, so for biosynthesis of chlorophyll, iron is required. That is what you people need to keep in your mind. So you can see porphyrin head, phytol tail is there. Now when you look at that, right, now when you look at that, porphyrin head and the phytol tail. Now we will discuss its structure a bit. Okay, we have to discuss its structure. So, okay, just 1 minute. So you can see that. So here you will be having a tetrapyrrole head, right? Tetrapyrrole. This is the porphyrin ring. Yeah, tetrapyrrole head is there. Yeah, tetrapyrrole structure is there. So tetra means 4. So just see 1, 2. This is 3. This is 4. These are that 4 tetrapyrrole rings. Central metal ion is already given. When you look at the chlorophyll A, so chlorophyll A, bache, this is very important. Chlorophyll A and B, I have mentioned the formula as well. We'll let you know why. So chlorophyll A is also the major photosynthetic pigment. What's that? It is also the major photosynthetic pigment, right? It is also known as universal pigment. Right, universal photosynthetic pigment. Now here in this case, in the case of chlorophyll, what you people will see that in the second pyrrole ring, here you will be having the methyl group. So CH3 is for chlorophyll A. And what is going to be different in the chlorophyll B? Chlorophyll B will have at the same place, it is going to have the aldehyde group. Another most important point. So chlorophyll A will have methyl group. Chlorophyll B will have aldehyde. That's what you need to keep in your mind. Is that clear? That's what you need to keep in your mind. Is that clear? So you can see here also, So this is basically the second tetrapyrrole ring here. So here also you can see rest everything is same. In chlorophyll B, you will have CHO. In chlorophyll A, you will have CH3. That is the methyl group. This is important. Okay, this is important. So you people can see here also, when it comes to the chlorophyll, it's a green colored pigment. Very important point is कि light is required for its synthesis. Light is required for its synthesis. If there will be no light, the chlorophyll will not get synthesized. A very important point again. So light is required for its synthesis, right? So here you can see soluble in, this point is written as well, a very important point, right? So it's soluble in organic solvents. So chlorophyll A, universal pigment in all oxygen liberating photosynthetic organism. So that is why it is also the major photosynthetic pigment. That is why it is also the major photosynthetic pigment, or you can say that you can also call it as the universal. So major photosynthetic pigment is there, universal pigment is there. So that's what you have to remember. So its color in chromatogram is going to be blue-green. We'll tell you what chromatography or chromatogram is. So it is blue-green. Another important PYQ formula we have already discussed. So it is having CH3 in its second pyrrole ring. So you have to be very specific here as well. So it is having CH3 in its second pyrrole ring. Now the next part is chlorophyll B. So it is the accessory pigment, right? Major pigment is A, accessory, shield pigment is chlorophyll B. It is in euglenoid, it is in green algae, it is in higher plants. So its color is yellowish green in chromatogram. Another important PYQ formula you people know. So it is having the aldehyde group. So this is another important question. Okay, so structure here looks like the tadpole. Already told you porphyrin head is there, which is water loving. Hydrophilic. Hydro is water, bacha. Philic means loving. So it is water loving. So it is having tetra, 4 pyrrole structure with magnesium at the center. With what is at the center? With magnesium as the center. So this is magnesium is the central metal ion here. That is also important. Magnesium is the central metal ion here, and iron is what? Iron is important for the biosynthesis. So phytol tail is hydrophobic. That is embedded in the lipid bilayer of the thylakoid membrane. Is that clear? So this one is hydrophobic. So biche, this is a very important thing, right? So chlorophyll will get synthesized from protochlorophyll. So it needs glycine and succinyl coenzyme. So glycine your simplest amino acid, glycine, the simplest amino acid. Other than that, succinyl coenzyme A is also there. So iron is very important. Fe, right? Very important here. Very important. So for biosynthesis, this is a very important point here. For biosynthesis, iron is required. That is iron. है ना? We call it as iron. Fe. For our central metal ion is magnesium. So glycine, succinyl coenzyme A. So this information is very important. So I have repeated it multiple times. This is what you have to keep in your mind, okay? Chaliye. Next pigment is carotenoids. They are also the accessory pigments. It is not just the chlorophyll which is present. Other pigments are also there, right? Other pigments are also there. So they are also lipid soluble. One should know which one is water soluble, which one is lipid soluble. So in green and non-green plant parts, you are going to find it. They absorb light energy. They'll transfer it to the chlorophyll A, which is there, which is the main pigment, right? And this carotenoid, they are very important, bacha, because they are going to protect your chlorophyll from the photooxidation, right? Light is not going to oxidize chlorophyll A, because this chlorophyll A, it is present in such a way that it is having, right, the accessory pigment around that, the shield pigments around that, which will include chlorophyll B, which will include your carotenoids, right? So that's what you have to remember. Right, so it is going to eat or destroy that nascent oxygen. It is going to basically convert that nascent oxygen into harmless O2. That's how it is going to protect the main pigment, the major pigment from the photooxidation, number one. Plus, the second important pointer here is that these shield pigments, right, because see, there is a certain wavelength of light, beta. Right, there is a certain wavelength of light. There is a certain wavelength of light that is going to absorb, right? That chlorophyll A is going to absorb, right? But these carotenoids, right? They can absorb different other wavelengths of light. So ultimately, they will also absorb light. They'll pass all the light to chlorophyll A, which is very important for the release of electron, and that is what we study in the light reaction. So that's the role of these pigments here. So this is basically the shield pigment. So here, yellow-orange pigment, they are on chromatogram. So they are even in chloroplast. They are even in chromoplast. So they are also present in universally, means this is not in eubacteria, otherwise they are all present, and it is the most stable pigment which doesn't need the light. So chemically, right, they are terpenes. So that's what you can remember. So here, if you will ask me the most important point, this is a very important point, that it is the most stable pigment. For this, you don't need the light for its synthesis. Very important, right? Yellow to yellow-orange in chromatogram, very important. Absorb light energy, transfer it to chlorophyll A, and protect chlorophyll A from photooxidation by squelching nascent oxygen, another important point, and they are lipid soluble. So from this, at least these points, right, they are PYQs, they are important as well, right? So if you genuinely done, so that's what you have to do. Done? Any doubts? Sure? Chaliye, next. Next is these carotenoids. So we will discuss, right? Here in carotenoids, we will discuss carotenes as well as xanthophylls. What are we going to discuss here? We are going to discuss carotenes as well as xanthophylls. What are we going to discuss here? Carotenes as well as xanthophylls here in the carotenoids. So carotenes, yellow-orange colored, purely hydrocarbon. They are yellow-orange. Jैसे here in carotenoids, overall I said that they are yellow to yellow-orange pigments. Yellow to yellow-orange pigments. What are they, bacha? Yellow to yellow orange pigments. Is that clear? Yellow to yellow orange pigments, number one. Number two, so yellow to yellow orange. Now we are discussing the types here. So when you talk about the carotenes, so they are yellow orange colored and xanthophylls are yellow colored. That's important. That's important. Is that clear? That's important. Is that clear? Yellow orange colored is carotene. Yellow colored is basically xanthophyll. So they are the derivatives of carotenes. Just in the case of in the autumn season, leaves, they turn yellowish. Why? Because of this lutein pigment. Lutein, right? Common in plants, responsible for autumn color of leaves. You have in the case of your dinoflagellates, your diatoms, right? Dinoflagellate, diatoxanthin they have, right? In the case of brown algae, do you remember? Brown algae is brown due to fucoxanthin. So that fucoxanthin is also the example of xanthophyll, or I would say carotenoids, right? So fucoxanthin. One second, bacha. So fucoxanthin. Fucoxanthin is the pigment due to which the brown algae, the Phaeophyceae, is brown in color. That is also an example here. So this is what you need to know about the pigments, right, students? So here these carotenoids, they can absorb the wavelength of different, they can absorb the light of different wavelength. So this is accessory pigment. I told you its role as well. So carotene, do you remember carotene, which is the example of this carotenoid? So I'll give you one very important example here, that is beta carotene. Beta carotene, which is basically a precursor of vitamin A, which is required for vitamin A. Your beetroot is also having this carotene pigment there. So this is important to understand. Please keep it in your mind. So the next part is what? Next part is basically the phycobilin, which is hot water soluble pigment. Important. Right? So here magnesium and phytol tail is absent. So if from the chlorophyll you remove the magnesium part, you remove the phytol tail, whatsoever is left, this is phycobilin. Just say phycocyanin, which will give blue color, phycoerythrin, red color. So in red algae we see both. Right? Just in red algae we discuss now. In red algae R phycoerythrin is present, that give red color, to red algae. Red algae. So it is found exclusively in blue-green algae and red algae. So this is also the accessory pigment. So this information is important, bache. This complete information is important. Please write down. Quick. Sure. Any doubts? Any doubts? Sure. Chali, let's move to the next part. So, structure of chlorophyll, this part is already done. So, chlorophyll A and B is also done. So, now, bacha, separation of photosynthetic pigment, another most important topic. Please underline this too, right? Please underline this too. This is again a very important topic, right? Most important topic it is. What is it, bacha? It is the separation of photosynthetic pigment that will be done by chromatography. And if I have to be very specific here, so what will I say? That this is the paper chromatography. It is the paper chromatography, that is basically going to separate, right? That is basically going to separate this. Is that clear? So what is this chromatography? What is the chromatography? Anybody? Done? Beta, in general means chromatography means chroma, color. So chromatography is the word that we have. Chromatography is the word that we have here. Now, what you have to understand here, like when you talk about the chromatography, when you people talk about the chromatography. So in the case of chromatography, basically what's going to happen? It is a method which is used in analytical chemistry, right? Which is used for separation of a components in a mixture. Used for separation of components in a mixture. So in a mixture, whatsoever components, right, whatsoever components are there, they will be separated. They will be separated with the help of this particular technique. Is that clear? They'll be separated and we can also identify that them. Is that clear? Is that clear? Done? So basically, right, so here you people will see, just I'm talking about the paper chromatography. I think in chemistry you guys have studied. So there is going to be a paper. That paper is going to be what? That paper is actually the stationary phase. In phases we discussed that, right? That paper is going to be the stationary phase, and there will be the solvent there, and that solvent is going to be the mobile phase, movable phase. So what you do, just say, let's suppose if you have to separate these different pigments, basically you can take the leaf, you can just with the help of mortar and pestle, you guys can make a mixture. You can put a drop here, right? Because we will see that this is only greenish in color, but then what is going to happen? The solvent will move, right? This paper is going to have that dot, right? Now what will happen? The solvent will move, right? So the solvent will move. This paper is basically showing us the stationary phase. So basically the principle is ki solubility of substance in the solvent and then absorption of substance onto the paper. First of all, solubility of the substance in the solvent, and the second principle is adsorption of substance onto the paper, right? So basically you will see when you will put a small spot of mixture on this part, right, paper in the solvent, this will not, without submerging, it will basically what will you do? The solvent will rise by the capillary action. So component, they will separate into different spots. Okay, so when this solvent will move by capillary action, just say, let's suppose here you have that dot, that mixture, the dot of that mixture. So basically its components will separate, right? It will not submerge, but rather its components will separate. And there are formulas that we use, beta. We check the retention factor, and that's how we calculate that. So it is used for separating the pigments as well, right? It is used for separating the pigments as well. So that is why when that pigments will get separated, you are going to see the colors. So that's what you say, that in chromatogram, that the chlorophyll A is bright here, blue-green in chromatogram. This is yellow-green in chromatogram. Xanthophylls are yellow. This carotenoids are yellow to yellow-orange, right? Carotenoids are yellow to yellow-orange. This is what we discussed, right? Carotenes rather, yeah, yellow to yellow-orange. So any doubts? Any doubts? So this is basically from the chemistry part, right? This is basically from the chemistry part. So that's what you have to keep in your mind. So if there is any doubt, please ask me. Okay, so carotenoids are yellow to yellow-orange. You can write it here. So they are yellow to yellow-orange. This one is going to be yellow. So that's what you have to keep in your mind. That's what you have to keep in your mind. Is that clear? That is what you have to keep in your mind. Done? Done? Chaliye, next is Visible light में you know that wavelength range is basically जैसे photosynthetically photosynthetically active radiation 400 to 700 nanometer. So here you can see the wavelength range is 390 to 760 nanometer, or in angstrom also you can see. So special light regions में we have red light. So red light above 700 nanometer beta you will call it as far red, and below this 390 nanometer is going to be ultraviolet. Above, beyond red, it is going to be your 760 nanometer. So this part is clear. This part is clear. You can even take a note of that. Quick, everybody, you can even take a note of that. Quick. Yes. Okay. So red light will have wavelength range. Done? So bacha, red light, you can say that red light will be somewhere in between 620 to 700 nanometer, right? 700 nanometer. So when it is above 700 nanometer, it is going to be far red. Is that clear? So far red will be somewhere around 700 to 750 nanometer, right? 700 to 750 nanometer will be far red beta. So when it is beyond the 760 nanometer, this is infrared, because this is beyond red. Is that clear? Is that clear? So you people can see violet, right? Vibgyor basically, violet, indigo, right? Blue, green, yellow, orange, orange, red and red. So one thing that you have to understand, already in, right, that T.W. Engelmann's experiment I told you, chlorophyll will basically show maximum photosynthesis in blue and red light. So that's the beauty of having other pigments also. Just that's the beauty of having the carotenoids also, because when you people will see the carotenoids, what you people are going to see, right, that carotenoids, they will show the absorption in different wavelength. Okay? Done? Yes? Quick, tell me. Quick, everyone. Okay, so if I talk about the carotenoids, they say, you know, carotenoids, they will not absorb the red light. Do you know that? So carotenoids, they absorb somewhere in, right? Somewhere, like if you will ask me the maximum region in which they absorb, they are going to absorb in this particular region, right? They are going to absorb in this particular part of the, this particular range, they will absorb, right? Somewhere in 400 to 500, and maximum will be there in this range. So they absorb blue or blue-green regions, right? So normally you will say 400 to 500 nanometer. In this particular range of light, they'll absorb, and the maximum absorption by carotenoids is in this particular range. So blue, right, so they will absorb. So you can see 400. So you can see somewhere around in blue or blue-green range. This is in this particular range they absorb. So that's the beauty of having different pigments, because let's suppose if chlorophyll A, B, they are absorbing blue and red light. So there are other pigments also which can absorb other different wavelengths of light. Ultimately they will pass it to the chlorophyll A, which is the main pigment, which will then release the electrons. So that's what we have to understand. I hope this part is clear. Now see some general points, bacha. Some general points. Dekho, leaves, they appear green. Why? Because they do not, the chlorophylls, they do not absorb green light, rather they reflect it. They do not absorb green light, rather they reflect it. Blue and red regions of spectrum, they are the most effective for photosynthesis. Told you already, T.W. Engelmann's experiment. When it comes to the blue light, you know that red light, its wavelength is more. Blue light, its wavelength is less. So blue light, it carries more energy. Red light, it carries less energy, but it is most efficient in the photosynthesis. Is that clear? Is that clear? And when it comes to the green light, because leaves, they are going to reflect it, they are least effective. That is why if under a tree canopy, mostly green light will pass through it, right? Green light will pass through that tree canopy. So therefore, plants which are growing under that will show very low photosynthetic rate. If there is a large tree, right, under that, if there are few, other trees are, other small plants are growing there, their photosynthetic rate is less. Why? Because they are getting the green light. So this is very important. So your blue light will carry more energy, and you know that its wavelength is less. Red light is having more wavelength, less energy. Chaliye, now these graphs, they are very important. So next topic that we have to discuss is absorption spectrum, beta, as the name is saying, right? Absorption. So it is basically the curve that is showing wavelength of light absorbed by the pigment. What is this, bacha? It is basically the curve that is showing the wavelength of light absorbed by pigment. It is basically the curve which is showing wavelength of light absorbed by the pigment. A spectrophotometer will help in its absorption. So this one will show the peaks here. So how can you, simply understand that? So you can see that this is basically showing absorbance of light by chloroplast pigment. So you know the colors. Chlorophyll A, bluish green. So chlorophyll A. So here, beta, you are having the blue region. Here you are having the red region. So you people can clearly see this part, right? You people can clearly see this part, right? This graph is very important, beta. So blue region, red region is there. Chlorophyll A, carotenoids, and chlorophyll B is given. Chlorophyll A, carotenoids, and chlorophyll B is given. So first of all, if I will talk about the chlorophyll A, dekho, from this curve, what are we able to depict? Chlorophyll A show absorption in blue and red light. है ना? It absorbs more blue than red. Number one. Chlorophyll B again shows absorption in blue and red light. For blue and red, I'm using capital B and R. It shows maximum absorption. In simple words, absorb more blue than red. है ना? Absorbs more blue than red. Absorb more blue than red. Now, the next part is what? So chlorophyll A is there. Chlorophyll B is there. Now the third part that you have to see. Look at this chlorophyll B. Look at this chlorophyll A. So can I not say that chlorophyll B absorb more blue light than chlorophyll A? Can I not say that? Obviously, chlorophyll B absorbs more blue light than chlorophyll A, and chlorophyll A absorb more red light than chlorophyll B, isn't it? Look at this. Whatsoever this curve is showing, that is what I am writing here. Whatsoever this curve is showing, that's what I'm writing here. And when you people talk about the carotenoids, they are showing absorption in, now you know the reason, showing absorption in blue light only. In blue light mainly. है ना? So I told you 400 to 500 nanometer and 450 to 480 is that maximum range. 480 to 450 to 480 is the maximum range. Is that clear? So chlorophyll A and B strong, blue and red light more strongly. Carotenes absorb, yeah, carotenoids absorb blue light, bacha. So red light is most effective, but blue light is also absorbed more efficiently. Blue light carries more energy, absorbed more efficiently. But out of blue and red, which one is most effective for out of blue and red, which one is most effective for the photosynthesis? Then you know that we have to mark the answer as red. So, bacha, these points are important. Please do not skip them, right? Please do not skip them. Is that clear? Do not skip them. Is that clear? Is that clear? Sure? Yes? Done, bacha? So, absorption spectrum will tell us ultimately that it is the blue and red light which will be absorbed which will be absorbed mainly, strongly, and already you know chlorophyll A, B, what are they going to absorb. Now, when it comes to the action spectrum, it will tell us the rate of photosynthesis at different wavelengths. And here also we know that, bacha, it is the blue and red regions which are more effective. So you can see here as well, right? In blue region, red region will show the maximum rate, and here you can see this is showing us the absorption as well as the action. Absorption as well as the action. So blue light, one second. So blue light, red light. Blue light is absorbed more, look at the rate. Red light comparatively absorbed less, but look at the rate. So blue and red light, they are the more efficient for that, most efficient for that. So that's what you have to understand. These are the diagrams from the NCERT. So this one is showing the absorption. This one is showing the action. This one is showing both. Right, so you have to be clear. So rate of photosynthesis measured by oxygen release, absorption of light, chloroplast pigment. So these are the graphs which are important, and they are also the previous year questions. Am I clear? Yes? Yes? Done? Chali. So before starting the mechanism of photosynthesis, right, two more pointers that I would like to discuss here, right? Please pay attention, bacha. So quantum yield and quantum requirement. Next is what? Quantum yield and quantum requirement. So what does it mean? Quantum yield and quantum requirement. So when I'm talking about the quantum yield, number of oxygen molecules evolved per quantum of light. Per quantum of light, how much it is, how much oxygen molecules it is making. So quantum yield is 1 by 8. Quantum yield. Now, when it comes to the requirement, basically number of light quanta which are required for the release of one oxygen molecule, it's 8. So now you can understand, basically 8 quantas are required for one oxygen molecule, for the evolution of one oxygen molecule. So definitely when we have to check the quantum yield, what are we going to do? It will be 1 by 8. It will be 1 by 8, right, bacha? So let's start the next two important topics. Let's start the next main important topic, that is the light reaction. So if, right, beta, chalo, let's discuss that, okay? Let's start. Done, beta? So, mechanism of photosynthesis, that's what we have to, right? Mechanism of photosynthesis, that's what we have to discuss now. So, when you talk about the mechanism of photosynthesis, there are two types of reaction, beta. Light reaction is there, dark reaction is there. Light reaction is there, dark reaction is there. It is also the photochemical reaction. It is also the Hill reaction. And when you are talking about this dark reaction, which is also known as biosynthetic phase, yeah, biochemical phase it is. Right, beta? And here, this is basically the word dark here. The word dark here and is the misnomer term. What is the meaning of bache misnomer term? So, misnomer term means it doesn't have the same meaning what it sounds. Like, for an example, right, I'm staying at a place, let's suppose I'm staying somewhere, right? It is not in China, it is in India, but outside my stay, there is a hotel, right? That is the China hotel. So it doesn't mean that that hotel is in China or it has to do something with China. So it's like China hotel, okay? So that is not like, if it is China hotel, it doesn't mean it is in China. Yeah, it is having some connection with China like this. So misnomer term, dark reaction, it doesn't mean that this reaction will take place in darkness. Right? It is like, you can say that it is light independent reaction. It doesn't need light directly. It is light independent. For light reaction, you need the light first, right? The light will be absorbed by the pigment molecules. That is how you have to connect here. But this is what? This is light independent reaction. What's that? Light independent reaction. So basically, it doesn't need the light directly. It doesn't need the light directly. It doesn't need the light directly, but is dependent on the products of light reaction. But is dependent on the products of light reaction. That is ATP, NADPH. So that's what you have to understand here, right? So that's what you have to understand here. So light reaction, where will it occur? It will be there in thylakoid membrane. Where your dark reaction will occur? Dark reaction, you know it very well. It is going to be there in stroma. It is going to be there in stroma. Is that clear? So it includes the light absorption, water splitting, oxygen release, ATP, NADPH formation. So one by one we will discuss it, but photosystems are required for the light reaction. So next important and the most important topic from this particular, right, from this particular unit is this part. This photosystems, they are the most important, right? They are the most important, they are amongst the most important topics of plant physiology, photosystems, okay? Photosystems, they are required. So beta, before photosystems, again, it is not given in NCERT, but it is important to understand the concept here. So photosystems are required for the light reaction. This is number one point. Second is how we got to know about the photosystems. That is what we have to discuss now. So we will talk about the Emerson effect and the red drop effect. Okay. Done. Okay. Sure. So Emerson and Arnold, they worked on chlorella. Already told you what is chlorella. It is the unicellular green algae. It is the unicellular green algae. So they gave the concept of two photosystems, right? So basically, what is the thing? See, they studied that, they gave some, effects and all. So firstly, I'll individually explain you red drop effect and the immersion enhancement effect. Red drop effect is like when you give chloroplast, the light, above 680 nanometer. As I told you that 662, this 700 or 680 will be in the red region. If it will go above 700. It is the far red region. Matlab above 700 will be far red region, right? Usually this is what you can understand. So far red light is above 680. या 700 nanometer या ये देखो जैसे greater than 680 usually around 700 nanometer. Already told you about the lights. So the quantum yield of the photosynthesis, that is oxygen evolved per photon, will drop sharply. Matlab they are saying if I am having a chloroplast and I'm not giving that chloroplast the complete light, I'm just giving it the light, far red light, right, beyond above 680 nanometer, usually around 700 nanometer when I will give it. So I'll see that there is a drop in the oxygen evolution. So this sudden fall in photosynthetic efficiency is the red drop effect. This is what they observed while studying, while conducting an experiment. This is what they observed. Ki when they are giving the chloroplast the light wavelength above 680 nanometer, around 700 nanometer, there is a drop in the oxygen evolution per photon absorbed. So why? Basically, in 1957, Emerson observed that if red light around 680 and far red light around 700 is given together, right, far red light above 7, done beta, when red light or far red light above 680 nanometer, it should be, let me rectify that. Hanji. So they were given together. Red light around this. 1 second, beta. Acha, red light around this is written. Okay, beta, it is right the way it is written. Ki if it is around 680, if it is above 680, or you can say somewhere around 700 nanometer. Somewhere around 700 nanometer, when they were given together, the rate of photosynthesis, it increased. The rate of photosynthesis, it increased. So red light around 680 nanometer, far red light above 680 nanometer, they were given together. The rate of photosynthesis was greater than some of their effects. So this is basically the immersion enhancement effect. Why is it taking place? After this, we got to know that there are two photosystems. There are two systems in the thylakoid membrane which are having pigments which are working like that. That's what we have to discuss. So two photosystems, they work cooperatively in light reaction. So red drop effect revealed a fall in efficiency with only far red light. So that's why red drop effect may what we notice that if we are giving it around 700 nanometer, the drop was there. Why? Because in photosynthesis, two photosystems are working together. Two photosystems are working together. So immersion enhancement effect is explained that there is photosystem 1 and 2. They are together required for the maximum efficiency. So this led to the discovery of your PS1 and PS2. This led to the discovery of your PS1 and PS2. Now, there are certain pointers which needs to be very clear. Beta, if we will complete this particular portion, then you will find the rest of the photosynthesis very, very, very easy. So please pay attention, okay? Please pay attention. Just see, you have photosystem 2 and photosystem 1, right? Obviously, usually what do we write? We write it like this, PS1 and PS2. We write it like this, PS1 and PS2. That's how we write. That's how we write. PS1, PS2, matlab 1, 2, 3, 4, that's how we write. Beta, this number here is as per order of their discovery. Order of their discovery, not as per their function. Not as per their function. That's what I'm trying to tell you. That's what I'm trying to tell you. That PS1 and PS2, what's the word that we have? PS1 and PS2. So it's not like if it is PS1, it is the first one which is going to do a task here in light reaction. No, it is the PS2. So basically, electrons move from PS2 to PS1. So the name is given as per the order of their discovery. Is that clear? As per the order of their discovery, the name is given. The name is not as per their function. This is the first thing. So immersion enhancement effect told us that because when they were giving the light above 680 nanometer, usually around 700 nanometer, the photosynthesis rate was low. But when they gave both 680 as well as 700, they are like photosynthesis is maximum. Means there are two systems which are working together, which are working in a cooperation. There are two systems which are working together, which are working in a cooperation, and that two systems are responsible for that. That two systems are responsible for that here, right? So PS1, PS2 is there. So let's understand what exactly is happening here. So basically your PS2, right, and PS1, here the electrons will move like that. So PS2, we also write P680. Why? Because it shows maximum absorption at 680 nanometer. PS1, you write it as P700 because it shows maximum absorption at, yeah, peak at 680 nanometer, maximum absorption at 700 nanometer. Is that clear? At 700 nanometer. Is that clear? Any doubt? Yes? Yes? Okay? Chali. So let's understand these photosystems. There are more points related to PS1 and PS2, but these were the basics. So imagine, just say, let's suppose in photosystems, what do you have? In photosystems, you people will see there are two things. One is reaction center. Second is light harvesting complex. LHC, which is also known as antennal complex, yeah, antenna complex. Right? Antenna complex or the light harvesting complex. So this is what you have. So you have reaction center, you have light harvesting complex or the antenna complex. So how are we going to understand that? So in a photosystem, in the reaction center, this is, let's suppose you have reaction center. So in reaction center, you are always going to find chlorophyll A because it is the major photosynthetic pigment. And now this reaction center will be covered by, right, it will be having so many, right, 100 to 200 other pigment molecules as well, right? It will be having other pigment molecules as well. Please pay attention, bacha. Other pigment molecules as well. So they are making the light harvesting complex or the antenna complex. It is making what? It is making the light harvesting complex or antenna complex. Light harvesting complex or antenna complex. Is that clear? Antenna complex. Is that clear? Is that clear? Done, beta? So what is happening? Just as sunlight will be absorbed by the reaction center. Sunlight will be absorbed by the reaction center. Then what's going to happen? Even these pigment molecules, because here what do you have? You'll see chlorophyll A, you'll see different carotenoids, you'll even see the phycobilins. So obviously, beta, they are going to have what? They are going to have what? They will absorb the different wavelengths of light. So it's not like that in a photosystem only reaction center will absorb. Yeah, only light harvesting complex will absorb. No, light harvesting complex, it is having these accessory pigments, shield pigments, which will make sure to protect or which are basically protecting your chlorophyll A. Is that clear? Which are basically protecting this chlorophyll A. That's the point that you have to remember, okay? That's the point that you have to remember. So these pigments are also absorbing the light and ultimately they will also pass it to the chlorophyll A. So that is how this photosystem works, and ultimately even after absorbing sunlight and getting sunlight from this, because it needs more energy to release its electron, it is going to release the electron, and then after that we will discuss the electron transport system here. We will discuss the Z scheme here, which we also call as non-cyclic photophosphorylation, which is basically the part of your light reaction. So it is very important for us to understand the photosystems first because it is very important. Their location is also very important. Now you will understand why that appressed and non-appressed region was required there. So every photosystem has a reaction center, and here you will have chlorophyll A. Accessory pigments will be there in antenna segment or light harvesting complex. So its function is to absorb light energy, pass it to the reaction center. So in PS1 beta, in both the reaction centers, chlorophyll A is there. So in PS1, peak is at 700 nanometer. So that's how you write it. In PS2, the peak is at 680 nanometer. So that's how you write it. Is that clear? That's how you write it. Is that clear? Is that clear? Is that clear? Now, when you talk about the location there, so process will start from PS2, right? So PS2, PS1 we have. So PS2, where will you find it? It's very important. It is in the expressed part, bache. Where do you find it? So where do you find it? You will find it in the appressed part. So appressed part means it is that non-exposed part which we have discussed earlier. So PS2 is there. PS1 is in the non-appressed part of grana. Plus it is also in the stroma thylakoid. It is not just in grana, it is in the stroma thylakoid as well. So grana thylakoid overall will have both, but stroma thylakoid will have only PS1. So it is present in the appressed part of granal thylakoid, not in stroma lamella, ya fret lamella, ya stroma thylakoid. Is that clear? Done, bache? PS1. So position, location is important. Already told you this is the appressed part. So appressed part, non-exposed part is having PS2. So here, this exposed part is having PS1. Even this is here also, PS1 is there. So in stroma lamella, you will find only PS1. Only PS1. This information is important. Kindly process it. Quick. This information is important. Quick. Done? Okay? Sure? So PS2 in appressed region, PS1 in non-appressed region, right? So this is non-appressed, this is non-appressed, right? Here it is the appressed region, here this is the stroma lamella. So in stroma lamella you will only find PS1. So PS1 is in reaction center, right? So its reaction center will show peak at 700 nanometer and its reaction center will show peak there, right, bache? So light harvesting complex, light harvesting pigments are present there. So the difference also I have mentioned, bacha. So PS1 is located at non-appressed part of grana thylakoid and stroma. Its reaction center is P700, but it is having reducing nature. Now we will understand the light reaction, then you will understand it in a better way. So it participates in both type of phosphorylation. This one will participate only in non-cyclic photophosphorylation. It is having oxidizing nature and it is only part in the appressed part. So this is very important, right? This is very important. So do keep it in your mind. Do keep it in your mind. Is that clear? It is very important. Do keep it in your mind. Done? Sure? Chaliye, so let's start the light reaction. If light reaction is clear, and after light reaction, I will also give you the break. So today I told you it's Sunday, right? We don't have any class. So today we have to complete the plant physiology in detail, beta. So what are we going to discuss? We are going to discuss light reaction. Now here in light reaction, right, we also talk about the non-cyclic photophosphorylation, and we will also talk about the cyclic photophosphorylation. So I'll break this topic first. Then we will talk about the light reaction or the Z scheme. So, bache, first word is phosphorylation. What is the first word here? Phosphorylation. What is the meaning of phosphorylation? Addition of phosphate. Addition of phosphate here. Okay, just say you have ADP. In ADP, we are adding inorganic phosphate. What will form? ATP will form here. ATP will form here. So, addition of phosphate, right? So, here, what is going to happen? The inorganic phosphate will get added in ATP. ATP will form. Now, the word that we have is photophosphorylation. Like here and in next chapter, you will discuss about 3 types of phosphorylations, right? 3 types of phosphorylations. Just we will also talk about photophosphorylation, substrate level phosphorylation, oxidative phosphorylation. So now here it is the photophosphorylation. So here you are talking about the light. What are you talking? You are talking about the light. In the presence of light, there is the addition of phosphate. That is what we discuss here in the light reaction. In the presence of light, there is the addition of phosphate, number 1. Now, non-cyclic. Whenever any process is non-cyclic, what is going to happen there? Just something is moving from A to B to C to D back to A. This is cyclic, but it is non-cyclic. Let's suppose in the presence of light, there is the addition of phosphate. So, light absorption will be there by pigments. Electron will release. Electron will pass from one pigment to another. Like this is what we have to discuss here. So, A, B, C, D, like this, non-cyclic, right? If it will be cyclic, let's suppose a process will start from A, then to B, then to C, back to A. This is the cyclic phosphorylation. So, this non-cyclic phosphorylation here, it is also the Z scheme, bacha, right? It is also the Z scheme, and here both PS1 or I would say PS2 and PS1 are involved. Both are involved here. Here only PS1. This will give you ATP, NADPH, that is nicotinamide adenine dinucleotide phosphate, and oxygen. From here, you will only ATP. So, this is the topic that we have to discuss next. Only ATP. Is that clear? Is that clear? Sure? Sure? Done. So, phosphorylation, addition of phosphate, right, in the presence of light is photophosphorylation. So, non-cyclic and cyclic photophosphorylation is there. Z scheme, both are involved. So, this is what we need to understand next. Any doubt? Any doubt? Sure? Are you sure? Chaliye. So, you can see this is the thylakoid membrane. Beta, here you have lumen. Obviously, in its surrounding, you have stroma. In the starting part, this is what we have discussed, right? This is what we have discussed in the starting as well. So, please keep it in your mind. So, what is happening? So, you will see overall, this is the Z scheme that we have to understand. I will, before starting this, I will also talk about the redox potential. So, here you will see electrons move from, that's what you have to keep in your mind, PS2 to PS1. Electrons move from a direct MCQ, beta. It's a direct MCQ. So, electrons, they will move from PS2 to PS1. Electrons, they will move from PS2 to PS1. That's what you have to keep in your mind. Now, in the thylakoid membrane, you will find there will be PS2, that is photosystem 2. Other than that, you are also going to see some other, right, electron proton carriers, plastoquinone, you will see cytochrome B6F complex, plastocyanins you people are going to see. First of all, we'll also talk about pheophytin, right beta, we'll talk about, right, pheophytins will be there, like one by one we will discuss it, So, plastoquinone, cytochrome, plastocyanin, PS1. So, this is the arrangement, that we have to see. This is basically the arrangement that we have to see. जैसे PS2 will exude the electron that will be accepted by your pheophytin. Pheophytin, which is the primary electron acceptor, will pass it to the plastoquinone, which is referred to as PQ. So in higher studies, so you will discuss like this. Special quinones are there. There is a plastoquinone pool. Cytochrome complexes will be there. So all that details we have to see. But ultimately, we have to pass the electron. So where this arrangement is. So these diagrams. Why am I showing you these diagrams here? Because here I want to show you, right, that how in that, just say we have discussed the diagram, bacha. In the chloroplast, we know that we have the granum. We have the granum. So we are talking about its appressed or non-appressed part. We are talking about these thylakoid membrane here. We are basically talking about the type of electron or the hydrogen carrier present there, which are basically conducting this particular system, which are basically showing this Z scheme to us. And here in Z scheme, the electron transport is there as per the redox potential. So all these electrons carrier or some are proton carriers also. So why do we call it as Z scheme? Because as per redox potential, they are arranged in a zigzag manner. That is why. So that is what we have to discuss next. So after understanding the redox potential thing, we will go ahead and we will first remember the sequence here. Right. We will first remember the sequence here. Right. That which one is placed like a sequence of these electron carriers, and then we'll discuss it next. So redox potential is our topic. Ready? Redox potential is our topic. Ready? Ready? Done, beta? So redox potential, also known as oxidation reduction potential, isn't it? We can also call it as oxidation reduction potential, isn't it? Isn't it? What's that? It is also the oxidation reduction potential. So it is the tendency. It is basically what? It is the tendency of a substance to gain or lose electrons. It is the tendency of a substance to gain or lose electrons. Is that clear? It is the tendency of a substance to gain or lose electron in a chemical reaction, or I would say it is a measure of that tendency of a substance. Is that clear? Is that clear? Okay. Done, beta? Okay, so oxidation, we know what oxidation is. Oxidation means loss of electrons. Reduction means gain of electrons, isn't it? So please understand that, because the same thing is required for the next chapter also. So oxidation means the loss of electrons here. The reduction means the gain of electrons here. So that's what you have to keep in your mind, right? So loss and the gain of a reaction. So if any substance is having, more, or I would say firstly higher, right? This is the language that you have to understand. Higher or more positive redox potential. Higher or more positive redox potential. Higher or more positive redox potential. So it will accept electrons quickly. है ना? And if it is having lower or less positive redox potential, it will lose electrons. Any doubts? Any doubts? Done? So if it is having higher or more positive potential, it will accept or it will lose electron. If it is having lower or less positive, or I would say more negative, है ना? Less positive means more negative redox potential, it will lose the electron or donate the electron. So this is what you have to remember. Okay? So let's suppose here you have A, here you have B, right? There is one entity A, there is another entity B. I am saying that B is having more. B is a redox potential. It is having more positive redox potential. So as per that, electrons will pass from A to B. Electrons will pass from A to B means when I am saying that out of A and B. So here out of A and B, B is having higher or more positive. B is having higher redox potential. So obviously electrons pass from A to B, yes or no? Electrons will pass from A to B, yes or no? Definitely. They will pass from A to B. That's how the things are. That's how the things are. Now, this is basically what? This type of transport. Can I not say that it is downhill? This is the downhill, na? Matlab, ideally this should happen. This is beta downhill transport. This is downhill transport. जैसे for an example, when you talk about the osmosis, then we say that there is a solution A, there is a solution B. Solution A is having more solvent. Solution B is having less solvent. It is having less solvent. So water will move from, right? Water will move from a solution having higher solvent to a solution having lower solvent through semi-permeable membrane. So in this case, this is actually downhill transport because the flow is along the concentration gradient, down the concentration gradient, because ideally this should happen. One is having more solvent, another is having less. That's how the things are. So here also don't be like, ki man, electrons are passing from this less positive redox potential to more, then how can you say that downhill? But here the context is different. Here you are talking about the measure of tendency of a substance to accept or gain electron. So out of this A and B, it is the B which is having more positive redox potential. It means this B is having more, its measure or its tendency to accept electron is more than A. So obviously this should be the downhill transport. But if this is going to be the case and I am saying electrons, right, B is having more redox potential, yeah, higher redox potential, but still if electrons, they pass from B to A, if still they pass from B to A, then you will call it as uphill. That is, it is against what should be done, right? It is against that ideal, it is against that, right beta, right? Sure, sure. Okay, done. Chaliye, very good. So this is required here, right? So you can see PS2 and all. So now before going forward, so it is in thylakoid membrane, everything is there. So their placement as per the redox potential is in zigzag manner. So now you have to remember something. What you have to remember? You have to remember photosystem 2. Then you have to remember pheophytin. We'll show you the arrangement. Then you have to remember plastoquinone, PQ. And then here you have to remember PQH2 as well. In this way you have to remember. So plastoquinone, and this one is plasto hydroquinone, or it is known as plastoquinol. And then you have cytochrome B6F complex. Then you have to remember plastocyanin. Short form for that will be PC. Then PS1. Then A. Then you will go for ferrodoxin. Then you have to remember ferredoxin NADP reductase. And finally, you have to remember NADP, oxidized. This is the sequence which should be very clear in your mind. If you genuinely want to understand the Z scheme, then this sequence should be very clear, okay? So you tell me guys, everybody here in the class, do let me know. This is basically regarding the Z scheme. So you people, please do let me know. Basically, ultimately what will happen? What will happen? Oxidation reduction, right? One will accept electron, that will be reduced. The one which is going to donate electron, that will be oxidized. So ultimately oxidation reduction, oxidation reduction, oxidation reduction will be there. So electron carriers are there. But here you have to remember your plastoquinone. It is not just an electron carrier, but also H positive proton acceptor. So now this is what we have to understand. Okay, what is happening, how, why, everything. But so PS2, right? What will be there? PS2, pheophytin, plastoquinone, that will form plastohydroquinone, which is also known as plastoquinol, cytochrome B6F complex, plastocyanin, PS1, A, then ferredoxin NADP reductase, whose short form is FNR, then NADP. So, who's going to remember it now? Who's going to remember it now? Quick. Who's going to remember it now? Quick, everyone. Quick, everyone. Who's going to remember it now? Done? Is it done? Quick. Ma'am, we are too busy in chit chat. Don't worry, when this question will come in final exam, we will skip it. Very good, Mr. Ames. Excellent, beta. Okay, do it quickly. So what is the point here? Photosystem 2, right? Photosystem 2, pheophytin, plastoquinone, plastohydroquinone, which is also known as plastoquinol, right? Then cytochrome B6F complex, plastocyanin, cyanin word, blue color. So obviously this one is having copper, right? This one is copper containing. This one is copper containing. So that's what first you have to understand. This one is copper containing. Then we are going for PS1. Then we are saying that there will be unknown electron acceptor A. And then after that, what is there? Very good, beta Dhruv. So after that, you have ferredoxin NADP reductase, and finally NADP. Very good. Okay. So let's revise it once again. What are we saying? Photosystem 2, pheophytin, plastoquinone. Then it will be plastohydroquinone or plastoquinol. Then you have cytochrome B6F complex. Then you have plastocyanin, then you have photosystem 1, then unknown electron acceptor A, then you have ferredoxin NADP reductase, and ultimately NADP. Okay? Now what is happening? Dekho beta, they all are present here in the thylakoid membrane. Just here, you will have your PS2. Okay? With it or in it, right? Because in NCERT also they didn't show it separately. You will find pheophytin. What are you going to find? Pheophytin. Now here in this thylakoid membrane, you will have this plastoquinone and you will have plastohydroquinone cycle. Matlab Q cycle we will see here. After that, you will have what? You will have cytochrome B6 या cytochrome F या simply cytochrome B6F complex in simple words. Then you have bacha plastocyanin, right? Unknown PS1, and then after that PS1, you discuss about the unknown electron acceptor A and all. So here towards the stroma side, you have ferredoxin and FNR, and in the stroma you have NADP. This is the thing. That is how it is placed. Just say you have PS2, you have cytochrome B6, you have PS1. You can see the positions as well. Plastoquinone in thylakoid membrane. Where is it present, beta? The positions are also very important. They are in thylakoid membrane. Where are you going to find it? You are going to find it in thylakoid membrane. Now, because I'm taking a part of thylakoid, right? This part, what am I trying to show you? I am trying to show you the thylakoid membrane here. I am trying to show you the thylakoid membrane here. Outside it's going to be stroma. Inside it's going to be lumen. Inside it's going to be lumen. Is that clear? Then plastocyanin. Then you have what? You have plastocyanin. So cyanin, it's copper containing. So it is towards lumen side. Location is also very important. So where do you see it, beta? You will see it towards the lumen side. Towards the lumen side. And then you have PS1, unknown electron acceptor A and all. So this is your ferrodoxin, FD. Ferrodoxin contains iron sulfur. So this FNR means your ferrodoxin, NADP reductase. So this particular thing, you will see this is absent in stroma lamella. And NADP, where is it in? It is in stroma. And these two, where are they present? Towards the stroma side, associated with membrane, but they are placed towards stroma side. Is that clear? Where are they present? Towards stroma side. Firstly, positions clear. I'm going very slow here so that you can understand this topic in depth. And after that, we will have break. After that, we will have break. Sure? Sure? So that is what we have. And that's what we have to study here as well. That is what we have to study here as well. So you people can see that, right? Plastoquinone, dekho. So then, this PS2, right? So this is that PQ pool here, cytochrome B6. Look at plastocyanin towards the lumen side, PS1, and with that unknown electron acceptor A, iron sulfur complex, FD, FNR, they all are associated. So the location, the position here is very important. The location, the position here is very important. And this point also, that with this PS2 towards the lumen side, you will find OEC. What will you find? OEC. So what is OEC? So OEC is your oxygen evolving complex. What's that, beta? This is your oxygen evolving complex. Let's repeat again. What's that? This is your oxygen evolving complex. Is that clear? Is that clear? So PQ, it is in thylakoid membrane, right? Here, plastocyanin is towards the lumen side, right? This ferrodoxin and all, they are towards the stroma side, and toward the lumen side, your PS2 is associated with OEC, that is oxygen evolving complex. So this is the place where the photolysis of water will occur, right? Where the photolysis of water will occur. Am I clear? Where the photolysis of water will occur. So let me start this part. Let me start this part. Just say you have PS2. You have PS2. You know that it is going to absorb the light. But it is not like that, right? I'll make it clear to you, right? If you think, beta, please listen to me very carefully. If you people think that the only PS2 will absorb the light first, then it will, then PS1, no, they are absorbing the light simultaneously, but we are discussing it in a sequence. So once this PS2 is going to absorb the light, it is going to, right, like after light absorption, it will be excited. It will be excited state. It will be in excited state, means it will get oxidized, right? When? So this P680 is going to pass its electron to pheophytin, right? To pheophytin. So this pheophytin, bache, this pheophytin is what? This pheophytin is the primary electron acceptor here. Primary electron acceptor here. But only here I would like to specify one thing, right? Only here I would like to specify one thing, that from PS2 to pheophytin, this electron movement is uphill. it is uphill. It is the uphill transport. So what's the meaning of uphill transport? Just now we discussed. So it is the uphill transport. What is the meaning of uphill transport? Like pheophytin is having lesser, lower redox potential than PS2, but still it is passing its electron to it. And now I'm not specifying 1 electron, 2 electron. That is not something we are going to discuss. So it is going to pass 1 electron to it, right? It is going to pass, it is going to pass electron to it. And this is basically uphill transport. But because PS2 is also getting the sunlight, it is absorbing the sunlight. So it is making it possible, right? It is making it possible. So it is basically passing the electron to pheophytin, but this from PS2 to pheophytin, this electron flow, movement is uphill, right? Now your PS2, after passing its electron, you know that it is in what? It is oxidized. Now this will be reduced. Now this pheophytin, it passes its electron to, right? This one, it passes its electron to plastoquinone. To whom? To plastoquinone, which is your PQ. Now, your PQ is not just, PQ is not just an electron carrier, but also H positive acceptor. That's what you have to remember, right? And here beta, there is a Q cycle which operates. Jaise plastoquinone to plastohydroquinone. See, this is not just the electron acceptor. So it will accept the electron, right? It will accept the electron. Now, what will it become? It will become your plastohydroquinone, which is also known as plastoquinol. Right? Already told you about that. So there is a Q cycle which operates here in between. So this one is not just accepting the electron, but this one is also accepting the proton from stroma. Right? Because in the surrounding there is stroma. Stroma is having H positive. So this plastohydroquinone, plastoquinol is going to accept the proton also. Is that clear? To proton also. So there is a Q cycle which operates in between, because beta, we have to regenerate this plastoquinone. So there are steps. In 2 steps this process occur, but trust me, that much depth is not required for you. That's why I'm not touching it. Otherwise you will be confused. Now, before going forward, let me tell you, if this one is getting electron, this one is getting oxidized, it has to come back to the ground state, na? It has to come back to the ground state, na? So oxygen evolving complex is helping here. So your OEC, it contain enzyme. Just here you have one exam, water oxidase. And here, what is going to happen? This is associated with the photolysis of water. What is photo? Means light. Lysis means breakdown. Photolysis of water will occur here. Photolysis of water will occur here. So what is happening in that photolysis of water? So your 2H2O, it will break. You will have 4H positive, 4 electrons, and oxygen will evolve. Just I told you, this is Z scheme. This is non-cyclic photophosphorylation, right? So this is how the oxygen is going to release. Oxygen evolving complex. So can I not say that if there is single H2O, one H2O is there, so there will be the release of 2 protons, 2 electrons, and there will be half O2. So if you need oxygen, O2, you need 2 H2O. And here, you have to use one trick, that is Man Ka College. Man for manganese, Ka for calcium, chloride. Man Ka College. That's what you have to remember. So here, what do you use? Manganese, calcium and chloride. They are used here in this reaction. So this is the photolysis of water. This is the breakdown of water in the presence of light. So it is Man Ka College. So that's how it works. That's how it works. So what is going to happen? The electrons, they'll be passed to, because where is it? It is in the, this is the thylakoid membrane where we find it. Hai na? We have already discussed that. This is basically towards the lumen side. So obviously the protons, they'll stay here in the lumen, and then this electrons will pass to PS2. So PS2, which was in excited state, it will come back to the ground state. It was oxidized. Now it will be, right? It will come to the ground state. So PS2, how the PS2 will come to the ground state? By accepting the electrons from oxygen evolving complex. Now, after this plastoquinone, it will pass the electrons to cytochrome B6F complex. cytochrome B6, yes, cytochrome B6 and then F complex. Now, when it is passing the electrons to this, so where will it passes the protons? So protons, it is going to pass in the lumen. It will pass the proton in the lumen. Then after this, cytochrome B6F complex, it will pass the electrons to plastocyanin. So plastocyanin, PC, Priyanka Chopra, so it's copper containing. That's what you can understand here. That's what you have to remember here. And this one is towards the lumen side. Its location, its position is very important, beta. So it is towards the lumen side. Where are you going to see this? It is towards the lumen side. So after this, you know that next to it is what? You have PS1. So it's not at all like this that PS1 cannot accept the, you cannot get excited. But here, that's how you can see this is a complete scheme. So when your PS1 is getting excited, obviously it is also, right, it is also coming to the excited state. So it is passing its electron to unknown electron acceptor A. Just we also see some amino acid and all. So we sometimes write it like this, ki unknown electron acceptor A is going to accept that, right? Yeah, 1 second. Let's suppose I'm writing it here. So it will pass it to unknown electron acceptor A. Then beta, it will pass to your FD, that is ferrodoxin. With it, FNR is associated. So that's how the electrons will pass. Now FNR will pass that electrons to NADP, which is in oxidized state here. So here I am writing, right, here I am writing FD. What's the meaning of FD here? It is the ferrodoxin. What is the meaning of FNR here? FNR means ferrodoxin NADP reductase. What's the meaning there? Ferrodoxin NADP reductase, right beta? And then after that, this NADP, what is this NADP? So this is going to be nicotinamide adenine dinucleotide phosphate. Dinucleotide phosphate. So that's what you have to keep in your mind. So electrons will pass here beta from this NADP, and NADP will pick up the protons from the stroma. It will make NADPH2. So if it is NADPH2 means it is having 2 electrons, 2 protons, right? So this is what you have. So NADPH will form here. This is the flow of electron. This is what your Z scheme is. So if PS2 is getting electrons from OEC, you can see here, PS2 is getting electrons from OEC. So you can also see PS1 is also getting excited, but who it is getting electron from PS2 via plastocyanin. So this is also the thing that you have to take care. So this is what Z scheme is. Why Z scheme? Because they are arranged, right? They're this redox potential thing is in zigzag manner. So here, this transport is uphill. This transport is uphill. Rest all will be downhill transport. So uphill transport means, so it will be against that redox potential. Like PS2 is having higher redox potential than pheophytin, but still it is passing the electrons to that. Is that clear? Is that clear? Done? Sure? Sure? Rest it is downhill. So cytochrome B6F, PC, so this ferrodoxin and then ferrodoxin NADP reductase. So this is to NADP. So this is the beta complete Z scheme. Here also I have mentioned that. So this H positive movement is important. H positive movement is important. Why? Why? Let me tell you. Beta throughout this, because it is also electron transport system. Right? So this is the electron transport system in the chloroplast that we discussed. So here in this electron transport system also, you are trying to build what? You are trying to build more H positive in lumen and less H positive in this stroma. Why? Why? Because we need this gradient. Why do we need this gradient? We need this gradient. We need this gradient because ultimately we have to do what? Now we have to talk about the 1 second. So now we have to talk about the ATP formation as well. So in the Z scheme, you got to know one thing very clearly. You got to know one thing very clearly, that when will the oxygen release. So you know that when you have the PS2 here, so oxygen evolving complexes towards the lumen side. So it is breaking down that, right, water, the water in the presence of light. So more and more H positive will be accumulated here. Now, you know that when plastoquinone is becoming plastohydroquinone, then also it is accepting the proton from the stroma, and while passing the electron again to cytochrome B6F complex, then it is passing that proton here in the stroma, in the lumen. Now after that, right, after that, when this, right, when this ultimately, after that, when this, you will see NADP, when this NADP is, this is oxidized, when it is getting reduced, when it is forming NADPH2, it is also picking up the proton from stroma. So ultimately, you are doing what? You are creating more H positive here and less H positive here, so that we can have this gradient, and this gradient is required for the ATP formation. And this CF0, CF1 is going to help in the ATP formation, right? So, just in mitochondria, you discuss the elementary particles. So here also we have this. So this is going to be the base, which is known as CF0. So this is head plus stalk, which is known as CF1. So CF0 is basically traversing. It is in membrane, bacha. It is in membrane. And when you talk about this head plus stalk, it is in stroma. So we have more H positive here, less H positive here. So it is maintaining a gradient. So through that, right, because protons cannot cross the membrane directly, they'll cross through it, conformational changes will occur. So that will help us to form ATP from ADP. So that's the complete process. And this part we understand as chemiosmotic hypothesis. This part we understand as chemiosmotic hypothesis. So this is what we're supposed to know, we're supposed to remember. So synthesis of ATP from ADP plus inorganic phosphate using light energy is photophosphorylation. So when it is the non-cyclic photophosphorylation, it is Z scheme. When it is the cyclic phosphorylation, it is only having the PS1. So beta, all the steps ki light will fall on green plant, they will are going to absorb it, right? This all the steps ki light will excite pigment of PS2, energy will be absorbed, right? P680 will become photooxidized, it will release electron, it will gain the energy, the electrons will be accepted by pheophytin, then they will be transferred to quinone. So here we have mentioned, right, here we have mentioned that after losing electron, P680 will be oxidized. So who's going to bring it to the ground state? The photolysis of water. So photolysis of water mein beta, it's Manka college. That is what we require here. The enzyme is water oxidase. So when there is the splitting of water, you can see for evolution of one oxygen molecule, you need 2 molecules of water. You need 2 molecules of water. So this will release 4 protons, 4 electrons. Electrons will be accepted by 680. Protons will be there in the lumen. We need it for increasing the proton gradient so that we can have the ATP synthesis. So electrons from PS2, they will excited, captured by quinone, enter the transport chain. So you know that plastoquinone, and this is going to be the step. So basically what is happening, your reduced quinone will pass the electron to cytochrome, it will get oxidized, but it is not just the electron acceptor, it is also the proton carrier, right? So PC, plastocyanin, will deliver the electron to PS1. So PS1 is also absorbing energy, it is becoming excited, it is passing the electron to primary acceptor A, and then bacha to ferrodoxin, and then to ferrodoxin and ADP reductase. So these are also the question. There's a ferrodoxin, it is basically iron sulfur protein, right? Outer surface of thylakoid membrane. So reduced ferrodoxin will pass the electrons to NADP. So all the things which we have discussed, they all are written here as well. So out of all this, what you have to remember, just a plastoquinone, it is a mobile electron carrier. So it picks up 2 electrons beta from PS2 by a pheophytin and also 2 protons from the stroma. So after reduction, it becomes plastohydroquinone or plastoquinol. Is that clear? So it delivers 2 electrons to cytochrome, but here it splits the pathway. I told you now a Q cycle is there in this PQ or PQH2, 1 electron to plastocyanin via this cytochrome F, then to PS1, but other electron through cytochrome B6 will come back to this plastoquinone pool, right? So this thing we don't have to understand in detail, not given in NCERT, but yeah, this is for the concept clarity, that's why I told you. So next is beta, while transferring electrons, PQH2 will release 2H positive into the thylakoid lumen. This contributes to the proton gradient for the ATP synthesis. So we have the Q cycle here, right? So this Q cycle, right, it is something that you will also study in respiration, but again, as I told you, not given in NCERT in detail, so that's why I'm not picking up. And special case, PS2 to PO phytin, this is also in the notes. So that's how you are getting the notes. They are simply amazing. Everything is written there, beta, and I have explained you each and everything. After the class, do read the NCERT. You are going to find it like very, very, very easy. Very easy you are going to find it. That I can vouch, and you are not going to get any question beyond that. Okay, so PS2, the reaction center here is this. It excites the electron from ground state to high energy state. So excited electron is then transferred to primary acceptor pheophytin. Is that clear? So this is uphill, beta. Why? Because PS2 is having more negative potential, or you can say, right, more negative potential. That is high reducing power. Let me rectify this. Its reducing power is more, right? Its reducing power is more means it can reduce, right? Right, its reducing power is more. Pheophytin is more positive potential, that is less reducing or more oxidizing power. So pheophytin is having more oxidizing power means it can oxidize something, right? Oxidize something means more oxidizing power. Loss of electrons. 1 second. Less reducing. Gain of electron, more oxidizing power. Loss of electron. It can, right? So its power is like that. So here, what you have to understand. So this PS2, its reducing power is more. Its reducing power is more. Is that clear? Is that clear? Sure. 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 One second, beta, I'll rectify this. Done, bache? So वैसे तो here I will rectify this part. Here I will rectify this part. Right? So 1 second. Beta, if we go as per NCERT, just 1 minute. If we go as per NCERT, if you will read that NCERT part, so in that NCERT part, they have clearly specified one thing. What they have specified there? They have specified clearly one thing there, that basically when there is the electron movement, right? If you will read that NCERT part, they have clearly mentioned that initially it is uphill, then downhill. So this part is as per the NCERT, which you have to remember here. Okay, so just open the NCERT, just read it once, and then we will come back to the cyclic photophosphorylation. So quickly do it. Quickly, beta. Go for it. Quick. Done? Okay. And beta, here, if you will read the NCERT, you're going to find that there is the uphill thing and there is the downhill thing. That initially the electron movement is uphill and then it is downhill. So uphill, downhill is associated with what? Uphill, downhill is generally associated with your this particular part that one is having a higher redox potential, but right, or I would say more positive redox potential. Other is having lesser redox potential, right, beta? And but still it is passing. One is having higher, but still it is passing the electron. That is uphill. But the point is, it is given in NCERT, and here, if you will go as per this part, just say uphill transport means, just say under normal condition, electron would not spontaneously move from P60 to pheophytin, but light energy will give the electron an energy boost, allowing it to move against its natural gradient. But here, if you will read this data is simply saying that if it is having more negative potential means less positive, means its reducing power is high. Reducing power is high means it can, right, its reducing power is high means it can basically reduce, or reduction means the gain of electron. It can basically reduce another entity, means it is having more reducing power, and this one is having more oxidizing or less reducing power, means it can, this pheophytin is having more positive. So I would say higher redox potential, or I would say less reducing, more oxidizing, like it can make other entity lose its electrons more easily. But here, right, if I will go as per NCERT, so in NCERT just uphill downhill thing is mentioned, that initially the transport is uphill then downhill. So just keep it in your mind, number one. Number two, here is if you will read the standard books, in that standard books they have clearly mentioned that PS2 to pheophytin, this transport is downhill. It's just that when it is PS2, it is absorbing the light energy, it is exuding its electron. That is because it is absorbing the light energy, then it is exuding. This is believed to be the uphill transport. So do not get confused here. Just forget this particular part. Don't get confused here. You stick to the NCERT part. And if you have better understanding of redox potential, then it is going to be a cakewalk for you. Then it is going to be easy. So normal condition mein electron is not spontaneously moving, beta, but light energy is giving the electron an energy boost, allowing it to move against its natural gradient. Is that clear? Is that clear? But light energy is giving the electron an energy boost. So electron is absorbing, or P680 electron is absorbing the energy, then it is moving uphill, right? So that's what you have to remember. So right now, just forget this part, right? And just read it from the NCERT. That is it. Now, next is the cyclic photophosphorylation. So in cyclic photophosphorylation, you have PS1 only. And you know that where do we find it? We will find it in stroma lamella, which is also known as fret lamella. Are you getting my point? It is stroma lamella, which is also known as the fret lamella. So here what you have to consider, or what you have to see, just in cyclic photophosphorylation, only PS1 is there. So it is there in stroma lamella or thylakoid membrane. So during cyclic photophosphorylation, electron is ejected from the reaction center. So this flow chart you have to understand, because here there will be just, right, there will be just this ATP formation, no oxygen release, right? There will be no oxygen release and there will be no NADPH, because stroma lamella, you can write it, it lacks PS2 and FNR as well. It doesn't have FNR. It doesn't have FNR. A very important pointer here. Is that clear? It's a very important point here. It's a very important point here. Is that clear? So stroma lamella, it lacks PS2 and FNR. So here you can see how the light is going to pass here. So you people can see, right, there is PS1, then it will pass its electron. Just 1 minute. Okay, so that's what you have to remember. So here you have PS1, it is going to pass its electron to FRS, so that is ferrodoxin reducing substance. That is ferredoxin reducing substance. Are you getting my point? That is ferredoxin reducing substance, and then it will pass to the ferredoxin. So beta, it will again pass to cytochrome B6, right? Then cytochrome F, then plastocyanin, then plastocyanin. So here ultimately from PS1 electron will come to PS2 only. Is that clear? From PS1 electron will come to PS1 only. So that is why it is the cyclic photophosphorylation. Is that clear? Is that clear? Sure. So ferredoxin, cytochrome B6 BF complex. So here I am also showing you this particular part. You can see that. So here what is happening, the electrons from reaction center of PS1, they will return back to this. So oxidation reduction is going on. That is basically energy will get released. So this released energy here also helping in creating a proton gradient. So this occurs occasionally. So ATP is formed here in this. Okay. Okay, so in stroma lamella beta, already I have mentioned here, I'm underlining this part also. In stroma lamella, there is no FNR, so no NADPH formation will be there. Is that clear? So cyclic photophosphorylation also occurs when only light of wavelength beyond 680 nanometers are available. So it's not a regular process, it is an occasional process. It is an occasional process. That's what you have to remember, okay? That is what you have to remember. Is that clear? So in cyclic ETS, no oxygen evolution is there, no photolysis of water, no NADPH formation. So this is basically the flow of electron. So from FRS, ferredoxin, then cytochrome B6F complex to plastoquinone, back to PS1. Is that clear? So when it is coming back to plastoquinone, again plastohydroquinone, that part will be there. H positive will again move. So here proton gradient will also be established. So that's what you have to keep in your mind. Is that clear? Is that clear? Sure. So here it is not showing, but yeah, from cytochrome B6 again plastoquinone will be involved, to which the electron will be passed. Is that clear? Is that clear? So any doubts? Any doubt? Sure? Any doubts? So after discussing it, we can have a break, है ना? We'll have a break for the lunch and then we will continue with the dark reaction. But so far, if there is any doubt, then do let me know. So far, if there is any doubt, then do let me know. Quick. Sure? Sure? So cytochrome B6 to plastoquinone, then plastohydroquinone. This is what you can write down. जैसे cytochrome B6 to plastoquinone and then that will be plastohydroquinone and then ultimately to cytochrome BF and then beta it will be plastocyanin, and that's how this process is going to be back again. Is that clear? Is that clear? So PQH2 then to cytochrome F and then further to plastocyanin. So that's how the movement is. This is what the diagram is. So next is chemiosmotic hypothesis. So beta, whatsoever diagram I have made, this is what they are showing, right? So H positive, that is proton from the photolysis of water, when plastohydroquinone is passing the electron to cytochrome B6, right? So that's how the protons will be there. And NADP, when it is getting converted to NADPH, it is also using the proton. So finally, through this elementary particle, CF not CF1 particle, you know that protons will pass from their higher concentration to lower and the ATP formation will be there. So this part is also important, high electrochemical potential gradient to low electrochemical potential gradient. Is that clear? So you can see thylakoid membrane, proton pump, proton gradient is required. So high H positive inside lumen, direct PYQ bacha. So this is what you have to understand. This is purely from the NCERT, right? KATP formed by breakdown of proton gradient. That's our PYQ. ATP formed by breakdown of proton gradient. Done beta? So NADPH formed by reduction of NADP. NADPH formed by reduction of NADP. So both ATP and NADP are used immediately in the Calvin cycle and fixation of CO2 will start then. So these are the steps beta. So this one, right, this chemiosmotic hypothesis is explaining the ATP synthesis. So just like in respiration, ATP here is also coupled to proton gradient, but we have to remember that H positive is more inside the lumen, less will be in the stroma. This is what we have to maintain. जैसे chloroplast में, in chloroplast, right, proton accumulate inside the thylakoid lumen, but in mitochondria, proton accumulate in the intermembrane space. So that's what we have to remember. Is that clear? So bache, this is again from NCERT, right? Everything is written here, ki splitting of water and all will be there. So I have explained everything to you in detail, so you can see where the low pH is there, where the high pH is there. So it is the CF naught which is there in thylakoid membrane. It is traversing, it is helping in facilitated diffusion. When do we say facilitated diffusion? When you are using a channel for the movement of things, but again from their higher concentration to the lower concentration. So CF1 is on the stroma side, so that's how the ATP will form. So that's what you have to keep in your mind. So after that there is a dark reaction which we will start after a break of 15 minutes. Okay, so let's restart after 15 minutes. Let's restart after 15 minutes. Done? Okay. Chale, let's restart, guys. Let's restart. I hope you all are done with your lunch. Huh? I hope you all are done with your lunch. So let's start the dark reaction. But before that, let's have some quick answers. So in Z scheme, who's involved? Your PS2 and PS1. These are the QRPs, quick revision pointers, right? Electrons move from PS2 to PS1. These are the points that you have to remember, right? So Z scheme में PS2 to PS1 electrons will move. Electrons move from PS2. So Z scheme में PS2 and PS1 are involved. Electrons move from PS2 to PS1. Now, photolysis, next point. Photolysis of water passes electrons to PS2 and it comes to ground state. And it comes to ground state. Done. Is it clear? Right. Next point is its PC passes electrons to PS1 and then it comes to ground state. PS1, जैसे unknown electron acceptor A is there, then ferredoxin, having that iron sulfur protein. So it is going to pass its electron to them. That's what you have to remember. So NADPH also remove. And yes, another thing is plastoquinone is not just an electron carrier, but also H positive acceptor. Now tell me any doubt. And PS2 passes its electrons to pheophytin. So pheophytin is referred to as primary electron acceptor here. So tell me if you guys are having any doubts, right? If you guys are having any doubts. So in cyclic photophosphorylation, we discuss PS1 only, no FNR. Where is it taking place? Stroma lamella. This one, where are you discussing? You are discussing here in grana thylakoid. Yeah, grana I would say. Right? Grana thylakoid. Yeah, grana I would say. Okay. So now tell me any doubts. Right? So maximum photosynthesis. Right? Blue and red light is there, but it is the red light which is more efficient. It is having less energy, but its wavelength is more. Okay, beta? So cyclic will occur when you will say the light is around 700 nanometer, or, you know, it is basically that far red light. So here, no NADPH because there is no FNR. There will be no oxygen evolution, only ATP. So proton gradient will be established. Done? Any doubts? Any doubts? Quick. Quick. Any doubts? Sure? So if there is any doubt, do let me know. This is, beta, these are the points that you people can refer for the quick revision. Is that clear? These are the points that you all can refer for the quick revision. Quick. You can even write it down. You can even write it down. Done? Sure? Sure, quickly write down these points and then please let me know if there is any doubt here, because today's the main agenda is to simplify this topic, okay? Right, what's the main agenda today? To simplify this photosynthesis topic, because many students, they complain here, right? That they don't understand photosynthesis well. Done? Sure. So in chemiosmotic hypothesis, already told you. So when will that proton formation occur? So chemiosmotic hypothesis. Chemiosmotic hypothesis. So here, if I have to go for the quick revision points here, QRPs here. So I will say that when membrane is intact, your membrane is intact, right? Lumen is showing what? Lumen is showing more H positive, that is acidic pH, that is low pH, and stroma will have low H positive, that is basic pH. So that is how it is going to be, right? Then we will see that there is the formation of ATP. Is that clear? Is that clear? Done, bache? Done? Done, bache? Okay. So now regarding this, there should be no confusion, right? That's why I simply cross that part. Ki do not just think about it, or do not write it. See, so uphill transport means when usually, right, electrons, they are electron when it is passing, right? When electron is passing from an entity to another entity, that entity is having what? That entity is basically having more negative, right? That entity is having higher energy level, or I would say more negative redox potential. So this is the uphill movement, right? This is the uphill movement. Is that clear? So normally electrons, beta, they move from high energy to low energy. Normally electrons, they move from high energy to low energy. But this time here, this electron from PS2 is going to gain the energy, but it will pass to the pheophytin, bacha, right? So when PS2 absorb light energy, electron is becoming excited with gaining extra energy. So excited electron is passing to pheophytin, which is even at a higher energy level. So that's the meaning here, right? It is at the higher energy level. It is pheophytin is already at the higher energy level. Usually electron prefers. So here this uphill and downhill is associated with what? Which is given in NCERT, right? So uphill means when the transport, usually electrons they prefer downhill transport, right? Like electron will pass to the low energy entity. So here what is happening in uphill, the electron is already passing from PS2 to pheophytin. When electron is passing, it is passing to pheophytin, which is already having the higher energy. Is that clear? Is that clear? Is that clear? Right? So this is what you have to remember. Okay. So here, let me rectify that. There is some error here in this point. So I will prefer to delete this slide so that you don't get confused here. So it is having more. So as per NCERT, we are going, right? Usually, no doubt, as per the standard literature, beta, as per the standard literature, they say that this is also downhill. But as per NCERT, I am mentioning you, right, that pheophytin is having more, right? Pheophytin is having, my bad, just a minute. So wait, let me rectify this part. So you can even check that out, and even you can just pass with it. It's that clear? So if there is any other doubt, that then you people can share that, right? That you people can ask that. Okay. So pheophytin, okay. So pheophytin is at higher energy level, that it is more negative. It is basically, beta, it should be more negative if I'll go as per NCERT. Okay. So this part from the notes I'll rectify before sending it to you in my own Telegram group and in our WhatsApp group as well. Okay. So these are the QRPs that you people can refer. So now let's talk about the dark reaction, right? Let's discuss this dark reaction part. Okay. So this is what we have to discuss. So we will talk about the C3, C4 cycle and the CAM pathway. That's what we have to discuss here. So chemiosmotic hypothesis diagram is also important. Kindly do not skip that. Okay. So next is your biosynthetic. Next is your biosynthetic phase. Next is your biosynthetic phase. Right. It is also known as the dark reaction. Dark word here is misnomer because it doesn't mean that the reaction is going to take place in the darkness. Rather, right, it is dependent on light and light reaction, but indirectly. If it will not get the products of light reaction, it will stop. So it is chemical dark reaction, light independent reaction, and it is also known as Blackman reaction. Okay, bache? So it needs ATP and NADPH. So what is going to happen here? What is going to happen here in this particular part? Right, it is taking place in the stroma of chloroplast, and this is responsible for the carbon dioxide reduction. Right, it is responsible for what? It is responsible for carbon dioxide reduction. So C3 pathway, also known as Calvin cycle, C4 pathway, Hatch and Slack cycle is there, and there is the CAM pathway that we have to discuss. So CAM pathway, basically it stands for crassulacean acid metabolism. Crassulacean acid metabolism. This is the name of this pathway. Is that clear? So it is also the Calvin-Benson experiment. It is also the Calvin-Benson experiment. Is that clear? So what do we have here? We have green algae chlorella. Like firstly, they conducted an experiment, right? And why do we call it a C3 cycle? Because the first stable product of carbon dioxide fixation is a 3-carbon containing compound. So green algae chlorella, which is a unicellular algae, told you already. Other than that, they use the radioactive carbon isotope 14C, right, in the form of 14CO2. Means they use the carbon dioxide which is having the radioactive carbon, and radiography was used here to track the labeled carbon in intermediates. Is that clear? So what they did, this algae, right, the algae were eliminated. It was given this 14CO2, right, for the carbon dioxide fixation, because they have to trace it, that what this CO2 is making after its fixation. So radioactive carbon quickly got incorporated into the compounds of the photosynthetic pathway. Then beta, at different time intervals, algae will be, killed, and what they checked, right, algae, it will be killed by dropping them into boiling alcohol. Then this preserved the intermediate compounds at that instant. Means, let's suppose you will have the chlorella, you are growing it, you are providing it deliberately, you are providing it radioactive carbon dioxide, means it which is having radioactive carbon. Now after that, you are allowing it some time, after time intervals, what you people are doing, you are just putting that algae in the boiling alcohol, because it is going to kill that. The metabolism will stop immediately, and this is going to preserve the intermediate compounds. That's how they traced everything. That's how they found a particular pathway. So the compounds later were separated by 2D paper chromatography, not required, right beta? So first happened is that here what they did, they did a very simple thing. Just say this is the C3 cycle. Why C3? Because here, right, first let me elaborate that. So why C3? Because here first stable product, is a 3-carbon containing compound. What's that? It's a 3-carbon containing compound. That's the first stable product. Is that clear? So what is it? It is 3-PGA. What is 3-PGA? It is 3-phosphoglyceric acid. Yeah, you can say that 3-phosphoglycerate. Right, 3-carbon containing compound. Rest what you have to remember, there is a difference in PGA and PGAL or G3P. These two are different things. Please do not confuse that. So 3-PGA is 3-phosphoglyceric acid or phosphoglycerate, carboxylic acid, carboxylate is there. Right, that is the functional group. Here, what do you have? This one is your phosphoglyceraldehyde. This one is your phosphoglyceraldehyde 3-phosphate. What's that? This one is your phosphoglyceraldehyde 3-phosphate, or it is also known as G3P, that is glyceraldehyde 3-phosphate. That is glyceraldehyde 3-phosphate. So that's what you have. So here the functional group is aldehyde, there the functional group is carboxylic acid, right? So first stable product is this. Now you are fixing CO2, you are getting this. So initially people were believing that the first carbon dioxide acceptor is a 2-carbon containing compound, but actually it is not like that. So C3 cycle, which is also known as Calvin cycle on the name of their discoverer, yeah, Calvin-Benson cycle, right? This will be there in all the plants. This is what now we have have to discuss one by one. We will split everything and we are going to talk about it. So here what will happen? So who's going to accept the CO2? Right. So here I'll write the important pointers directly. So CO2 acceptor is beta. It's a 5 carbon containing compound. It's a 5 carbon containing compound. And what's that? What's that 5 carbon containing compound? That is RUBP, or you can also write it like ribulose. Ribulose 1,5-bisphosphate. What's the word here? It's ribulose 1,5-bisphosphate. So what is the meaning of ribulose here, bache? Ribulose, ul is the word here. Ul, basically it is showing that it's a ketose. It's a ketose. Here the functional group is a ketone. Jaise ribose means aldose, ribulose means ketose, xylose means aldose, xylulose means ketose, erythrose means aldolose, erythrulose means ketose. So ribulose is there. So it is basically a pentose, that is, it is having the 5 carbon. So that's why here the word is RUBP, ribulose. If you will write ribose here, it is going to be incorrect. So please pay attention. So 5 carbon containing compound it is, ribulose 1,5-bisphosphate it is. So this RUBP, right, RU15BP. So this is going to accept the CO2. This process is known as, this is the very first step here of this particular cycle. It is carboxylation. And this one I already told you, it's a cyclic process. So if you are starting from A, you have to come back to A. So regeneration is also required here. So you have carboxylation here, you have carboxylation here. So this 5 carbon compound is going to accept the CO2. So the enzyme used here is Rubisco. So we have to discuss its property as well. But firstly, you will get to know that there will be 2,3 PGA here in this particular step. Right. So after this beta, next step will be of reduction here. So it is a cyclic process. 3 steps here we are going to see: carboxylation, reduction, regeneration. Carboxylation we have discussed. I already told you that it is ribulose here. That is important. So next important point is related to bacha Rubisco. So what is this Rubisco? Rubisco is a very, very, very important point here. Rubisco is the most abundant enzyme of biosphere. Most abundant enzyme of biosphere. Jaise you have most abundant protein of biosphere, of animal word is your collagen. Yeah, the most abundant protein of extracellular matrix is collagen. Same way, most abundant enzyme या protein of biosphere is, right, your most abundant protein of biosphere is your, biosphere is the word, then it is going to be Rubisco. That it is going to be Rubisco. Now, when you people talk about this Rubisco, its full form is ribulose 1,5-bisphosphate carboxylase oxygenase. Ribulose 1,5-bisphosphate carboxylase oxygenase. That's the name of the enzyme. This enzyme, what is special about this? What is special about this? This particular enzyme can show carboxylation as well as oxygenation. Carboxylation as well as oxygenation. Both the things this enzyme can show. Right, mostly it is the carboxylation, addition of CO2 only beta. It is the addition of CO2 only, right? But if let's suppose it depends on the relative concentration of CO2 and oxygen. So that is the speciality of the Rubisco, and that is something we all need to know. This is a very important topic. So here you can see what is this Rubisco. As I said, it is in the stroma of chloroplast. It is the main enzyme in the C3 cycle, and it is the most abundant enzyme on the biosphere or on Earth. So it is the primary gateway for inorganic carbon to enter in the biosphere. It is the one that is helping in this reduction, bache. So here, what is the speciality? Dual activity. So it can fix the carbon dioxide to RUBP, forming 2 molecules of 3-phosphoglyceric acid. And that is important to start the Calvin cycle. But it is also having the oxygenase activity, which will result in a wasteful process, right? That can result in C2 cycle, which is also known as beta photorespiration, which is a wasteful process for a particular plant. What's that? It's a wasteful process for a particular plant. So in presence of high oxygen, as I said, the dual activity depends on the relative concentration. So in presence of high oxygen and low CO2, rubisco binds to oxygen instead of CO2, and then there will be only one molecule of 3-PGA, one molecule of phosphoglycolate, which is 2 carbon containing. Right? So this is the photorespiration. So this is a kind of drawback of rubisco as well. So here in the very first step, what are you doing? You are using the rubisco. Now the second point here is, second step here is reduction. Reduction. In reduction, beta, what you have to remember, right? What you have to remember here, it's very simple. What you have to remember here, it's very simple. In a reduction, 2 ATP, 2 NADPH are used. 2 ATP, 2 NADPH are used. Here, your 3-PGA, what is it going to make? Then it will make triose sugars, and then beta, that will further enter in the processes. You will see the starch. Here, this is the complete cycle. See this, triose phosphate, my bad, not triose. Waise to it is going to be triose phosphate here, then you will have starch. So here in this process, what you have to remember, 2 ATP, 2 NADPH. Third step is of regeneration. So for any cyclic process, regeneration is very important, right? If there will be no regeneration, then you cannot continue that process. So in regeneration, the story is very simple. The triose phosphate, right? They will further make our RUBP. For that, you are going to use 1 ATP. 1 ATP. So now this is what we have to understand. This is from where our question is going to come. So this cyclic process should be very clear. So carbon dioxide and water is there, right? So carboxylation, which enzyme is used? Rubisco. So in the NCERT itself, you can mark it is having dual activity, which depends on relative concentration of oxygen and CO2. Now, bache, here in this particular step, 3-phosphoglycerate will form, means 2 molecules of your 3-PG are going to form. Then you will have 2 ATP here, 2 NADPH here, right? Right? So here, that is the requirement. Jaise light reaction will make ATP and NADPH. That is how it is going to be. Then there is triose phosphate. Finally, sucrose and starch will be there. This is going to be the regeneration. This is going to be the regeneration. And why are we writing sucrose here, beta? So please do mention it. Sucrose, which is a disaccharide. Sucrose, which is a disaccharide, which is having glucose plus fructose. Beta, it is an invert sugar. What sort of sugar it is? It is an invert sugar, right? It is also known as cane sugar. It is also known as table sugar, sucrose. So it is an invert sugar. And bache, why do we call it as invert sugar? This is your homework. This is your homework. This is related to chemistry, biomolecules. Why do you call it as this invert sugar? Now, the second point is, when you people talk about the sucrose, right, this is basically what? Non-reducing in nature. So in the phloem, right, in the phloem, what you people will see, right, in the phloem, because this is the phloem that is going to do what? It is the phloem that will help in translocation of food, right? This is the phloem that will help in translocation of food. So in the phloem part, beta, you will see it is the sucrose that is getting translocated. It is the sucrose that is getting translocated. Now, here you have regeneration. So this is one turn of Calvin cycle. What is it? So this is one turn of Calvin cycle or C3 cycle. So can I not say that? Can I not say that, bacha? Please pay attention. Can I not say that one turn of Calvin cycle is using 2 ATP, 2 NADPH in reduction and 1 ATP in regeneration? Can I not say that? So this is the information which is required for you, everybody. This is the information which is required for you. है ना? So 2 ATP, 2 NADPH in reduction, 1 ATP in regeneration, right? So, but for making glucose, 6 turns of C3 cycle are required. How many turns are required, bache? There are 6 turns. Why 6 turns? Because it is C6H12O6. It is C6H12O6. So how many turns are required? 6 turns are required. How many turns are required? 6. So if in one turn you are using 3 ATP, 2 NADPH, then for 6 turn, that is for 1 glucose. For 1 glucose, it is going to be 3 into 6, 2 into 6. It will be 18 ATP. It will be 12 NADPH2. Is that clear? That's the information which is important, which is required. Is that clear? Is that clear? Sure? Done? So this is important. This is important. Is that clear? So this is something that you have to keep in your mind. Okay? This is something that you have to keep in your mind. Just keep it in your mind. Just keep it in your mind. So this is what we have for making glucose. 6 turns of Calvin cycle, C3 cycle are required. One turn will use 3 ATP, 2 NADPH. So this is what we have, right? So this is what you have to remember for the C3 cycle. So all that steps are also written here, right? This is the table which is given in NCERT. So this is what you can say, you can discuss. Now, if somebody will ask me the kind of question, how many ATPs are required? How many ATPs are required for synthesis of, let's suppose, 200 glucose molecule? Tell me this. 200 glucose molecule. Right. Next question. How many ATPs are used in regeneration step for synthesis of 20 ATPs, sorry, 20 glucose. So etc. in one way or another, I can ask you the questions like that. Is that clear? Is that clear? Right? So that's what you people can keep in your mind. So how many ATPs are required for synthesis of 200 glucose molecule? Beta, one glucose you have read it is using 18 ATP. So it is going to be 200 into 18. Don't be like that there are 3 ATPs. 3 ATPs are used per Calvin cycle, and for one glucose, because it is C6, you have to take 6 turns of the Calvin cycle. So that's how you are going to answer it. Now here they have specified glucose are 20, but they have also specified that in regeneration step. So obviously for one glucose, or for one glucose, 6 turns will be there. So 6 turns means 6 regeneration steps means 6 ATP. So 6 ATP for one glucose. So 6 multiply by 20, you will have your answer. So do not get confused here. This is all about your C3 cycle. Then comes the C4 cycle, bache, right, where we will see the Krebs anatomy, right? So C3 cycle will be there in all the photosynthetic plants. So, but C4 cycle, where do you find? So C4 cycle, which is also known as Hatch and Slack pathway, it is also known as dicarboxylic acid cycle, right? You can also call it as Hatch and Slack pathway. You can also call it as carboxylic acid cycle. So it is in certain plants. Which plants? That is again a PYQ. In which plants you are going to find that? You are going to find in certain plants which are adapted to beta very high temperature, hot, dry and high light environments. Where will you find? You'll find it in certain plants which are adapted to hot and dry condition, high light environment. So MD Hatch and C.R. Slack in 1966, they discovered it, bacha. So these plants are known as C4 plants, because here the first stable product that we are going to see is a 4-carbon containing compound, that is your oxaloacetate. So whatsoever I am highlighting, bache, they are important previous year questions, right? They are important previous year question. Make sure you write it. Is that clear? So why C4 pathway? What? It's simple. Some plants, they are surviving in high temperature and the light. And you know that when it comes to the rubisco, it tends to bind with oxygen instead of CO2. When the temperature is very high, it binds with oxygen, not with the CO2, and photorespiration occur, which is a wasteful process, right? So energy and the carbon dioxide, both will be, carbon fixed will be wasted. So C4 plants, to minimize this photorespiration, they have evolved this pathway. In their cases, you will see C3 and C4 cycle, they will occur at different places. So here we have to study known as Krebs anatomy. Krebs anatomy. So Krebs, which is a German word which means wreath. Right? So what we have to understand here, we have to talk about the Krebs anatomy. So Krebs means wreath. Krebs is a German word which means wreath, means ring-like arrangement of cells. So this is what we call as wreath. For this we use the word wreath, right? So this circular, this ring-like arrangement is there. That's what you have to, yes beta, that is what you have to discuss here. Is that clear? That is what you have to discuss here. Is that clear? Is that clear? Are you getting my point? So let me explain you that with the help of diagrams first, and then we will discuss it further. Beta, this is a normal C3 plant. Now, you will see the inner section, the anatomy of a leaf here. Now, when you look at the anatomy here, this is the vascular bundle that you have. This vascular bundle, right, this vascular bundle here is covered with, it is covered with the bundle sheath. This vascular bundle is covered with the bundle sheath in the surrounding. You know that if it is dicot, the palisade and spongy parenchyma, otherwise like this ground tissue is basically the mesophyll there. This is the basic arrangement. Now when I look at the C4 plants, what do I see? No doubt there will be the vascular bundle. No doubt that vascular bundle will be covered by bundle sheath cells. But here what do we observe? Here we observe, right? We observe that these bundle sheath cells, right, their cell wall is so thick, that it is impervious to gaseous exchange, means there will be no gaseous exchange through that, impervious to gaseous exchange. That's the first point that you have to take care here. Now, secondly, if you will look at the mesophyll cells, can I not say that these mesophyll cells around here are arranged just like a wreath? Are arranged just like a wreath here, like in a circular ring-like form. This is the one thing that you have to understand. Now, the second thing that you are going to understand here is that the chloroplast is dimorphic here. Right, so Krebs anatomy, I told you what is the meaning of Krebs here. I told you where do you find it. So what will you see here? You will see that the chloroplast, right? Chloroplast is basically dimorphic. Chloroplast is basically dimorphic, means it is having two forms. It is having two forms. So what will you see here? What will you see here? You will see there is large chloroplast. There is a small chloroplast as well. There is a large chloroplast and there is a small sized chloroplast as well. Number one, this large chloroplast, where do you find it? You are going to find it in bundle sheath cells, and these bundle sheath cells, bache, they even lack papcase, have rubisco. I'll tell you what's that. Let me write it first for you here. This one, it is in mesophyll cells, and this one will lack rubisco but have papcase. That's the point that you have to take care. But have papcase, right beta? That's what you need to know. So mesophyll cells, they perform the initial CO2 fixation. So here there is an enzyme, papcase is present. What is the purpose of this particular enzyme? What is the purpose of this particular enzyme? So this particular enzyme is phosphoenol pyruvate carboxylase. Phosphoenol pyruvate carboxylase. This is the enzyme for C4 cycle, bache. That's what you are going to see here. Now, in the bundle sheath cells, what do you have? You will have rubisco, but here you are not going to see no pepcase. So that is what you have to keep in your mind. That is what you need to remember, okay? Okay, so you people can see here, it is written as well, right? The chloroplast of bundle sheath cells, what is it? So chloroplast is dimorphic here, it's larger, rich in rubisco, but it is going to lack phosphoenol pyruvate carboxylate. So it contains starch grains, but it lacks grana. It doesn't have grana, it has starch grain. So that light reaction part will be there in the chloroplast of the mesophyll cells. Next, chloroplast of mesophyll cell is normal type. Small size means normal type it is, right? It is having grana beta, but it doesn't have rubisco. So one thing that I am clear here, one thing that is clear to us is that in chloroplast of mesophyll cell, we will have C4 cycle, and obviously grana is there. So light reaction will also occur here. Light reaction will also occur here. And here in bundle sheath cells, you will have C3 cycle here, right? And here you will not see the light reaction. So this is something which is very important. Is that clear? This is something which is very important, right? So mesophyll cell is performing C4 cycle, right? And here bundle sheath cell is performing C3 cycle. So here you will see why. So this is basically, there will be two times carboxylation occur here, and one time decarboxylation also. 2 times carboxylation occur here, and there is one time where decarboxylation. So this is a cooperation between these two cell type. So this is also the cooperative photosynthesis. So look at the diagram here. Look at the diagram here. Is that clear? Is that clear? So you have mesophyll cells, you have bundle sheath cells, but they have plasmodesmatal connections in between, right? So in simple words, this atmospheric CO2 in bicarbonate form. So here who's the, in C4 cycle, first of all, I'll write the location here. These are the C4 plants. C4 cycle, location, mesophyll, chloroplast, chloroplast, normal, no rubisco, pepcase required for, pepcase is required for C4 cycle here, right? This is what you have to remember. Any doubts? Any doubts? Sure? Are you sure? Done, bache? Done. So this is the bundle sheath cells that you have. So bundle sheath cells, once again I'm repeating this so that you don't make mistake. Impervious to gaseous exchange. So here you people will see pepcase absent, rubisco present, large chloroplast. So in C4 cycle, primary acceptor of CO2 is a 3-carbon containing compound. Okay, so this is the thing that you have to understand here. It's a 3-carbon containing compound. What's that 3-carbon containing compound? That 3-carbon containing compound is phosphoenol pyruvate, right? That is PEP in simple words. So that PEP will combine. Bicarbonate is there, PEP is there. So its fixation will be done. Its fixation will be done, and here you have an enzyme pepcase. right? So C4 acid will form, bacha, and that C4 acid will get transported to bundle sheath cells. Am I clear? So that C4 acid will go to what? Will get transported to bundle sheath cells. So this is what you have to remember. Is that clear? This is what you have to remember. Is that clear? Is that clear? Okay. Any doubts? Any doubts? So that's an important topic. Tell me if there is any doubt. Done. So very first thing that you have to understand here. Say, let's suppose this is your phosphoenol pyruvate, right? The CO2 is getting fixed here. The enzyme here is peptase beta. So you will have OAA. That's a 4 carbon containing compound. And PEP is 3 carbon. So which cell it is? It's your mesophyll cell. It's your mesophyll cell. Done. So this oxaloacetate, bache, it will get converted to malate. That is again a 4 carbon containing compound. Right? So C4 acid. So oxaloacetate, then you have malate. Oxaloacetate, then you have malate. So here, bache, NADPH will be used. This is, okay, this malate. I'll change the color here also. So this is your bundle sheath cell. So this malate will pass through plasmodesmata. This malate will pass to, this malate will pass here, and now bache, it's decarboxylation occur. Yes, this is the step where again decarboxylation occur. So if decarboxylation occur means CO2 will release. So where that CO2 will go? Where that CO2 will go? That CO2 actually enters in C3 cycle. And moreover, your bundle sheath cell is impervious to gaseous exchange. There are no gaseous exchanges here. It is impervious to gaseous exchange, beta. So that is why even if in the surrounding there is less CO2, it is not going to affect this process here. And meanwhile, after this, you will get what? You are going to get the pyruvic acid, and in this process, beta, the NADP which was used here, it will reform. It will be reduced. So NADPH2 you are going to get here. So this is what you have to remember. So yes, there are 2 steps of carboxylation. There is 1 step of decarboxylation as well. So malate to pyruvate या pyruvic acid will form, right beta, which is again a 3 carbon containing compound. So let me write it here. So it is, I'm writing all in ionic form. So I'm writing this one in ionic form too. So pyruvate, which is a 3 carbon containing compound. So this pyruvate will find its way to the mesophyll cell again. And here, bacha, this pyruvate has to convert into phosphoenol pyruvate. And here, in each step, 2 ATP will be used. Actually, 2 ATPs are going to be used here. Right? So overall here, if I have to see, देखो, C3 cycle, right? For 1 glucose, you all are well aware. For 1 glucose, you all are well aware that there will be 12 NADPH2, there will be 18 ATP, right? Now, in C4 cycle, bacha, in C4 cycle here, this per turn, है ना? Here, for regeneration of PEP, 2 ATP are required. 2 ATP are required. Is that clear? So 2 ATP are required here. 2 ATP are required here. So per turn for regeneration of PEP, 2 ATP are required. So for 1 glucose, we know that there will be 6 turns. So 6 into 2, 12 ATP. So total, you know that in C4 plants, for 1 glucose, you can see that, right? In totality, what is going to happen here? So you people can see 12 ATP plus 18 ATP, that is 30 ATP, and there will be 12 NADPH2 that are going to be used here. Is that clear? That are going to be used here. Is that clear? So that is the important point that you have to understand, that you have to know. So pyruvate to phosphoenol pyruvate, ATP to AMP will form. So that's how you have to relate it, and you have to understand that. Done? So you should know which enzyme is present where. So see here, C4 acid is given. So you can simply remember, you will have OAA that will convert into malate. That malate will pass to this, and here the C3 acid is nothing but the pyruvate. You can also write it here, bache. So this is going to be your pyruvate. So here the regeneration of phosphoenol pyruvate will be there. So it is as simple as that. Is that clear? Is that clear? Done, bache? Okay, so this is the Krebs anatomy. So important C4 plants are your sugarcane, maize. Maize is C4, but wheat is C3. Do not confuse that. So sorghum, Digitaria brownie, Amanthus, Artiplex, etc. So sugarcane, maize, sorghum, they are important examples, right? So this is what you have to remember. So the best part is that here you are not going to find the photorespiration, or it will be less. So that is what we have to remember. So all the steps, everything is written here in detail as well, but energy requirement part should be clear to you. So this is all about the C4 cycle. So you know that there is no photorespiration. Why? Because spatial separation is there. CO2 fixation and Calvin cycle, C4 cycle and C3 cycle is there. So there is a spatial, by space they are separated. So bundle sheath cells are having high CO2 concentration. So Ruben, and it is moreover impervious to gaseous exchange. So when your C4 acid will get decarboxylated there, CO2 will go for the C3 cycle. So here the photorespiration part will not be there. So light reaction is also there in the mesophyll chloroplast. Dark reaction occurs in the bundle sheath chloroplast. So it is having higher productivity and yield because photorespiration is not there. So it is having, pepcase is having high affinity for CO2 even if CO2 is low. So they have better adaptation for higher temperature. They have efficient water use as well. Thick bundle sheath cell also. Storage of starch grain is also there. So these are the pointers that you have to remember. So this table here I have added. So this is important. You people can see that. Is that clear? This is important and you people can see that. Is that clear? So there is a comparison also, my dear students. There's a C3 plants, C4 plants are there, right? So we are about to finish it. So 3-PGA, OAA, right? So primary CO2 acceptor, RUBP. Here you have phosphoenol pyruvate. Enzyme is Rubisco. Here both are used. Calvin cycle, C3 cycle here is in mesophyll cell, here is in bundle sheath cell. Photorespiration high or absent. ATP use per CO2 is 3. Per CO2, here it is going to be 5. Right? 5. Is that clear? Is that clear? Per CO2 is the point here. So CO2 fixation and also comparison table is also there. Baki beta, there is one thing known as CAM pathway, right? So after that we will talk about the photorespiration and then we will just simply start the factors affecting the light reaction. So from scratch we have completed this topic. After that there will be respiration. From scratch we have completed this topic. So if you guys do not know, right? If you people do not know anything, right? If you don't know anything related to the photosynthesis, this lecture is basically for you. So here, right, CAM stands for what? This is crassulacean acid metabolism, bache. So basically it is named after the plant that belonged to the family Crassulaceae, in which first time it was observed, right? So basically what you have to remember, it is a photosynthetic adaptation found in succulent plants. You know, succulents which survive in xeric condition, dry condition, so that live in hot and dry environments. Okay, so here water loss is minimum by continuing photosynthesis under arid condition. Under dry condition, photosynthesis is there. So cactus, opuntia, your aloe vera, aloe, you know, in aloe also you will find this. So that's what, in pineapple also you will find this. Okay, so here beta, what is the funda? Here also C3, C4 cycle is there. Let me write it. See, no Krebs anatomy, no Krebs anatomy, but C3, C4 cycle are not separated by space, but by time. So this is, here in this case, what is happening? Stomata can't stay open during the day. Daytime, because this is growing in dry condition. So because it is growing in dry condition, so dry conditions are there. We have to conserve the water. So stomata should be closed during daytime. Right? So water loss due to transpiration will not be in the form of vapors, that is transpiration will not be there. So stomata can't stay open during the day, right, to avoid transpiration. So plants, they need CO2. So CAM plants, they open their stomata at night, so they absorb CO2 during night time, right? They'll keep it closed during the day to conserve water. So CO2 fixation, that is firstly your C4 cycle and the Calvin cycle, they'll be in the same cell but at different times. So this is what you have to remember. So night time mein what will happen? Carbon dioxide fixation will be there, right? The C4 cycle will occur, stomata is open, CO2 fixation by pepcase. Daytime mein beta, because stomata is closed, this CO2 from that same cycle will be done here. No Kranz anatomy, so no separation of C3, C4 cycle by space but by time here. That's it. Baki photorespiration, one thing that you have to remember, Chintu, Papa, Mummy. First Chintu, then Papa, then Mummy. So what does it mean? In the photorespiration, which is also the C2 cycle, right? Here you people will see that the rubisco, right? The rubisco, its oxygenase activity is there. So that is why here you will have 1 3-PGA plus you will have 1 phosphoglycolate, a 2 carbon containing compound, phosphoglycolate. So it is a wasteful process because here the CO2 which was fixed will be used here because it is respiration. You know that in the respiration what will happen? CO2 release, right? CO2 will release. So carboxylate activity fix CO2, but oxygenase activity will do photorespiration. So chloroplast, peroxisome, mitochondria, these three are involved there. That's a PYQ. So Chintu, Papa, Mummy, in this sequence you have to remember because in this sequence it will be completed. Okay. Okay. So RUBP, it is going to form 1 molecule of 3-PG or 1 molecule of 2-phosphoglycolate. That is important. Rest bit are not required for you, but this is the only part which you need to remember. Is that clear? Is that clear? Baki beta, when you talk about the factors that affect the photosynthesis, right? The factors that affect the photosynthesis. So you know that, right? Law of limiting factor will be there, which is given by Blackman, because here when a process is controlled by more than one factor, the factor which is in least, that is going to decide its rate. Jaise ki when a process is conditioned as to its rapidity by a number of factors, means here number of factors are responsible, so its rate will be limited by the pace of slowest factor. So here many factors are there, so the one which is limiting, that will decrease the rate. So if carbon dioxide is limited, even if you are going to increase the light, even if photosynthesis will not occur, because one factor which is required for this process is limited. So light beta, you know that 2% of solar energy is reaching Earth, right? Right, you know that photosynthetically active radiation is 400 to 700 nanometer. So light intensity, in case of light, you see the intensity, the duration, all the factors are considered. So light intensity, it is directly proportional to the photosynthesis up to a certain limit, the intensity of light, right? Beyond a saturation point, obviously rate will not increase, but its intensity is required. Just as shade plants, skyophytes, they are low light tolerant, they need less light. Sun plants, heliophytes, they need more. That's it. Baki light quality, matlab obviously if red and blue is more and red is more, the rate will be high. If you see plant is getting the light but it is a green light, the rate of photosynthesis will not be there. And for certain plants, you know that long day, short day plants, they need a required photo period, so duration also the matter. So light compensation point is also there, the light intensity at which CO2 uptake and CO2 release is equal. A saturation point is also there, where light intensity at which maximum photosynthesis occur. Beyond that, further increase will not be there. So this part is given in NCERT, this graph is given in NCERT. So I am showing what is the meaning of A, B, C, D and E here. So A is showing us the part of curve where light is limiting. Here light is the limiting factor. B mein light is only limited by 50%. C means light is not the limiting factor now. Some other factor is, because it is in this optimum amount. So some other factor is going to be limiting, bacha. So D is like ki this is the point where the light photosynthesis is maximum, and E is representing rate of photosynthesis. So this graph is important. This graph is important. Chali, next part. So CO2 concentration, you know that it is important. It is low in atmosphere, right? It is low, very low in atmosphere due to the pollution. No doubt it is increasing. So it's an increase in concentration up to 0.05% can cause increase in CO2 fixation rate. But beyond that level, beta, it is damaging. So C3 and C4 plants, they respond differently to the CO2 concentration. We know it very well, right? So at low light, when light conditions are low, neither group respond to high CO2 condition. So basically, first of all, you have to provide them the optimum light. Then at high light intensity, we know that C3, C4 plant will show increase in rate. But what is here to important, that C4 plants, beta, this information is important. It is from NCERT. C4 plants, they show saturation at about 360 microliter per liter. So here C3 respond to this value. So this is right to this concentration, and then there is the saturation. This is important, right? And CO2 is limiting to C3 plants mainly. We know that, right? C3 plant, they respond to higher CO2 concentration by showing increased rate of photosynthesis. They have better productivity. So this example, this is some greenhouse crops like tomato and bell peppers, right? We see when CO2 concentration is increased, their productivity also increases. Is that clear? Is that clear? And then beta, temperature, you know that dark reaction enzymatically based. So enzymatic process it is. So if the temperature is not optimum, the enzymes will be denatured. So that is how it will be affected. Okay. So effect on different plant types. C4 plants, they respond better to higher temperatures. We know it very well. They are adapted in such a way. They have more photosynthesis in that condition. C3 plants will not do that. Okay, so C3 plant will respond better when CO2 concentration is high, because then their rate will increase. So that's how you have to relate the things, right? This is it. Baki water, water is one of the reactants, but its effect as a factor is not that much. But you know that if plant will not get the sufficient amount of water, then water stress can occur, which can result in stomata closure and all. So in this way, it is going to affect. That is it. This is all about it. Baki beta, oxygen is there. High O2 decreases photosynthesis. It is Barberg effect. Pollutants and inhibitors are also there. Oxides of nitrogen, hydrocarbon, they also inhibit photosynthesis, right? Then you have factors affecting internal factors, may structure of leaves, their number, their size, their thickness matters, right? Accumulation of end product matters. Genetic disposition is also the way. That is it. Now let's start the PYQ practice. What say, guys? So this is all about the chapter. Now let's start the PYQ practice. Yes? Done? Sure? Sure? Chaliye, how many ATP and NADPH are required for one molecule of glucose through Calvin cycle? One of the simplest question. Again, your NEET UG 2026, in that also this question was there. Quick. Quick. So it's very easy, right? Just now we finished it. One molecule of glucose through Calvin cycle, right? Calvin cycle, C3 cycle, one molecule of glucose is written. So we know that there will be 18 ATP and 12 NADPH, right? So A is going to be the correct answer. So you can download the pylons to answer this question. Next part, the enzyme required for carboxylation in the C3 Kelvin cycle is. So again, easy, very easy it is, right? Very easy question it is. Quick everyone. Yes, what should be the answer here? What should be the answer here? So you know that it is going to be Rubisco. So I am ending the poll here. Exactly, very less number of students are answering the question. So guys, do one thing, download the pylons. Okay, download the pylons, right? Download the pylons and then you will be able to answer here in the poll. Chaliye, next question. I have started the poll, again a very simple one. It's a NEET 2025 MCQ. So here you people can see they are asking about the chromatogram. So yes, what should be the answer now here? What should be the answer now here? Quick. Chlorophyll A, B, xanthophyll and carotenoids. Chlorophyll A, B, xanthophyll and carotenoids. Xanthophyll, go for yellow, isn't it? Isn't it? Go for yellow. Eliminate these two options. Then chlorophyll A, we know that it is going to be blue-green, right? Chlorophyll B is going to be yellow-green, and here carotenoid is yellow to yellow-orange. So D with 4. So this is going to be our answer. This is going to be our answer. Is that clear? Is that clear? Done. Chaliye, next question. Next question, which of the following statement about Rubisco is true? Which of the following statement about Rubisco is true? And everybody quickly answer the question. Everybody quickly answer the question and make sure that you subscribe to our channel. Quickly, guys. Do it. So which of the following statement about Rubisco is true? About Rubisco, which one is true? That it is having higher affinity for oxygen than CO2. It is an enzyme involved in photolysis of water. Catalyze the carboxylation of RUBP. It is active only in dark. So obviously it catalyzes the addition of CO2, carboxylation of RUBP. Beta. Okay. Beta, on what basis you decided the B as the answer only? I told you it is the relative concentration that is going to be the deciding factor. Do you remember that? It is the relative concentration that is going to be the deciding factor here. It is the relative concentration that is going to be the deciding factor here. So you tell me, on what basis you said that it is the B which is going to be the correct answer? So it clearly indicates you didn't study well, isn't it? Yes, isn't it? And it's a 2025 PYQ. It's a 2025. People say the 2025 paper was difficult. You can just look at it. You can just look at it. Hanji, what should be the answer here? Beta, when did I say it is having? I told you that oxygenation and the carboxylation thing, it depends on the relative concentration. If CO2 is less, oxygen is more, then it will go for that, right? So it is, fourth is the most appropriate answer here. Okay, chaliye, next question. Again, NEET 2023 MCQ it is. So the reaction center in PS2 has an absorption maxima at. Absorption maxima at. Hanji, absorption maxima at. So you know that it is PS2. So it is going to be 680 nanometers. So I'm ending the poll. Okay. So this is it. This is all about photosynthesis. Let's discuss the next part. that is respiration. What say? Let's talk about the respiration now. Okay? Ready? Let's talk about the respiration now. Okay? Oh, so no recap. Chaliye, let's go. Let's start the respiration in plants. So we will start with our handwritten notes. But before that, I would like to specify few basic details from that. Okay? Right? We'll discuss few topics or few things here in detail, and then we are going to proceed. So what is the topic that we have here? We are going to talk about the respiration in plants. That is our topic. Okay? See, in the chapter breathing and exchange of gases, we discuss the 5 steps, and at the end we talk about the cellular respiration, that the cell is going to use the oxygen, it is going to break the oxygen, and then the energy will be released, right? So then this respiration thing, this is the one that we are discussing here. So what exactly is happening here in the respiration? So here in the respiration part, beta, so there is the breakdown. So breakdown of this carbon-carbon bond is going to be there. Just photosynthesis, it is associated with the build-up, but this one is associated with the breakdown of carbon-carbon bond. So when there is a breakdown of carbon-carbon bond, that too in multiple steps, that too by using enzymes, each step need enzyme. So multiple steps, enzymes are there, right? So that the energy release can be trapped in the form of ATP. This is what respiration is. So what is this respiration? A process in which there is the breaking of carbon-carbon bond, that too in multiple steps by using enzymes. And why in multiple steps? So that energy later can be trapped in the form of ATP. So this is what we have to understand when we talk about the respiration. So on that basis, we say that it is catabolic, right? Remember, kata means cut. There is a cut. Kata means cut, means there is a breakdown. So, but overall, by the end of this chapter, right, no doubt, we are saying that there is a carbon-carbon bond breaking. So it should be the catabolic reaction. But here, beta, we are going to see that it is amphibolic in nature. This respiration, this process is actually amphibolic in nature. That's what we need to understand here. That it is amphibolic in nature. So amphibolic in nature means dual. Amphi means dual, that there is catabolism as well as anabolism. So if I have to, because this is how I'm going to write the basics here, so you people can clearly see cellular respiration, it's a process in which breaking of carbon-carbon single bond is there, right, through oxidation within the cell. Oxygen is used, beta, and in a step-wise, in multiple steps this is occurring. It is not going to occur in the single step, because if it will occur in the single step, heat will release. We have to trap the energy in the form of ATP. That is why, right, so energy during cellular respiration is released in multiple steps, because in single step it may be converted to the heat. So ATP does form during respiration is broken down whenever energy needs to be utilized. Whenever, because ATP will form, we can use that. ATP is our energy currency, isn't it? So the compounds which will oxidize during this process, they are the respiratory substrate, beta. So usually it is carbohydrate, but your fats can be there, and rarely proteins are also used. Some plants, they also go for some acids also. That's what we have to discuss. But main respiratory substrate is your carbohydrate, which is also the glucose. It is in less or unavailability of carbohydrates, fat, and then proteins will be there. Is that clear? So it is amphibolic in nature. Now look at the gross calorific value. And let's talk about the physiological value here. Let's discuss that. Right? Let's discuss that. The gross calorific value and the physiological value. Just say, let's suppose you decide that from tomorrow I am going to study for 10 hours a day. Continuously I'm going to study. I'm not going to waste my time. You make your timetable as per that. Isn't it? Beta, indirect breakdown is there, right? But see, one by one. In multiple steps, you are breaking it. In multiple steps, you are breaking it. And one more reason is there. We'll discuss it one by one. So now listen to me what is happening. Just say, let's suppose today you have decided that I am going to, right? I am going to do this. Hannah, I'm going to study a lot, right? Yes? Yes, that I am going to study a lot, isn't it? Isn't it? And because today you are highly motivated, you are like, I am going to study 12 hours a day, take 14 hours a day, and then all of a sudden what happened? Next day, you got the reality check. You got the reality check. Just many students were like, ma'am, please, 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 right? Please, madam, do not, right? Do not just, please ma'am, start the plant physiology, please do this, do that. And then when I took the session, you were like, nahi yaar, now I'm not interested in studying. Means whatever you decide and whatever you actually do, there is a difference in that. There is a difference in that. Same is the case here. Gross calorific value is there, physiological calorific value is there, right? So these are the two things that we are discussing, right? These are the two things that we are discussing here. So first of all, I'll tell you what exactly is the meaning of calorific value. Right? So it is amount of energy released when 1 gram of substance is completely oxidized. It's the amount of energy released when 1 gram of substance is completely oxidized, beta, yeah, फिर it completely burned. Is that clear? Is that clear? Yes or no? This is basically the gross calorific, this is basically the calorific value. Now I am saying gross and the physiological. Gross is like the timetable that you have decided. Physiological is what you guys have achieved that. So gross calorific value is the total amount of heat energy, beta, which will be released when 1 gram of substance is completely oxidized, right, beta? So this is your gross calorific value when 1 gram of substance is completely oxidized. Okay, beta? So carbohydrate ke liye, right, when you check that in calorimeter and all, 4.1 kilocalorie per gram. But when you check it, right, inside the body, then it is a 4 kilocalorie per gram. Fats, look at it, 9.45 kilocalorie per gram. There it is 9 kilocalorie per gram. Protein 5.65 there. So basically what am I trying to tell you? First of all, I told you about the respiratory substrate. I told you that we have the carbohydrates, right? Carbohydrates are there, proteins are there, fats are there, then proteins, right? Overall, what is happening? Carbohydrates are a main respiratory substrate. You can see the energy that they are providing us. There is no major difference. There is no major difference in what? In gross and physiological value, isn't it? Isn't it? Yes or no? We do not have major difference here in gross, right? That is basically when you are, oxidizing it outside the body. Physiological means when it is oxidized, right? It is oxidized after, or I would say the actual energy which you are getting after digestion, absorption and metabolism inside the body. So after carbohydrates, our body, it goes for fat. If you will see, fats are giving us more energy. Can you see that? Can you see that? Fats are giving us more energy, beta, but still fats are not our primary respiratory substrate. Reason is very simple. You know that oxidation of fat is not that easy. In our body, we need an energy source which can immediately, which can quickly oxidize and can give energy. But fats, they take time. Their absorption, you know that it is not direct. Lymph vessels are also involved there. So their oxidation is not that easy. So fats are not preferred as the main energy source. This is the thing. And when it comes to the proteins, our basic framework, our enzymes, they are proteinaceous in nature. So if body has started feeding on proteins, that is a protoplasmic respiration, and it means kind of degradation is going on. Is that clear? So overall, this is what respiration, these are the basic steps. So cellular respiration is amphibolic in nature, and here the energy is going to release. Is that clear? Baki in NCERT, they have given a very simple part, beta, where they have discussed, do plants breathe? They don't have special respiratory organs, right? They don't have, with the handwritten notes itself, we are going to complete. So they don't have special respiratory organs, right? Their needs are also not that much. Why? Because they are sessile, they are fixed to a place, right? They are having the lenticels, beta. Just say, let's suppose this is a plant, right? They are having these lenticels by which they can just do the gaseous exchange. They can just do the gaseous exchange. So they don't have very high metabolic needs like animals, right? So here you can see each part can take care of its own needs. Jैसे leaves are also having the stomata, the stem part it is having the lenticels. Their demand is less, right? They are adapted in such a way that they can take care of their own needs. Baki beta, they have loose parenchyma cells to which also air spaces are there, air will be there. So that is why. Baki bache, phases of respiration mein external, internal and cellular is there, which we have even discussed in human physiology. So in external, the exchange is there between organism and environment. Like if I relate it with the human body, you know, in between alveoli and blood capillary. In internal respiration, blood and tissue. Here you will see the gaseous exchanges there. Cellular mein, right, the cell is going to use the oxygen, cell is going to oxidize the glucose. So this is what you need. So it is the exchange of respiratory gases between organism and environment. It is the exchange of respiratory gases between tissue and extracellular environment. So enzymatically controlled multi-step process is basically cellular respiration. So the basics should be clear. Baki beta, types mein you have floating respiration, which is more favored, right? The respiration using carbohydrate and fats. Fats are only used, right? See, they have more energy, but they are not preferred because we have to break them into intermediates, then we can use that. They cannot be directly utilized. We have to make acetyl coenzyme, glyceride, the glyceraldehyde, then we can use it. Rest beta, protoplasmic respiration, that will be in a starved cell, when a cell is not getting the food, right? So what is happening in this case? Proteins, right, which are rarely used, they'll be preferred. Okay, so obviously this respiration can deplete protoplasm, right, because structural and functional unit is protein. So toxic ammonia will be released and some seed during germination, they can use that. Okay, okay, chale. Now after this part, right, after this part, let's come to this. Okay, okay. General things clear: protoplasmic, floating, external, internal, cellular. Why do plants breathe or not? All the basic points we have covered. Now, respiration we are discussing. We have to understand few points here. What? Dekho. In the case of respiration, right, in the case of respiration, what is happening? Dekho, we have aerobic respiration, bache, and we also talk about the anaerobic respiration. Aerobic respiration is also there, anaerobic respiration is also there. Aerobic means oxygen will be used. Anaerobic, no oxygen. In the absence of oxygen, it is going to be. This is what we know. Now, if I relate it with the evolution, right, if I talk about the origin of life, we know that initially, right, the prokaryotes were there and initially anaerobic organisms were there. Then from that anaerobic organisms, aerobic organisms, they evolved. Isn't it? Isn't it? Yes or no? Isn't it? Yes or no? Exactly. So, aerobic respiration is there, anaerobic respiration is there, oxygen is used, and in anaerobic, oxygen will not be used. But we know that if in terms of evolution, if we see, so we know it very well that it is the oxygen, like aerobic organism, they evolved after the anaerobic organism. So what is the story here? Yeah, what is the point that we have to understand? Now, when you are talking about the respiration, the very first step is common, that is your glycolysis, which is also known as Embden-Meyerhof-Parnas pathway. Embden-Meyerhof-Parnas pathway, which is also known as EMP, right? This process, bache, this is basically oxygen independent, and its site is what? It's cytoplasm. Now I'll tell you why am I talking about this. You talk about the evolution, you know that first prokaryotic cells came. So in that prokaryotic cell, cytoplasm was there. We know it well. And even if I talk about eukaryotic cell, which originated from, like after prokaryotic cell, eukaryotic cell came. So we know it very well. We know it very well, beta, right? That in their case, we have mitochondria, which is associated with the respiration part. But cytoplasm is common to both. So when it comes to the respiration, the very first thing that you understand is glycolysis. That you have to understand is glycolysis. What you have to understand, guys? Glycolysis. So what is the meaning of this? Lysis means breakdown. What is the meaning of lysis, bacha? Lysis means breakdown. Glycolysis. Here you are talking about the glucose breakdown, and that breakdown is in 10 steps. In 10 steps, there is the breakdown of glucose, and each step needs the energy. Each step need a particular enzyme, right? 10 step. Each step is enzymatically controlled. Got that? Each step is enzymatically controlled. So what do we have here? We have glycolysis, which is basically breakdown of glucose. 10 step. Each step is enzymatically controlled. So it is Ambiden-Meyerhoff-Parnas pathway. So it is also known as EMP pathway. So here you people will see it is oxygen independent. The site is cytoplasm. So in 10 steps, what exactly is happening? So glucose, which is a 6-carbon containing compound, its breakdown will be there in 3-carbon containing pyruvic acid. In 3-carbon containing pyruvic acid, or you know that we can simply write pyruvate. So imagine this is what is going to happen first. Right now, I'm not talking about aerobic and anaerobic. If it is oxygen independent, means it doesn't need the oxygen. So you have a 6-carbon containing compound. You are breaking it into. So obviously you will have 3-carbon containing compounds. 2 pyruvate will be there. Now, beta, if this pyruvate, what will happen to this pyruvate? If oxygen is available, if aerobic conditions, if oxygen is available, if aerobic conditions are there, then what will happen? This pyruvate will enter in the transition reaction, या gateway reaction, or the link reaction, or in simple words, I will say it will go for Krebs cycle. It will go for Krebs cycle, then it will go for electron transport chain, and finally we know that terminal oxidation will be there if oxygen is available. But now students, if let's suppose oxygen is unavailable, if oxygen is unavailable, right, then what's going to happen? It will enter in a process. Anaerobic conditions are there. It will go for fermentation. And here in fermentation, we are going to discuss 2 types. One is alcoholic fermentation, right? Alcoholic fermentation, and another is lactic acid fermentation. Another is lactic acid fermentation. Is that clear? Is that clear? So that's what you see here, okay? That's what you see here. So first of all, in respiration, we are going to talk about the glycolysis. Once glycolysis is clear, what will be the fate of pyruvate? That depends on the oxygen availability and oxygen unavailability. If oxygen is available, there will be complete oxidation. There will be complete oxidation of glucose into CO2 plus water. Right, it will release carbon dioxide and water. But if oxygen is unavailable, there will be incomplete, right? What is going to happen? Incomplete oxidation of glucose. Less energy from glucose will be obtained. Is that clear? Less energy from glucose will be obtained. Is that clear? Done. So the very first thing is glycolysis that we have to discuss. Is that clear? So it's a 10-step process. So how are we going to discuss that? Let me show you. So glycolysis, it's a 10-step process, oxygen independent process. So do remember, first 5 steps, right beta? Do remember this thing. First 5 step and next 5 step. So what's going to happen here? Yes, everybody. What is going to happen here? Please pay attention. First 5 steps, which are also referred to as preparatory phase, right beta? And here you people will see there will be payoff phase as well. So in the preparatory phase, what will happen? Here the ATP is used. ATP used. Okay? Where? Step number 1 and 3. Now, here, next 5 step, ATP form. Okay? Here what will you see? You will see substrate level phosphorylation is there, right? Step number 7 and 10, we will see the substrate level phosphorylation will be there. Is that clear? So steps are also important. So it's not like as such step, you should know which reactions are going on there, because this is the kind of question they do ask. So substrate level phosphorylation will be there. We will discuss what is the meaning of substrate level phosphorylation. You don't worry about that, okay? Right? So this is what we have. So first 5 step preparatory, next 5 step payoff. So here ATP is used, ATP will form, right? Other than that, in glycolysis, you will see step number 1, 3, 7 and 10. They are irreversible, right? They are irreversible steps. This is what you have to remember. But rest all steps are reversible. In step number 7, there is a catch which we have to discuss. So this is what you have to know. So substrate level phosphorylation, in which step it is taking place, what are the reactions there, all you have to understand. So by the end of the glycolysis, 2 pyruvate, 2 NADH, it is NADH, right? 2 NADH, 4 ATP will form, but 2 are already used, so net gain will be 2. So that is what we have to discuss. So glycolysis is our topic. So already told you, bacha, already told you, right, that it is oxygen independent and it is in cytoplasm. So your ribosome, sorry, your RBC, right? Your mature RBC, which doesn't have mitochondria, you all are well aware. It doesn't have mitochondria, you all are well aware. So in the mature RBC, what you people will see, in the mature RBC, what you people will see, you people are going to see that they are dependent on, RBC depends on the glycolysis. Okay, so what do you study first? Dekho, 5 steps now. So you have glucose, you have glucose 6-phosphate. So here ATP will be used, because how? Then it is going to add the phosphate, right? So here the enzyme used is glucokinase या फिर hexokinase, right? You can even write hexokinase or glucokinase. I'll tell you why do we write it. We will discuss that, and what is the difference in two, that is also we are going to discuss. And magnesium is used as cofactor in all the enzymes of glycolysis. Magnesium will be used as the cofactor in all the enzymes of glycolysis. Now, firstly, I will write first 5 steps, then I will explain it to you. Okay? Then I will explain it to you, the story of these first 5 steps, and then, bacha, we will discuss the next 5 steps. So, glucose is giving you glucose 6-phosphate. Glucose 6-phosphate is giving you fructose 6-phosphate. Then that fructose 6-phosphate is giving you what? It is giving you fructose 1,6-bisphosphate beta, fructose 1,6-bisphosphate, and then it will undergo cleavage. This step is also reversible one. Here the aldolase enzyme is used. I'll write the other enzymes also. So here you will have the glyceraldehyde 3-phosphate, that is your G3P, or you can also write it like PGL, or there will be dihydroxy acetone phosphate. This is again a 3-carbon containing compound. 3-carbon containing compound. Short form here is DHAP, and these two are also isomers. So glucose to glucose 6-phosphate, glucose 6-phosphate to phosphoglucoisomerase. First, I will discuss these 5 steps, then we will go for next steps. So you make sure you remember them, okay? Please at least try to remember it, please. Phosphofructokinase, very important enzyme. Aldolase This step is also reversible. So, here you have triose phosphate isomerase. For all the enzymes here, right, for all the enzymes here, you need magnesium ion as the cofactor. Now, these are the first 5 steps. So, step number 1, step number 2, this is the step number 3. Here also ATP will used as ADP. This is the step number 4. This is the step number 5. So, what are they? This is the preparatory phase. Which phase it is? Preparatory. It is not payoff, preparatory. It's like I always give this particular example to students. You just imagine it like you have to set a business or you have to start a business. First, you have to invest money in that. So, it is that investment phase. That's how you can remember. Now, see, glucose to glucose 6-phosphate. You have added the phosphate. You have added this phosphate. So, you have used the ATP. So, this step is irreversible. Now, it is also the locking step. That's what I would like to tell you. It is also the locking step. Why it is a locking step, bacha? Because this glucose 6-phosphate is impermeable. It is impermeable to cell membrane. Like this glucose 6-phosphate cannot cross the cell membrane. Just say, for an example, this is our cell. In cytoplasm, this process will occur. Your glucose will enter here. So, here you are converting that glucose into G6P because you have to go for this process. So, this is basically a locking step. Which step it is? It is basically a locking step because it is G6P is impermeable to cell membrane. So, in masters, one question was there. I gave one, this entrance examination in that question was there that why in a hospital patients are directly given this thing, na, glucose. Why can't we give glucose 6-phosphate to the patient? Patient is already weak, right? Then why are in solution we are putting glucose? Why can't we directly put glucose 6-phosphate? Because glucose to glucose 6-phosphate conversion takes ATP. So, we should give the G6P directly. But what is the explanation here? G6P is impermeable to cell membrane. It cannot cross that. So, we cannot give that patient G6P, right? This is the first step where the ATP is used. Now, glucose 6-phosphate in its isomeric form, it will change. You have fructose 6-phosphate. Then, fructose 1,6-bisphosphate is the word. Why this is important? Dekhiye, the word here is bis. Just 1, 2, 3, 4, 5, 6. You have 6 carbons. How many carbons do you have here? You have 6 carbons. So, to this 1 carbon phosphate is there. To this 6 carbon also phosphate is there. So, in one compound, 2 different carbons, here phosphate is attached. So, you will use the word bis. You will use the word bis. To if say and to same carbon, right? Beta, each step is enzymatically controlled and each step is having different enzymes, right? Each step here is having different enzymes. Let's discuss that, Karan. So, bis means it is attached to 2 different carbons, right? In NCERT at places they have mentioned di, but actually it is bis. When do you say di? If this phosphate will be attached to the same carbon, then you will write diphosphate. But we are writing here bisphosphate. Bisphosphate means it is attached to 2 different carbons here, to carbon number 1 and 6. So, again, one phosphate you have added, you used ATP. Another phosphate you have added, you have used another ATP. So, in these 2 steps, 1 and 3, which are irreversible also, you have used the ATP. Now, you are cleaving it, you are breaking it down. Matlab, it is something like that. 1, 2, 3, 4, 5, 6. Here also you have phosphate. Here also you have phosphate. Now, you are breaking it down, right? So, you are having 1, 2, 3. 1, 2, 3 means you are breaking it. There is a cleavage here. So, aldolase is going to do that. Aldolase is going to do that. So, here you will see that this is glyceraldehyde 3-phosphate. It is again having 3 carbon. It is again having 3 carbon. So, it is G3P. It is PGAL. It is G3P. It is PGAL. Isn't it? Isn't it? So, it is glyceraldehyde is the functional group. This is dihydroxyacetone phosphate, DHAP. Di means 2, hydroxy. So, this is a ketose, this is a aldehyde. So, they are also isomeric, right? So, triose phosphate isomerase is their 5th step. Now, up to 5 steps, all clear? All clear in these 5 steps, all clear. Why do we call it as the locking step? Why do we call it as locking step? Now, here, another important thing is your phosphofructokinase. This enzyme is a very important enzyme. There are 3 regulatory enzymes that you people are going to see here. So, phosphofructokinase, it is the main regulatory enzyme. It is the key regulatory enzyme. Let me tell you what is the meaning of this main या फिर key regulatory enzyme. Let me discuss that here. Let me discuss that here, that why do we call it as main or the key regulatory enzyme, right beta? So this is important, this is very important. So far if you guys are having any doubt, do let me know. Done? 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 So any doubt here, bache? Sure? So this is the main. It's not like that we have just one regulatory enzymes, we have many, but this is the main regulatory enzyme. So wherever you will find the kinase, it has to do something with the phosphate. Always remember, isomers, isomerase, isomerization. Aldose is associated with cleavage. Mutase is associated with rearrangement. Now, this enzyme, main regulatory enzyme, so bacha, it is inhibited by ATP and is stimulated by, right, this enzyme is inhibited by ATP and is stimulated by ADP plus inorganic phosphate, right? Mainly means if there is excess of ATP in the cytoplasm, so this enzyme will be like no need to go for the process, no need for glycolysis. But if there will be more this ADP and inorganic phosphate, so this enzyme will be like ki yes, chalo, let's go. So this is the main regulatory enzyme. There are other enzymes also, but key enzyme, main enzyme is this. And this is an allosteric enzyme, bacha. It is having, right, it is an allosteric enzyme. It is actually a dimer, right, 2 subunits. Matlab, one part will attach to this ADP-PI and this another part will attach to the substrate. So this is the main regulatory enzyme. These are the first 5 steps. So that's what you have to remember. Is that clear? Is that clear? So glycolysis, Embeden-Meyerhof pathway, Embeden-Meyerhof-Parnas pathway, EMP pathway in 1930, they have given it. I told you, right here there will be the sugar splitting. That too in 10 steps, be it aerobic respiration or anaerobic. This is the first common step here, right? After that, you know that glucose breakdown into two molecules of 3 carbon containing pyruvate will be there, right, bacha? So here you know that in anaerobic organism, it is the only process in respiration, right? So plants mein what is happening? We know that it is the sucrose, right? The non-reducing sugar. It is basically translocating. So this sucrose, bache, right? This is the product of photosynthesis and then the starch is there. So sucrose will break, glucose fructose is there and then they will enter the glycolytic pathway. It is the simple process. So first 5 steps I told you, next 5 steps. So phosphofructokinase is the main key regulatory enzyme. It is also known as pacemaker of glycolysis. I forgot to write it here. So I'm writing it again. So this is also known as pacemaker of glycolysis. What do you call it, bacha? You call it as pacemaker of glycolysis. This one, this is the key enzyme, right? This is the key in the regulatory enzyme. So that's what you have to keep in your mind. That's what you have to keep in your mind, right, beta? So allosteric enzyme it is, and not just the ATP, I told you, citrate, right? It is also inhibited by high concentration of citrate. Right? So allosteric enzyme hai, one half of dimer will have ATP binding site, other will substrate. So it is having separate allosteric binding site also that can bind with either with ATP or ATP, that I have already told you. Other than that, hexokinase and pyruvate kinase is also there, right? But main regulatory enzyme is that one, right? But main regulatory enzyme is basically that one. Is that clear? So main is your Pacemaker of glycolysis is this, but they are also the regulatory enzyme. So your hexokinase, it is inhibited by G6P. If it is G6P is in excess, it will be inhibited. And pyruvate kinase, it is stimulated by fructose 1,6-bisphosphate and it is inhibited by ATP and acetyl coenzyme. So these three are the regulatory enzyme, but main one is this one. Okay? So steps are also given, right? First 5 steps are also given. So before let's move to the next steps. Then there are some other details also that we have to discuss. So G3P, 3 carbon, and this is the 5th step now. This is where we dropped our 5th step, DHAP, 3 carbon. Now what's going to happen, right? We have two 3 carbon containing compounds, right? Now what's going to happen? This DHAP will get converted to G3P. This DHAP will get converted to G3P. Why? This DHAP will get converted to G3P again. But why is it so? Why? Because bacha, the further enzymes, the enzymes that further we have. One second, huh? Done. So G3P to DHAP. What do we have? G3P to DHAP. So further the enzymes which are there, right? They are basically to hydrolyze this DHAP. They are not. They are to hydrolyze this G3P. They are not for DHAP. Right? Right. This is one simple explanation that you can remember. So you will have. Obviously, if this DHAP will get converted here, so don't you think that you are going to have 2 G3P here? That you are going to have 2 G3P here, 2 glyceraldehyde 3-phosphate, right? That you are having 2 glyceraldehyde 3-phosphate here. Isn't it? Isn't it? Isn't it? So now further steps will be as per this. So this is the 6th step that we have next. This is the 6th step that we have next. So it is glucose glyceraldehyde 3-phosphate. Now we will have 2 glyceraldehyde 3-phosphate. So beta, this will form what? What will it form? Glyceraldehyde 3-phosphate. This is the 6th step here. And in this particular 6th step, what will happen? Please pay attention, everybody. Here, right? Here in this particular step, what is going to happen? There is an amazing phenomena that will occur here in this particular step. Here you will see oxidation and phosphorylation, both will be there. Right? Oxidation and phosphorylation, both will be there here in this particular step. Oxidation will also be there and phosphorylation will also be there. Oxidation and phosphorylation both will be there in this particular step here. So here, why am I writing 2? 2pi means inorganic phosphate. Inorganic phosphate. And here you will see 2NAD, because now why 2? They'll form 2NADH2. 2NADH. So why NADH? So it is NADH. It is nicotine amide adenine dinucleotide. Why 2 bacha? Because we have 2 glyceraldehyde 3-phosphate now. Is that clear? We have 2 glyceraldehyde 3-phosphate now. Is that clear? Is that clear? So the enzyme used here is glyceraldehyde 3-phosphate. It is glyceraldehyde 3-phosphate dehydrogenase. This is the enzyme which will be used here. Glyceraldehyde 3-phosphate dehydrogenase. Is that clear? So here what will you get? You will get 2,1,3-bisphosphoglycerate. This will form. So 6th step clear? 6th step clear? It's 3 carbon containing obviously. It's 3 carbon containing obviously. So it is 1,3-bisphosphoglycerate that is going to form. that is going to form. So beta, this is the 7th step. Here you will get Y2, now you know Y2, okay? Here you will get 3-phosphoglycerate, 3-phosphoglycerate. Now please pay attention. Dekho, this is the 7th step. This is the 7th step. Which step is there, beta? 7th step. It's 1,3-bisphosphoglycerate. Means it is having 2 phosphate. This is 3-phosphoglycerate. Means it is having just 1 phosphate. So where that 1 phosphate will go? Where that 1 phosphate will go? Beta, it is very simple. It is very simple. Here, your ADP, your 2 ADP. Why 2? Because we have 2 glyceraldehyde 3-phosphate now. So they will form 2 ATP. So enzyme used here is phosphoglycerate kinase. Phosphoglycerate kinase. So this particular step, which is the 7th step, because these are the steps where they ask the question. So this is the 7th step. So what exactly is going on here? This is the step, beta, where substrate level phosphorylation is occurring. Where substrate level phosphorylation is taking place. Where what is occurring, beta? Substrate level phosphorylation is taking place. So phosphorylation, you already know, addition of phosphate. ADP plus inorganic phosphate will make you ATP. So it is substrate level phosphorylation. Here your substrate itself is helping in the ATP formation. Here your substrate itself is involved in the ATP formation. Is that clear? Is that clear? Yes, beta. So this is the step of substrate level phosphorylation. This is the step of substrate level phosphorylation. So ATP is formed by direct transport of phosphate group from the substrate to the ADP. Is that clear? Is that clear? Sure? Sure. So this is substrate level phosphorylation. So this reaction is important to understand. So here this is the payoff phase. Now after that you will have 2-phosphoglycerate. Now what will change here? Dekho, what is happening here? Look at this. You have 3-phosphoglycerate, means phosphate is attached to 3rd carbon. 2-phosphoglycerate means phosphate is attached to 2nd carbon. So this is simply the rearrangement. So for rearrangement, enzyme used is mutase. Because it is simply the rearrangement, so the enzyme used is mutase. Right? It is phosphoglycerate mutase. So rearrangement means simply there is. Right, rearrangement means simply there is the, right, like phosphate from 3rd carbon is shifted to the 2nd carbon. Right, phosphate from 3rd carbon is shifted to the 2nd carbon. That is it. That is it. Is that clear? Now, after this, what will happen? Again, here 2-phosphoglycerate, now you will have phosphoenol pyruvate. Phosphoenol pyruvate. In short form, we write it as like PEP. It is again 3 carbon containing. Enzyme used here is enolase, right beta? So in 2-phosphoenol pyruvate, en, double bond, ol is also there. So here the water will release. That's how the double bond is coming. Phosphoenol pyruvate is there. And after that beta, after that, so this is basically 6th step, 7th step, 8th step, 9th step. The last step is there. Right, the last step is there. 1 second. So you can see this is the last step. So in last step you will have the pyruvate, which is a 3 carbon containing compound. The enzyme used here is pyruvate kinase. The enzyme used here is pyruvate kinase. Again, bacha, because phosphoenol pyruvate, it is having phosphate. This is pyruvate only. So here also what is occurring? This is also the substrate level phosphorylation. This is also the substrate level phosphorylation. So this is the 10th step where the substrate level phosphorylation is going on. So these are the 2 steps in the glycolysis. These are the 2 steps in the glycolysis where substrate level phosphorylation is taking place. Is that clear? Now, this enzyme, phosphofructokinase, if it is stimulated, if it is not inhibited, means it will form fructose 1,6-bisphosphate. So if it has formed, it is clear that now the glycolysis will continue. So that's why we said that this pyruvate kinase, for that enzyme, for pyruvate kinase, your fructose 1,6-bisphosphate is just like a stimulant. Yes, now we have to continue the process. And when it comes to the hexokinase, it is even inhibited by G6P. So that's how everything is related. Main key enzyme is this. Baki beta rest, all the things are mentioned here. And some books, they use this thing. Either they write hexokinase or they write glucokinase, right? But they have little difference. Jaise hexokinase, which is in the brain cells, glucokinase mainly in the liver, right? Pancreas, it is in brain and muscle cells. Beta, hexokinase, what's the thing? It can bind to the glucose even when its concentration is low, right? So that's what you have to understand. Means, dekho, hexokinase is nearly in all the cells because it is having more affinity for glucose. So if an enzyme is having more affinity for glucose, if it is having more affinity for glucose, means if that particular substrate, even if it is in less concentration, that enzyme can bind. So it means, right, whenever you know that, this is basically your Michaelis-Menten, right? High affinity means there will be low Km. Substrate concentration at which the maximum velocity, substrate concentration at which the velocity is half maximal, and there will be low Vmax. This is what we already know. If affinity is high, the Km and Vmax is going to be low. So your your hexokinase, it is having high affinity. It is having high affinity for glucose. So if it is having high affinity for glucose, its Km is low, right? Right, beta. So it needs, even when glucose is in less amount, it can bind. Now, in liver, where we have to, the glucose will be converted to glycogen. If there also enzyme will have high affinity for glucose, then obviously the enzyme will immediately bind to the glucose, then it can immediately break it. But this is not what we want. We want that in liver cells, when glucose is in excess, then only they should undergo glycolysis, otherwise glycogen should form. So that is why here, this particular enzyme is having low affinity for glucose, means high Km and high Vmax. Okay, and this one is not at all inhibited by G6P. Is that clear? Is that clear? Baki beta, step 1, 3, 7 and 10 irreversible. For 7th step, there is a catch. We'll discuss that. So step 7 and 10 is of substrate level phosphorylation. So in 1 and 3, ATP used, 7 and 10, ATP form. In step number 6, NADH2 will form, which we will send an electron transport chain, and here from here also we will get the ATP. So we will discuss that. Now, one of my students asked me this thing, that ma'am, in some literatures it is mentioned that 7th step, just here, I told you that this is from one of my favorite books, Stryer, Biochemistry. So this step is what? Here it is irreversible, but some people say that this step is reversible. But actually, what is the funda? Just here enzyme used is phosphoglycerate kinase. Phosphoglycerate kinase. Beta, in glycolysis, it will be like this, right? In glycolysis, it is going to be like this. Means, what does it mean? Just phosphoglycerate kinase. That's the name of enzyme. Yes? One second. Dekho, so it depends on the phenomena. Just when it is gluconeogenesis, when glucose synthesis from a non-carbohydrate source is there, then this step is going to be the reversible one. So it depends on the need. It depends on that demand. So when it occur in glycolysis, then this particular enzyme is transferring the phosphate to ADP, forming ATP. So the reverse reaction can occur, but the reverse reaction is taking place in the gluconeogenesis. So in glycolysis, this is how it is going to be. In glycolysis, it is going to add the phosphate into the ADP. It will form ATP. So that is the catch that you have to understand for this particular enzyme. Is that clear? Is that clear? So this is important. I hope it is clear to you. So now this pyruvate, if this pyruvate will get oxygen, then it will enter in the link reaction and all. If it will not, then fermentation. We will discuss the fermentation first. So you can see glucose, 4 ADP, 2 inorganic phosphate, 2 NAD, 2 ATP are used. You are getting at the end 2 pyruvate, 4 ATP, 2 NADH, 2 ADP. This is important. Okay. So from glycolysis, what we obtained? In simple words, this reaction is from NCERT. Do write it. You are getting 2 pyruvate. You are getting 2 NADH2. And you are getting, see, 4 ATP. In totality, 2 ATP. But how come 2? Ma'am, we got 4. So again, the same funda. In preparatory phase, beta, you used 2 ATP. In payoff phase, you made 4 ATP. So net gain, when I will see the total here, so I have to, whatsoever money you put in investment, after from profit you will subtract that 2 now, then you will say ki this is my profit. So this is 2 ATP. That is why it is 2 ATP. Is that clear? That is why it is 2 ATP. Is that clear? So next is beta fermentation, a very easy topic. The NCERT paragraph or the NCERT diagram for fermentation is very important. Okay, that diagram is very, very important. I hope this diagram, it is very, very important here. Definitely question will come. Okay, definitely question will come. So you are having pyruvic acid. See, in the bacteria and in muscle cells, animal muscle cells, you know that pyruvate can form the lactate. Pyruvate can even form the lactate. Just in our muscle, when we are doing some exercise excessively, when we are using our muscles in that way, you people know that, right? That muscles, they'll be like, insufficient oxygen is there. So pyruvate, right? Pyruvate is like oxygen is not available. Pyruvate will enter in lactic acid fermentation. Pyruvate will enter what? Where? Pyruvate will enter in the lactic acid fermentation. Are you getting my point? Are you getting my point? So this lactic acid accumulation in the muscle cell is actually the fatigue. It is actually the muscle fatigue. So here your NADH is beta. This your NADH will again be oxidized. It's a one-step process. It's a one-step process, and the enzyme used here is going to be, right, this is even given in your book. So this enzyme is important, lactate dehydrogenase. Which enzyme is used here, bacha? This is lactate dehydrogenase. Which enzyme is used here? Lactate dehydrogenase. So NADH2 is reoxidized. Now, another step is alcoholic fermentation. It's a two-step process, right? Here it is heterofermentive. Two different products are there. So for 2 steps, the trick is DR. The 2 steps, the trick is DR. What is the meaning of DR? D means decarboxylation. D means decarboxylation. So here CO2 release is also there. Then comes the reduction. So these are the 2 steps here in alcoholic fermentation. Here also NADH will be reoxidized. NADH will be, it will be reoxidized. So ethanol plus this CO2 will be there. So they are basically what is happening here. So it's a 2-step process again, beta. It's a 2-step process again. First decarboxylation, right? Your pyruvate will be decarboxylated, right? Beta acetaldehyde will be there, and then further it will be reduced to ethanol. So these are the 2 steps, right, bache? So here you people can see this is what we have even in the NCERT. So you have CO2 and ethanol. So that is also a PYQ, in which fermentation carbon dioxide is released, right? So here pyruvic acid decarboxylase is the enzyme, and then alcoholic dehydrogenase is the enzyme. Pyruvic acid decarboxylase is the enzyme, alcoholic dehydrogenase is the enzyme. Rest is that table which you have to remember. So this is something very important, right? This is something which is very, very, very important, right? So after glycolysis, which is common to both, right, aerobic and anaerobic, you will have homofermentive or heterofermentive. What will you have? Homofermentive or heterofermentive fermentation. One type of product, homofermentive, lactic acid, 2 or more than 2 types. So alcoholic may, which is in fungi, which is in higher plants, what is happening? Your pyruvic acid, by using pyruvic acid decarboxylase, magnesium ion and TPP, thiamine pyrophosphate, it is forming what? So do remember the NCERT here. You will get acetaldehyde and CO2. So from 3 carbon, it will become 2 carbon containing compound. So carbon dioxide is released. So DR, you want to be the doctor. So it's DR, decarboxylation and reduction. So acetaldehyde, here also NADH will be reoxidized. So alcohol dehydrogenase, then ethyl alcohol will be there, and then beta energy will be released. Energy will be released. right? And here in the case of lactic acid fermentation, lactate dehydrogenase is the enzyme, and here zinc is used as a cofactor, and here you have FMN, that is flavin mononucleotide. So that's what you need to remember. It's flavin mononucleotide. That is what you're supposed to remember. Flavin mononucleotide, that is important. So here beta lactic acid is formed, right? So here the complete glucose is not oxidized, and here you know that when lactic acid is accumulated, fatigue will be there. So in this both type of fermentation, less than 7%, what will be there? Less than 7% of energy in glucose is released, and not at all the energy is trapped in the form of ATP. Less than 7% of energy in glucose will be released. So this process is hazardous. Either acid or alcohol, both are bad. And beta, this is given in NCERT, right? It is something that I discuss in microbes chapter as well, that yeast poison themselves to death when the concentration of alcohol reaches about 13%, right? So yeast will poison itself to death when alcohol concentration which reaches about 15%, because then it won't be able to reproduce. So the beverages which form due to the fermentation, they are having less alcohol from 4 to 13%, right? So one molecule of glucose yields only 2 molecules, those formed in glycolysis, when it is the fermentation in an organism. When what is there? When fermentation is there, so 1 molecule of glucose yields only 2 molecule of ATP. So this is regarding your fermentation process beta, right? So overall here also I would like to specify a few other pointers. जैसे ये देखो aerobic or anaerobic. So aerobic respiration is also known as intermolecular. This one is also known as intramolecular. So anybody who can explain this? Anybody who can explain this? Yes? Why this aerobic respiration is intermolecular respiration? Done? Quick, anybody? Yes? देखो बेटा, intermolecular, inter means in between. So oxidation reduction is going on in between, I would say in different molecules, ठीक है? जैसे and intramolecular, one molecule is getting, right here. What is happening in the intramolecular respiration? You will see oxidation reduction is taking place within the same molecule. Jैसे glucose is making the ethanol and then it is releasing the CO2. But here in the case of aerobic respiration, you people will see in the case of aerobic respiration, जैसे glucose and oxygen, two different things are involved, right? So electron transfer will occur from one molecule to another. That's what you have to see. So in aerobic respiration, NADH is oxidized to NAD vigorously. Matlab it will be oxidized vigorously here. This is going to be, right, very slowly. So these 2 points from aerobic and anaerobic respiration, they are important. And about the energy uptake also I told you. Next topic that we have to discuss is the Krebs cycle. But before Krebs cycle, we are going to discuss the link reaction. Is that clear? We are going to discuss the link reaction, the gateway reaction, right? The reaction which is linking this glycolysis and the Krebs cycle. Now, next process, where is it occurring? It is occurring in the mitochondria. First, let's discuss the mitochondria, powerhouse of the cell. है ना? What is mitochondria? Powerhouse of the cell. Yes or no? Yes or no? Again, when we talk about mitochondria, what do we see in the mitochondria? What do we see in the mitochondria? Here also you are having outer membrane. Here also you are having intermembrane. And in between we have intermembrane space. Beta, be it chloroplast or mitochondria, outer membrane is having porins, right? The protein channels, the pore-like structures, right, through which things will pass. So outer membrane, even in chloroplast also, it is less permeable. Outer membrane, bacha, it is less permeable. And if I talk about the inner membrane, it is more permeable. It is, I would say, selectively permeable. It is selectively permeable, but when you people talk about the outer membrane, it is more permeable. That's what we have to keep in our mind. Now here inside, what else do we have? We have matrix. And we know that here you will have circular DNA. What do you have here? Circular DNA. Here you have 70S ribosome, right? 70S ribosome is there. Now, bacha, here, these finger-like structure, yeah, I would say these infoldings here, they are known as cristae. The cells in which respiration is high, the cristae will be more. And here you are also going to find the structures. These are the structures that we have to discuss here, right? These are the structures, these lollipop-like structures which we have to discuss here. What are they, bache? They are the oxysomes, right? What are they? They are the oxysomes. Is that clear? Is that clear? So these are the oxysomes that we have to discuss here, right? We discuss the same in the case of chloroplast as well today. So oxysome, they are also known as F0, F1 particle, right? But this is also known as elementary particle, right? We also call it as elementary particle, and they are, this is also the ATP synthase, ATP synthase. And here, this part is basically F0, this is base, and this part, beta, this is head plus stalk, this is F1, and it will be in matrix. This one is in inner mitochondrial membrane. So before discussing anything, right, it is traversing there. So before discussing anything, the structure here is important. So it is again a semi-autonomous organelle having circular DNA, 70S ribosome. It is a powerhouse of the cell because it is involved in ATP formation. So this matrix is the site, bacha, where Krebs cycle will occur. This matrix is the site where what is going to happen? In this matrix, bache, what will happen? There will be the Krebs cycle. What will be there? There will be the Krebs cycle. Okay, in the inner mitochondrial membrane, you people will see electron transport chain will be there. Okay, right? So now what is happening? Why are we discussing that? It's simple. After glycolysis, pyruvate will form. Now that pyruvate, it is in cytoplasm, it has to enter here. It has to enter here. Where? It has to enter here inside the mitochondria because the link reaction will occur. So what is going to happen further? What is going to happen further? In mitochondrial, basically, with the help of specific carriers protein, specific channel proteins, with the help of specific carrier proteins or the specific channel proteins, pyruvate will be transported into mitochondria, right, when sufficient oxygen is available with the help of specific carrier proteins. Now, once it is in mitochondrial matrix, right, the oxidative decarboxylation will occur. So this is what we discussed, bacha. So oxidative decarboxylation, the word here is decarboxylation, means CO2 will be released. Today, I will also explain why in NCERT 38 ATPs are given and that is incorrect. All the things we will discuss in the detail here. So pay attention. So oxidative decarboxylation means CO2 release will be there in this step. So this is the step which is also known as link reaction. This is the step which is also known as gateway reaction. So that is what you need to remember. That is what you need to remember. Is that clear? That is what you need to remember. Is that clear? Now, in mitochondrial matrix, what is happening? Your oxidative decarboxylation is taking place. It's a link reaction. It is linking your glycolysis and Krebs cycle. So you have 2 pyruvate. Here 2 coenzyme A will be used. 2 NAD will be reduced. Basically, pyruvate dehydrogenase complex is the enzyme that you are using. Magnesium is the co-ion there. Beta, here 2 acetyl coenzyme A will form. So like in simple words, if I have to tell you, there are 2 pyruvate here, right, 3 carbon containing. So that 2 pyruvate will give you 2 acetyl coenzyme A. So acetyl coenzyme A is 2 carbon containing. So it is the CO2 release is there. So 2 CO2 will also release, and 2 NADH will also form. So the enzyme used here is pyruvate dehydrogenase complex. That is again an important PYQ, right? So coenzymes are TPP, that is thymine pyrophosphate, thymine pyrophosphate. Then beta you have FAD, flavin adenine dinucleotide. Flavin adenine dinucleotide, and then you are having lipoic acid. Then you are having lipoic acid. Is that clear? Is that clear? So oxidative decarboxylation is there. CO2 will release. It is the link reaction. So 2-pyruvate, it is going to give 2-acetyl coenzyme A. So 2 coenzyme A are used. 2-acetyl, 2 coenzyme A are used. 2 NAD are used. So 2 coenzyme A, 2 CO2, 2 NADH2. So this is going to happen here. So that is what you need to remember, right? Right. So after that, this is the acetyl coenzyme A. So 2 acetyl coenzyme A, 2 NADH2 will form, 2 CO2 will release. So please keep it in your mind. Kindly keep it in your mind. Now next point is what? Now beta, next is the Krebs cycle. Next is the Krebs cycle. Again, it's a cyclic process. It is also 10-step process, but some steps are unstable, which we do not discuss. It is amphibolic. It is a dual pathway, catabolic as well as anabolic. And where is it taking place? It is occurring in mitochondrial matrix. So who discovered that? Hans Krebs. And this process, this cycle is also known as citric acid cycle. Will tell you the reason. This is also known as tricarboxylic acid cycle. It is also known as tricarboxylic acid cycle or TCA cycle. These are the names that we have for this. So you can call it as Krebs cycle, citric acid cycle, tricarboxylic acid cycle or TCA cycle. Now, in mitochondrial matrix, it is taking place. So here you have to remember something. There is a trick. It works for me. I don't know whether it will work for you or not. So this is come in and kick, scooty, scooter, fastly move on. Come in and kick, scooty, scooter, fastly move on. It's a cyclic process. Overall, it is also having 10 steps, but there are some unstable compounds in between, which we do not discuss, not given in NCERT. In NCERT, even this part is not given, but still they have asked the questions from that. So I'll show you with the proof. So come in and kick, scooty, scooter, fastly move on. That is what we have. Come in and kick, scooty, scooter, fastly move on. That is the trick that we have for this Krebs cycle. So as I said, we are calling it amphibolic. We are calling it as mitochondrial matrix. We are also calling it as Hans Krebs. So come in and kick. So here, firstly, you have citrate, or you can call it as citric acid, a 6-carbon containing compound. Then you have isocitrate. Isocitrate, again a 6-carbon containing compound. I is telling us this C. And kick, it's telling me alpha keto glutarate. That's a 5-carbon containing compound. Then you have succinyl coenzyme A, which is a 4-carbon containing compound. Then you have succinate, which is again a 4-carbon containing compound. Then you have fumarate, again as I said, a 4-carbon containing compound, malate and oxaloacetate. Again a 4-carbon containing compound. So first of all, remember this, right? First of all, remember this. In today's lecture, we are going to remember everything. So what are we saying? Come in and kick, scooty, scooter, fastly move on. Citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme A, succinate, then you have fumarate, then you have malate, and then you have oxaloacetate. This is what you have to remember. Now, one more thing that from this part itself you can remember. It's a cyclic process, beta. It's a cyclic process. So regeneration is required. It's a cyclic process, so indeed regeneration is required. So come in and kick, scooty, scooter, fastly move on. So here you can see 6 carbon, 6 carbon, 5 carbon. If you are getting something like from 6 carbon, if it is 5 carbon, means CO2 is released, decarboxylation. Then it is CO2 is released. 2 steps of decarboxylation. One by one, we will break it, we will understand it. We will break it, we will understand it. So come in and kick, scooty, scooter, fastly move on. You write it, you repeat it at least 10 times. If you are learning this for the first time, minimum 10 times you have to repeat it. Trust me, everything will be on your fingertips. So we are saying come in and kick, scooty, scooter, fastly move on. So citrate, isocitrate, alpha ketoglutarate, सक्सिनाइल कोएंजाइम है, सक्सिनेट, फ्यूमरेट, malate, oxaloacetate. ठीक है? Now, one by one, as I told you, we are going to learn that. Overall, what do you see in this particular Krebs cycle? Here we will see 2 steps of, that's what we need to remember as well. 2 steps of decarboxylation. So you people should know in which step decarboxylation is taking place. Here, there will be 3 steps in which beta NADH2 form. 3 steps in which NADH2 form. There will be 1 step where your ATP will form, ATP/GTP form, and this is again substrate level phosphorylation. This is again substrate level phosphorylation. Please understand that. There will be 1 step where FADH2 form. This is what we have to understand here. This is what we have to understand here. Is that clear? So come in and kick, scooty, scooter, fastly move on. So there will be citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, malate. This is the first thing. Then we discussed here that there will be 2 steps of decarboxylation, 3 steps in which NADH2 will form, 1 step in which ATP/GTP will form, 1 step where FADH2 will form. Is that clear? Now, here, substrate entrant means which substrate will enter here in this particular process. It's a 2 carbon containing acetyl coenzyme A. Now, substrate acceptor, yeah, primary acceptor here. Yeah, who's going to accept this? Who's going to accept this? This is going to be oxaloacetate, OAA, 4 carbon containing compound. This one, short form OAA, oxaloacetate. First stable compound, that is your 6 carbon containing citric acid या citrate. So bacha, that is why you are calling it as citric acid cycle. Why? Because the first stable compound here, जैसे ये देखो, acetyl coenzyme A is the substrate entrant, which is 2 carbon containing. Oxaloacetate is the substrate acceptor. It is the one with which your, what is the meaning here? It is the one with which your acetyl coenzyme A will bind. Then citrate is the first stable product here. This one is the first stable product. Then we will talk about all the enzymes and everything. First, try to remember that. Quick, everybody. Quick. Entrant, acceptor, first stable product. The cyclic process is in front of you, and as I said, this is going to be amphibolic as well, because here also breakdown buildup both will be there, right? So overall, that's the process. Then we will understand all these compounds, but I hope this part is clear to you. So this is basically the gist that you have to remember. Chaliye, now let's move to the next part. Okay, so even in the notes, the trick is also given, right? Everything is given, but come to this slide. Ye dekho. As I said, dekho, first of all enzymes required for Krebs cycle are present in mitochondrial matrix. This is important, please remember, are present in mitochondrial matrix, except all enzymes required for Krebs cycle are present in mitochondrial matrix except succinic dehydrogenase. Whatsoever I'm writing like this, they are important for you. They are very important for you. Except succinate या succinic dehydrogenase, right? Succinic या succinate dehydrogenase. I'll prefer to write succinate here. ठीक है? So succinate dehydrogenase. This is what you have to remember. So it basically link Krebs cycle and electron transport chain. It is actually the complex 2. So except succinic dehydrogenase, right? So this enzyme, it is present in inner mitochondrial membrane. That's the beauty of this enzyme. Where is it present, beta? It is present in inner mitochondrial membrane. This enzyme is present in inner mitochondrial membrane, right? Towards the matrix side. Towards matrix side. Done? Sure? So first stable product here is citrate. Okay? So now one by one, let's break it and let's understand it. Just say your oxaloacetate, which is the 4 carbon containing compound, right? This is the first or the primary acceptor here. This is the first or the primary acceptor here. Yes or no? This is the first or the primary acceptor here. Yes, everyone. So oxaloacetate, you have acetyl coenzyme A. This is very important. Please pay attention. This is the acetyl coenzyme A. So here you will also see water is also there. Enzyme used is citrate synthase. It is not synthetase. There is a lot of difference in synthase and synthetase. It is synthase. Now you will have citrate here, which is a 6 carbon containing compound. And here you will see, because it is in ionic form, I am writing look at the structure now. Yes, everyone. Look at the structure now. Can you see that? Sure. Sure. Any doubts here? So now look at the first stable product. Don't you think? Yes, please pay attention. COH means this is an ionic form, otherwise 3 carboxylic acid. That's why the Krebs cycle is tricarboxylic acid cycle. That's why you are even calling it as citric acid cycle. So this is what you have to remember here. Now, bacha, this citrate, okay, this citrate ultimately there is a step in between, right? There is an unstable step in between. Cis aconitate forms. And then again, there is the formation of isocitrate. This part, it is not even in the trick, right? But it is a very old PYQ. This is a very old PYQ. That's why I am explaining it here, right? It is a very old PYQ. That's why I'm explaining it here. So this is what you need to remember. So your oxaloacetate is forming citrate, then there is cis aconitate, then there is isocitrate. So enzyme used here in both the cases is aconitase. So here H2O will come, here H2O will leave, but enzyme in both the cases aconitase. So this unstable step we do not discuss, but it is a very old PYQ, right? It's a very old PYQ, so that is why we are discussing it here. Is that clear? Is that clear? Sure. One second, beta. This part here, I made the mistake. Water will leave here. Water will enter here, will be used here in this process. This is what we have. Enzyme is aconitase. So oxaloacetate, citrate, isocitrate, in between there is an unstable compound also. This cis aconitate is there. So you will solve the PYQs, you will find this MCQ. Now, what about this isocitrate? Beta, this isocitrate is giving us what? This part is clear. Now, this isocitrate, right, come in and kick. So we have alpha ketoglutarate here. And we know that it's a 5-carbon containing compound. So CO2 is released. Meanwhile, here what is happening? Your NAD will be reduced. You will have NADH2. So here, beta, here what is going to be the case? Here what is going to be the thing? The enzyme used is going to be isocitrate dehydrogenase. What is the enzyme here? It is going to be isocitrate dehydrogenase. Now, next step, your alpha ketoglutarate. It is going to give you succinyl coenzyme A. So you know that here also beta, it's a 4-carbon containing compound. CO2 will go out. Here in this step also, this is the second step where you will see your NADH, right? Your NAD is reduced. You will have NADH2. And this is basically succinyl coenzyme A. Here the enzyme used is going to be alpha ketoglutarate dehydrogenase. What's the name of enzyme here? Alpha ketoglutarate dehydrogenase. What's the name of enzyme here, students? Alpha ketoglutarate dehydrogenase. So succinyl coenzyme A is going to make succinate. So that is what you have. And here in this particular step, beta, this is the step where substrate level phosphorylation is occurring. I'll tell you how come the substrate level phosphorylation here, and here in this particular step, that's how you write. Okay, ADP, either you write like this, ADP, GDP, they'll form ATP, GTP, and in another way also I'll explain it. So this is the thing that you have to remember. These reactions which are there in front of you, they're very, very, very important. Right, so succinyl coenzyme A will form. Right, okay, one second, alpha keto dehydrogenase, theek hai. My bad. Succinyl coenzyme A will form. So here you have added the coenzyme A also. That is why you have succinyl coenzyme A. So next is the succinate. Okay. Done? Any doubt here? So here the enzyme used is succinyl coenzyme A. Synthetase is the word. Synthase or synthetase, they are two different things. We'll let you know about that too. Succinyl coenzyme A synthetase or succinate thioester is also the name that we will give it. But it is the step of substrate level phosphorylation. Now you have to tell me, right, this is a PYQ. So these reactions, question will come from this part. But why do we call it as substrate level phosphorylation? Quickly tell me. Why do we call it as substrate level phosphorylation? Quick. Yes, everyone. Yes, everyone. Anybody? But in NCERT, if you will read this particular part, they have clearly mentioned that there, right, the GDP to GTP synthesis and then ATP to ATP synthesis is a coupled reaction. What is the meaning? Means here GDP to GTP will form. How? Inorganic phosphate will enter. Then immediately that GTP breaks and forms the ATP. Right? That is the meaning of having the coupled reaction. Is that clear? Is that clear? Right? Yes? Done. Now, here you might think that why it is the substrate level phosphorylation, because it is the succinyl coenzyme A, ma'am. It doesn't have any phosphate to it. So how can you say that? Beta, basically this succinyl coenzyme A, it is a high energy compound. What's that? It's a high energy compound, right? So when it's thioester, when its thioester bond breaks, it provides energy for adding inorganic phosphate to ADP. Let's say we are considering ADP. So that is why, beta. So substrate level phosphorylation, जैसे in the glycolysis we have learned that the direct transfer of substrate from phosphate from substrate to the ADP was there. Here the substrate itself is providing the energy so that we can, because it's high energy bond. When it is having this thioester bond, which is the high energy bond, right? So what is this thioester bond? It is the high energy bond, beta. So when this bond it's breaking, it releases the energy which is required for doing this, right? Which is required for this particular process here. So that is why students, this is what you have to keep in your mind. So that is why here this is the substrate level phosphorylation. So this is the point that you have to remember. This is a very, very, very important point. Okay. So after that, your succinate is going to give you what? The succinate is going to give you fumarate. Right? So this is again a 4-carbon containing compound, and here the enzyme used is going to be succinate dehydrogenase. And I told you already, this is not present in the mitochondrial matrix. It is in the, right, it is present in, yes beta, it is present in inner mitochondrial membrane. It is present in inner mitochondrial membrane. It is not present in the mitochondrial matrix. So succinate to fumarate will be there. Succinate to fumarate will be there. Are you able to understand that? Succinate to fumarate will be there. So if you guys are having any doubt here, you people can ask me. Right? Right? So succinate to fumarate will be there. Sure? Done? So synthetase enzyme, then fumarate, then after this fumarate you have malate, right? Fumarate to malate, malate back to oxaloacetate. So succinate to fumarate, right beta? When there is the formation of succinate to fumarate, here in this particular step, what is happening? Your flavin adenine dinucleotide is getting reduced to FADH2. So this step is very important, because here we will see that this is the one which is going to link it with the electron transport chain, then fumarate to malate, malate to oxaloacetate. So when the malate to oxaloacetate will also form, so you will again see NADH2 formation. So 3 steps where the NADH2 form, they are discussed here. The CO2 release steps are also discussed here, right? So the step where substrate level phosphorylation will form, where FADH2 will form, is also discussed here. This is basically a very important topic. Tell me if there is any doubt. Tell me if there is any doubt. Guys, quick. And make sure you hit the subscribe button as well. Okay? Done? So this is the complete process. So oxaloacetate regeneration is very important. Why beta it is very important? Because it is a cyclic process. So you can see, oh, I missed the enzyme malate dehydrogenase here, fumarase here. Baki succinate dehydrogenase location is inner mitochondrial membrane towards the matrix side. So succinate to fumarate will give you FADH2. So this is the complete cycle which I have added here, right beta? So there are 2 diagrams. There are 2 diagrams which I have added. All the PYQs are also discussed there. So you people can see citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme, succinate, fumarate. So this reaction here is coupled, right? So as per NCERT, NCERT is like GDP will form GTP. Immediately that GTP will break, right? Will break, and your ADP is going to accept the inorganic phosphate and ATP will form, right? Right? So this is what we have. This is what we have. This is what is given in NCERT. So this is about the Krebs cycle, beta. So synthase and synthetase, these are 2 different enzymes, right? Synthase will not use the ATP, but synthetase will. So you can see Krebs cycle. It is on the name of German biochemist Sir Hans Krebs, who discovered it and won the Nobel Prize for his work in 1953. So site is mitochondrial matrix. It's aerobic. It's amphibolic in nature. First product is citric acid. That's why the cycle is citric acid. So structure here, because it is having 3 carboxylic group, so it's tricarboxylic acid cycle. So 2 steps of decarboxylations are there. So these reactions are important to remember. 1 step of substrate level phosphorylation, succinyl to succinate, that is also written there. After that, you can see this is NCERT note. During conversion of, that's how you should make your notes, guys. So during conversion of succinyl coenzyme to succinate, a molecule of GTP is synthesized. This is substrate level phosphorylation. So in a coupled reaction, bacha, GTP is converted to GDP. GTP will form, then it is converted to GDP, and then there is the synthesis of ATP from ADP. NCERT. So trick is also added. Right, everything is added here. What is formed, what not is, that is also added, beta. Just ATP, CO2, everything is written here. Now, in Krebs cycle, oxidation or dehydrogenation occurs at 4 places. So that reactions are also given. That reactions are also given. Okay, so this is what you have to remember. So this is all about the Krebs cycle, which we have discussed in detail, beta. Baki acetyl coenzyme for fatty acid, raw material for fatty acid, steroid, carotenoid, terpenes, it is. Alpha ketoglutarate can form amino acid glutamate. OAA forms pyrimidine alkaloid. Succinyl coenzyme, you know that it is even for the chlorophyll formation. So baki this step I have already discussed. Done. Baki synthase will not use the, it will synthesize the molecule without using ATP. Synthetase will use that. Now, we will go for terminal oxidation, that is ATC. I will make sure that I'm going to make it very simple, very, very simple for you. I will make it very simple for you, ETC part. So I'll give you a break as well, right? I'll give you a break as well. But before that, let's write. You got glycolysis. First, you have glucose. है ना? This is important. It will give you 2-pyruvate. So, if it is giving me 2-pyruvate, है ना? This is 10 steps. And here, what else we got? 2 ATP, 2 NADH2. Beta, right now I'm just focused here. So if you have 2-pyruvate, how many pyruvate? If you have 2 pyruvate here, how many pyruvate? 2. So there will be link reaction. There will be link reaction, या gateway reaction, या oxidative decarboxylation, or the transition reaction. These are the different names that we use. Here what we got. Here what we got. Because 2 pyruvates are there, so you will have 2 acetyl coenzyme A, obviously coenzyme A also will add. 2 CO2 will release, and here in this step 2 NADH2 form. That's what we have covered. Now after that there will be Krebs cycle. So beta, that will also be 2 times, na? Because you have 2 acetyl coenzyme A. Don't you think so? If 2 acetyl coenzyme A are there, so there will be 2 Krebs cycle. So from 1 Krebs cycle you will got what? Just in Krebs cycle, in 1 Krebs cycle what happened? 3 NADH2 form, 1 FADH2, 1 GTP. Chalo, let's simplify it. Let's write ATP and 2 CO2 release. Isn't it? Now, this is from 1 glucose. We are doing it from 1 glucose. So, second KC will do what? Same thing. 3 NADH2, 1 FADH2, 1 ATP, 2 CO2. देखो, in totality, 2 CO2 will release here, 2 CO2 will release here, total 4, 2 CO2 will release here, 6 CO2 will be released from one glucose when there is complete oxidation, and we used 6 CO2 to fix 6 CO2 fixation to make one glucose. So here you can see one glucose is releasing 6 carbon dioxide, number 1. So overall, can I not say that that if I have one glucose is giving me 2 ATP, 2 NADH2, here 2 NADH2 again from link reaction, and total 6 NADH2 plus 2 FADH2 plus 2 ATP from Krebs cycle. Can I not say that? So this one is from glycolysis, this one is from link reaction, this one is from Krebs cycle. So when you read NCERT, what you people see, that here they have mentioned that from after glycolysis you are getting 8 NADH2. So sometimes they count them together, but you know this is what we got. This is what we got. Is that clear? Any doubt so far? If that is the case, right, this is important, beta. Whatsoever I am writing here, this is very important. This is very, very, very important. Please pay attention. Any doubt? Done? So now we have to reoxidize these NADH2 so that our process can continue. So what will happen? Now comes the terminal oxidation. Now we will talk about the electron transport chain and the final oxidation here, right? Oxidative phosphorylation here. We have to reoxidize these NADH2. We have to use their electrons. We are going to basically make the ATP from that. So this is something that you have to remember, that one glucose in totality is giving you around 10 NADH2, 4 ATP, 2 FADH2, or link reaction Krebs cycle. Individually it should be clear that what exactly is happening. Now what's the next point, right? We have to get ATP out of that. So here I am writing the actual concept. What am I doing, bacha? Here I am writing the actual concept. 1 NADH2, when you pass it to ETC, electron transport chain, it will give you 2.5 ATP. And 1 FADH2, when you will pass it in electron transport chain, it will give you 1.5 ATP. Are you getting my point? 1.5 ATP. Are you getting my point? This is the actual concept, right? This is the actual concept. This is the correct concept, I would say. Now, in old NCERT, new NCERT mein properly changes are not there. In old NCERT, it was given that 1 NADH2 gives you 3 ATP and 1 FADH2 gives you 2 ATP. Matlab they are like ki chalo yaar, why to write points, write like this. So this is incorrect concept. This is incorrect. But yeah, it was there in old NCERT. People used to we used this thing to calculate our answer, but now it is an incorrect concept, and now it is clearly mentioned that if something is given incorrectly in the NCERT, don't go for that, go for actual concept. This is the actual concept. So this is given in old NCERT. Okay? This is given in old NCERT. Now, how that electrons will form, how that ATP will form, that part I'll come later. But here I would like to explain something to you. जैसे if this concept, if I apply actual concept, है ना? चलो, I'll write it separately because this slide we need है ना, this slide we need, so I will write it separately. ठीक है, so what I just said कि glycolysis is giving me 2 ATP to NADH2. That's what I said. So if I will send it for ETC, if I go as per actual concept, to 2 into 2.5 will be 5 ATP. So I am going to have total of 7 ATP from glycolysis after terminal oxidation. Right? All our assumptions will go one by one. Now, link reaction is giving me what? 2 NADH2. So that NADH2 will give me again 5 ATP. Now, Krebs cycle, I'm taking total 6 NADH2. I'm multiplying with that 2.5. 2 FADH2, I'm again multiplying it with 1.5. And already we have 2 GTP or 2 ATP there. Okay? So this is going to be 3 ATP. This is going to be 3 ATP. And what about this, beta? What about this? Hanji, quick. 6 multiplied by 2.5 is going to be what? 6 multiplied by 2.5 is going to be what? 15 ATP. So from this Krebs cycle, 15, 16, 17, 18, 19, 20 ATP you get after terminal oxidation. This is 25. 25 plus 7, what is it going to be? 32. Am I right? So we write it like 32. Once again, I think I made one mistake somewhere. 20. 32. है ना? 32. So total 30 ATP. So we 32 ATP. So we write it like this, beta. Here you got 32 ATP, or we also write it like this. Either it can be 30 or 32 ATP. It depends on shuttle system, which we will discuss further. This is the correct concept, right? The shuttle system thing we will discuss. This is the correct thing. Now, if I go as per old NCERT, because if you will solve PYQ, you are going to find the answers as per that. So old NCERT says कि glycolysis is giving me what? 2 ATP, 2 NADH2, which I am going to multiply with 3. So I am getting 6. So total 8 ATP. So when you solve the old PYQs, you will get the answer as 8. But now it is incorrect. Earlier also it was incorrect, but it was given in NCERT like that. So we used to discuss that. Now, in the link reaction, they are like 2 into 3. Again, 6 ATP. Again, Krebs cycle, 6 into 3, 2 into 2 and 2. So you know that 18 ATP, 4 ATP, 2 ATP. So as per the old thing which is given in NCERT, which is incorrect as well. So for its Krebs cycle itself is giving you 24 ATP. Just imagine 24 here, है ना? 24 here plus 6, 30 plus 8, 38. So here as per old NCERT 38 ATP, or it is what we used to write 36 or 38 because it depends on shuttle system. So but actually it is 30 or 32, and if you don't consider the shuttle system, you can consider this. So in the NCERT, when you read the paragraph, at the end they have mentioned that 38 ATPs are given. So this is incorrect concept. So now I told you how come 38, how come this 30 or 30, how come 32? This is what I told you. Now I will explain the logic behind, but that is the funda. So this is incorrect. It is given incorrectly in the NCERT. We are not going to accept it. Okay. So this is the thing that you have to remember. So if you will solve some PYQs, you will get, right? You people are definitely going to get this point, right? But You don't have to accept it. Okay? So terminal oxidation is what we are going to discuss. So we are going to discuss about certain electron complexes also. So ETC, trust me, I will simplify it. So now you tell me, do you need a break, or let's finish this chapter, then let's have a break? What say? Then let's finish this chapter and have a break. Let's finish it, hai na? Chalo. Terminal oxidation, what is the site of terminal oxidation? Inner mitochondrial membrane. So it is basically the last step of the aerobic respiration, beta, right? Here, the transfer of electrons and protons of these coenzymes, that is NADH2 and FADH2, will be done. Their electrons and protons will be taken, right? So we know that they are produced during glycolysis, Krebs cycle, and oxidative decarboxylation. So ultimately, their electrons and protons will be taken and they will be transferred to the oxygen. It is the terminal acceptor. What is this oxygen? So this oxygen is the terminal acceptor ultimately. What's that? It is the terminal acceptor ultimately. So that's what you have to remember. So we are going to discuss two steps here. So one is your electron transport system, that is ETS or ETC, electron transport chain. And then you have oxidative phosphorylation. Now you are well aware of photophosphorylation. You know what substrate level phosphorylation is. This is oxidative phosphorylation. So here beta, you people will see a gradient is going to establish, and that gradient will help in the ATP formation. So here beta, we have to discuss. So here we will discuss about certain complexes. So we will talk about complex 1 to 4 in the case of ETC. Right, and when there is the oxidative phosphorylation, you will talk about the complex 5. This is what we have to discuss. So if in the paper, do read the statement carefully, So if in paper they are asking electron transport chain and oxidative phosphorylation, if in the paper that statement is including both oxidative phosphorylation and ETC, so how many complexes are there? 1 to 5. 1 to 5, but in ETC you have 1 to 4, and then in oxidative phosphorylation you have 5th complex. So ultimately what is happening? Nothing differently here. All the reduced hydrogen acceptors, right, NADH2, FADH2, we will pass them to this electron transport chain where they have to release their hydrogen. They have to reoxidize so that we can use them again. So this electron transport is down to the energy gradient. Just we discussed the redox potential, so down to the energy gradient it is. So this is why this is used for making ATP. So this is why this is the oxidative phosphorylation, right beta? But this direct transfer of electrons to oxygen is not possible, bacha. Direct transfer of electrons is not possible because of the redox potential difference. That's why we have certain complexes which are going to do that. So here you can see these are oxidized through electron transport system and electron are passed to the oxygen. We cannot do that directly. Why? Because of the redox potential difference. So that's what we have to understand. So terminal acceptor for electron and proton is oxygen. So there are total 5 complexes, but first 4, and where are they present? This is our mitochondria, bache. This is the mitochondria. This is the inner membrane of mitochondria. In the inner membrane of mitochondria, I already told you about the oxysomes. Yes, I already told you about the oxysomes. So if we zoom this part or this magnify this part, so actually in this inner mitochondrial membrane, you people are going to see some complexes are arranged. What will be there? Some complexes are arranged here. That's what you have to remember. So actually here in this inner mitochondrial membrane, you people are going to see these protein complexes here. And let me tell you, this oxysome, which we have already discussed, bache, right? You know, elementary particle, F0, F1, where F0 is in inner mitochondrial membrane, F1 is in matrix. So this is making our 5th complex, ATP synthase. Okay? So NADH dehydrogenase, succinic dehydrogenase, cytochrome C complex, cytochrome BC1 complex, cytochrome C oxidase, and finally ATP synthase. So again, I am writing the name of the complexes. Even if you don't know anything, right? Even if you remember the sequence here of the complexes, your name here of the complexes, that will be more than sufficient for all of you. Trust me on that. That will be more than sufficient for all of you. Is that clear? So complex 1 will be your NADH dehydrogenase, right? NADH dehydrogenase complex. This one is succinate dehydrogenase complex. Succinate dehydrogenase complex. Then beta, there will be cytochrome C reductase, or the name which is given in NCERT for this is cytochrome BC1 complex. The fourth one will be cytochrome C oxidase. Done. So that's how I remember this NADI, UCD. This is basically looks like suicide. Then I remember Cyto2RO. This is my way to remember that. If it works for you, fine. If it doesn't, right, you can simply remember that. Okay. Okay. Done. Sure. Sure, beta. Chali, let's move. So, NADH dehydrogenase complex, right? Succinate dehydrogenase complex, cytochrome C reductase, cytochrome BC1 complex. So, cytochrome C oxidase is also there. Fifth one, which is involved in oxidative phosphorylation, it is your ATP synthase. So, beta, these are 5 complexes. So, it's 18:06. So, let's restart. Let's restart at 6:15. Okay, so let's sit for 10 minutes, and then, because till then you all can process, and then we are going to continue. So you know that ETC, and after that RQ value, there is a flow chart from the NCERT where I will tell you why is it the amphibolic one, because energy part I have already explained, and why I already explained I'll tell you with the help of the ETC. So maximum after that 1 hour respiration will be over, and 1.5 hour I need for PGD. Okay, so maximum by 9 you people will be free. Okay. Chaliye, let's restart, guys. So, wow, I'm on time. I'm happy. So, this is what we have. So, it is NADI, you see the cyto2-RO. So, this is an NADH dehydrogenase, succinate dehydrogenase complex, cytochrome C reductase, which is also known as cytochrome BC1 complex, cytochrome C oxidase, and then comes ATP synthase. Now, you have to understand two routes. Ultimately, this particular thing is for taking electrons and H positive from what? From your NADH2 and FADH2. So we have to know about routes, right? So basically, I am breaking this topic into some parts. So in first part, I told you what is the meaning of terminal oxidation and I told you about the complexes. Now, I will tell you about the two routes of electron transport chain. What am I going to discuss? Two routes of ETC, electron transport chain. So first of all, let's talk about the route number 1. In route number 1, beta, you will have NADH2. Electrons will pass from NADH2. Then you will have route 2. In route 2, we will see that how electrons will pass from FADH2. First point clear? In two routes, we are going to understand this. So, in first route, NADH2 will pass the electron to who? To complex number 1. Then it will pass the electron to complex number 3. And then it will pass the electron to complex number 4. Matlab, it's like passing the parcel from 1 to 3, 3 to 4. This is what you have to remember. Finally, you will have your oxygen, O2. Right? Finally, you have your oxygen. Okay? Look, I'm writing it. Finally, you have your oxygen. In the case of route 2, FADH2, first it will pass its electrons to. First, it will pass its electrons to complex number 2, 2. Then there is complex number 3. Again, then story is same. Complex number 4. And then finally, beta, again, oxygen will be there. Are you getting my point? So 1, 3, 4, 2, 3, 4. 1, 3, 4, 2, 3, 4. 1, 3, 4 oxygen, 2, 3, 4 oxygen. These are the routes. Now, complex 1, complex 3, complex 4 and even your complex 2, complex 3, complex 4, beta, they are, right? They are what? They are basically fixed there. They are basically fixed there. Now, if just a complex 1 is present there in inner mitochondrial membrane, it is a complex, means it is having like other proteins also, right? Other things also. We'll discuss what do they have. Now, 1, 2, 3, 3 to 4. In between 1 to 3, you will have ubiquinone or you can also call it as coenzyme Q. Either you can call it as, just say in photosynthesis, it was plastoquinone. Here you have UQ, that is your ubiquinone, or you can also call it as COQ, that is coenzyme Q. Anything you can use here, that's okay. In between complex 3 and 4, you have cytochrome C. So 1, 3 or 1, 3, 4 is also fixed here, 2, 3, 4 is fixed. So if we have to pass the electrons, we need to have the mobile electron carriers or movable electron carriers. So this is what we are talking. We are talking about the cytochrome C and the ubiquinone here. So this matlab or and you have cytochrome C. So they both are what? They both are mobile electron carrier. They both are, right, movable electron carrier. So two are fixed and then these two will carry electron from 1 to 3, 1 to 3. First, tell me this part clear. If in parts you are going to understand it, it will never be a problem. It will never be a problem. Now, beta, your complex 1. Once again, beta, your complex 1 is made up of FMN, then it is having FES. You should know which one is having iron sulfur, right, which is having iron sulfur protein. It is made up of FMN, right? It is made up of FMN or FES. Complex number 1. Complex number 3 is having cytochrome B, right? In short form, I am writing cytochrome B. Then again, FES, cytochrome C1. B, FES, C1. B, FES or first, B first, C1. Then this one is having copper center A. Then it is having cytochrome A, cytochrome A3. Then there are 2 copper centers here. And then finally oxygen. I know right now it is difficult for you, but if you are studying it for the first time, you might find it difficult. Just allow me 5 more minutes. Everything will be very easy for you. So this is made up, this is having FM and FPS. Complex 1. Complex, if it is complex, means it is not a single thing, it is having many. Now complex 2 is having FAD, FES. Complex 3 is having, again same, it is the same na, cytochrome B, FES, C1. This one is having 2 copper centers, copper center A, then cytochrome A, cytochrome A3, and then you have copper center B. Then finally oxygen is there. Finally oxygen is there. So in between, these are the mobile electron carriers that we have, right? So if you do not know the full form, let me write it here. Let me write it here that FMN is flavin mononucleotide. Flavin mononucleotide, and this FAD is flavin adenine dinucleotide. So this is how you have to understand. 2 complexes, clear? Right, these are the 2 complexes which you have to keep in your mind, right? These are the 2 complexes which you have to keep in your mind. They are important, FM and FES. Now understand that, right? If roots are clear, then everything is going to be easy for you. Come to this diagram. Please come to this diagram. This is your outer mitochondrial membrane, inner mitochondrial membrane, intermembrane space. So here you will see this clearly. You will see this clearly, right? Do not focus how many H positive or something. Complex number 1, complex number 2, complex number 3, complex number 4, complex number 5. That's how they are placed. So complex 1 in membrane, 3 in membrane, 4 there in membrane. This second one, which is your succinate dehydrogenase, it is attached to inner membrane. It is in inner membrane. It is in inner membrane, but more towards matrix side. है ना? Look at this cytochrome C. Look at this cytochrome C. It is more towards intermembrane space. That's something you have to remember, right? One more diagram to show you this. Look at this. Complex number 1, complex number 2, complex number 3, complex number 4. So second one attached to inner membrane, but it is more towards the matrix side. Now complex number 4, it is attached to inner membrane, it is mobile, it is movable. This is your ubiquinone or quinone. This is your cytochrome C. This is movable. It is towards intermembrane space. The position clear? This is 5th one. Right now we are talking about ETC. Right now we are talking about ETC. So our main focus is this part. Right now we are talking about ETC. So our main focus is this particular part. Okay, so one more diagram here. Now you must be thinking, ki madam, why that FM and FES? Come to NCERT. So this is the third part of the ETC. First part, I told you about the definition or the complexes. Second part, I told you about the roots. Third part, right? Third part. In third part, I'll be discussing the diagram of NCERT. So you have complex number 1, which will carry the electrons from NADH2. So this is obviously matrix side, inner mitochondrial membrane, intermembrane space. 2 electrons will pass to FMN. Dekha? Now diagram will be easy because I break, right? I am basically breaking this topic so that you can understand. FMN, FES. So complex 1 is made up of FMN, FES. Electrons are passed to ubiquinone, and here again just like photosynthesis, it is going to form UB hydroquinone. The Q cycle will operate here as well. A little bit of idea I'll definitely give you. Then this will pass the electron to complex number 3 because one root is 1, 3, 4. One root is 1, 3, 4. So complex 3, cytochrome B, FES, cytochrome C1. After that, it will pass the electron to cytochrome C. Told you, mobile electron carrier. Then it will carry it to the complex number 4, which is having copper center A, then cytochrome A, A3 and copper center B. Then finally, oxygen, number 1. Now, let's take the story of FADH2. FADH2, which is basically the second complex, which is basically an enzyme of Krebs cycle, which is basically linking your Krebs cycle and etc. What will happen here, beta? Here, FADH2 will pass its electron. FADH2, so you can relate. It is having FAD. Then FES, then story is same. ubiquinone, UB hydroquinone, cytochrome B, FES, C1, and finally again to the fourth complex, right? And meanwhile, you can remember 1 is pumping 4 proton, 3 is pumping 4 proton, 4th is pumping 2 proton, but your FAD is not pumping any proton. Why and how, that we have to discuss. So if this part of the, right, if this part of the topic is clear, right, if these roots are clear, beta, that diagram of NCERT will never ever be a pain for you, right, will never ever be a pain for you. I am telling you, I am making it very easy for you, right? So what is happening? NADH2 is passing its electron to complex number 1, which is having FM and FES. Then through ubiquinone, it will pass to complex 3, which is made up of cytochrome B, FES, cytochrome C1. Then cytochrome C, mobile electron carrier placed more towards the intermembrane space, will pass it to complex 4 having copper center, and then cytochrome A, A3, and finally oxygen. Come to this part, FADH2 is passing its electron to complex number 2, FAD, FES, then ubiquinone, then cytochrome B, FES, C1, then cytochrome C again, right, mobile electron carrier, and then again copper center A, then cytochrome A, A3, cytochrome B. Is that clear? Is that clear? Then finally oxygen. Roots clear? Beta, this is what you have to remember. Even up to this part, if you can remember everything, then trust me, this topic is not at all going to be difficult for you, because this is how the electron form. So always remember, NADH is going to give its electron to complex 1. FADH2 is going to its electrons to complex 2. Now, ubiquinone, which is also the coenzyme Q, it is lipid soluble. Important. This is beta can accept 2 electron, 2 proton at a time. But it will transfer only 1 electron to complex 1. And when it is taking the electron from complex 2, then obviously, right, basically, it is ubiquinone. Its work is to, you know that, transfer the electrons from complex 1 to 3 and complex 2 to 3. That's its job. That's its job. It's a mobile electron carrier present in between 1 and 3 or 2 or 3. It has to pass the electrons like this. This is an important information, right? Important information. So it can accept 2 electron and 2 protons at a time. And obviously here also beta the story is going to be same. UQ will form UQH2. You can either call it as UB hydroquinone or you can simply call it as ubiquinone. Right? In the photosynthesis part also, we have discussed this particular portion. We have covered this particular portion. Now, it can carry 2 electron and 2 protons at a time. So that is why we are saying it can transfer electrons from complex 1 and even from complex 2 and even from complex 2 to complex number 3. Now, cytochrome C, which is water soluble. So don't you think this information is very important? So ubiquinone is what? Lipid soluble. Again, mobile electron carrier. Cytochrome C is also water soluble. Again, electron carrier, mobile electron carrier. Right. So cytochrome C. So it lies in intermembrane space. So it is, where is it placed? Loosely attached to the outer surface of inner membrane, NCERT language. So it is towards the intermembrane space. It is loosely attached to the outer surface of inner membrane. So it also transfers the electron from 3 to 4. Their role should be clear, but it can carry only 1 electron at a time. So it's like a person is there which is carrying 2 electrons and 2 protons. Let's suppose you are doing a job, right? You might have seen, right, on the construction site. Let's suppose a person has picked up 2 bricks and the next person is like, I cannot pick up 2 bricks. I can just pick up 1. So So that person has to keep it somewhere na, so that it can pass in next cycle or can revert it. Something like that is happening here. So ubiquinone can accept 2 electrons and 2 protons at a time. But cytochrome C can go for only 1 electron at a time. So they have the high mobility, these 2 electron carriers, so they quickly shuttle, quickly move between the complexes. So roots I have already discussed with you all. Now diagrams I have already discussed with you all. Now let's understand the topic. So what is happening? This is full-fledged NCERT. Dekho beta. Don't go for when it is sending 2 electron, when it is sending 4 protons, don't go for this. Remember, your complex 1 is throwing in intermembrane space because we have to make the proton gradient. That is required for ATP. 1 is giving 4 protons, second is giving no proton, third is giving 4 protons, last is giving no proton, 2 protons. So electron from NADH, which is produced during TCA, they'll be oxidized by complex 1 NADH dehydrogenase. Electrons will be transferred to ubiquinone, which is located within inner membrane. It gets reduced, it will form ubiquinol. Yeah, ubiquinone will get reduced ubiquinone or ubiquinol. So this is then oxidized with the transfer of electron. It has accepted the electron, it will transfer it to complex number 3. But it will transfer it to complex number, right, 3 beta, and then from 3 to it will ultimately pass it to complex number 4 through cytochrome C. So this line of NCERT is saying ki ubiquinol, that is UQH2, is then oxidized with the transfer of electron to cytochrome C. Means ubiquinone, which is again a mobile electron carrier, when it will accept the electron, it is not just accepting the electron beta, it will also accept the proton from the matrix. Right, just like the one we have discussed in the photosynthesis, here also the story is going to be same. So it has to pass the electron to next electron, mobile electron carrier, but in between complex 3 is there. So this is like NCERT is just trying to confuse you. You can simply say 3 to 4. Here who's coming? Here you have ubiquinol. It will pass the electron to 3. Now we are saying it will then oxidize with the transfer of electron to cytochrome C via complex 3. That's what they are trying to say. This is the route na, UQH2, then 3, then cytochrome C, then 4. So obviously this is going to pass the electron to cytochrome C via complex number 3. So do not get confused with this language of NCERT. So what is cytochrome C? A small protein, a mobile electron carrier attached to the outer surface of inner mitochondrial membrane. It is towards the intermembrane surface side. So its job is to transfer complexes between 3 and 4. Very simple topic. Very simple topic. Done. Now, complex 4, it is having cytochrome A and A3, 2 copper centers. Important MCQ. Now, this is it. Then finally the oxygen. Now, same for FADH2, complex 2, then beta to ubiquinone, which will become ubiquinol, right? Yeah, ubiquinol, that will pass the electron to cytochrome C. Same story here. If roots are clear to you, then this is not going to be the problem. And ultimately, what are we saying? Ki when electron pass from one carrier to another, right, they are coupled to that 5th complex ATP synthase beta, right? So this will help in the ATP production. Baki, you know that 1 molecule of ATP, 1 molecule of ADP, FADH2 will give you 1.5, 2.5 ATP. Oxygen is the final or the terminal acceptor here. Then beta, this is as per NCERT. This is as per NCERT. Okay, beta. Now, moving ahead, I will not directly jump to this. I'll come to this part. Here, dekho. Complex 1 is accepting. These are the short notes, brief notes for you. It is accepting electrons from NADH, right? It is accepting electrons from NADH, transfer them to FMN, then to FES, then to ubiquinone. It is pumping 4 proton from the matrix to intermembrane space. Sometimes students ask, why? Because beta, see, though I will suggest you not to enter into that, simply remember, right? Just simply remember that there will be complex number 1, 2, 3, 4. This one will pump 4 protons. This one will pump 0. This one will again pump 4 protons. And this one is going to pump 2 protons. This is what you have to remember ultimately. You can consider it as 4th part of ETC, which you can directly remember. So NADH2, these electrons are having more energy. They are able to pump that much protons. So that is how it works. And moreover, your complex 2 is more towards the inner mitochondrial matrix side. So that's why it cannot pump any proton, right? So see, no protons pumped here. So third one will also, which works by Q cycle, it also pump 4 proton. And fourth one, right, it is also having copper centers, right, heme groups, so it will finally pass the electrons to the oxygen. So here you can see Q cycle, beta, there is no need to write the Q cycle, there is no need to write the Q cycle, but ultimately what is happening when ubiquinone, or you can also call it as a coenzyme Q, when it can accept 2 electron and 2 proton at a time, but cytochrome C cannot accept that, right? So Q cycle solve this mismatch. So when you know this complex 3, it will pass, right, the complex 3 na third complex, right, to which ubiquinone will pass the electron, it is actually having 2 binding sides, right, CQH2, and this is outer, which is near intermembrane side where ubiquinol is oxidized. There is another side which is towards the inner side where this ubiquinone is again reduced. One side where it will be oxidized, one side where it is reduced. So first half mein beta, first half mein that this ubiquinol actually, it binds to that Q0 side, right beta, outer side, it releases its protons to intermembrane space, it will give up 2 electron, 1 electron beta, it will pass further to complex 3, but another electron via that cytochrome BC, right, B1, it will again pass to the inner side of that complex number 3. So that's how it works. So I told you Q cycle is not required for you, do not go for this. Now focus on this part so that I can simplify this topic further. So roots, focus on the roots beta, focus on these 2 roots, everything will be clear. Just I told you 1, 3, 4. This is for NADH2. For FADH2, it is going to be 2, 3, 4. Beta, it has pumped 4 protons, 4 protons, 2 protons. Am I right? It has pumped 0 protons, 4 protons, 2 protons. Now, when NADH2 is passing its electron, they are pumping total 10 protons, where in intermembrane, pumping 10 protons in intermembrane space, yes or no? They are pumping 10 protons in intermembrane space, yes or no? And this one is pumping. See, so its electrons are not having that energy. 6 protons in intermembrane space. That's what you have to remember. 6 protons in intermembrane space. Now, what you have to remember, beta, 4 protons are required for making 1 ATP. That's the next thing. 4 protons are required for making 1 ATP. Done? जैसे वैसे तो 3 protons for ATP and all, 1 is for movement, but in totality for making 1 ATP, that is what you have to remember. That 4 protons are required for basically, 4 protons are required for basically moving, for making 1 ATP. So here it is having 10. So 10 means 4 protons here, 4 protons here, 2 like this. So 1 ATP here, है ना? 1 ATP here, and this one is going to be 0.5 ATP. So that is why it is giving me 2.5 ATP. So old NCERT was explaining it incorrectly. Now here there will be 4 protons, there will be 2 protons, so there will be 1 ATP, there will be 0.5 ATP. So ultimately it is going to be 1.5 ATP. So that is how the story is. That is why, that is what we have here. Have a look. That is what we have here. So roots are important here. Roots are important. How are they pumping it? This is what we need to know here. Okay? So that is why beta, net ATP, which we have already discussed. So in NCERT, in the notes also 3 ATP, 2 ATP are given, but actual is this. So we have to go as per actual. So if we go as per actual, so 2 plus 2 into 2.5, total 7 ATP here. Right? We have already discussed that. Once again, I'm repeating it. So 2 into 2.5, 5 ATP here. Now 2 plus 6 into 2.5 plus 2 into 1.5. So here we know that it is going to be 15. It is going to be 3. 20 ATP here. So that is why beta our total is going to be 32 ATP. I'll tell you why 30 or 32. 32. Now, if I go as per old NCERT, so I will write what? 2 plus 2 into 3. Right. So it is going to be 8 ATP. 2 into 3, 6 ATP. 2 plus 6 into 3 plus 2 into 2. So here it is going to be 24 ATP. So 38. So this is what they have mentioned still in the new NCERT, but this is incorrect. This is incorrect. This one is correct. Right. So on the basis of shuttle system, either it is 30 or 32. Right. Either it is 30 or 32. That is it. Right. That is it. So this is what you have to keep in your mind. And I hope up to this part, everything is crystal clear to everybody. Right. If still it is not, after the class, do check this part again. It will be easy. So let's talk about the shuttle system. Dekho beta. Through glycolysis, we know that 2 NADH form. We assume that these NADH2 will enter in the ETC. It's not like that. In our body, when required, they cannot be used. We assume so that we can understand this process thoroughly. So if there is a shuttle system, just say, let's suppose, in the investment, different type of investments are there. Let's suppose you are putting your money in a fixed deposit. There is a certain interest you are going to get. Same amount of money you are putting in a different mutual fund, but there you got better benefit. It is like that. You have same amount of money. Either you can put it in a fixed deposit or you can put it in a mutual fund as per the risk or what. So from one side you are getting more profit, from another side you are less, but the amount is same. That is something that we have to understand. NADH2 is having the electron. It has to pass its electron to the inner mitochondrial membrane so that they can further, enter in the process. So NADH2, it is having two choices. Two shuttle systems are there. Because NADH itself is impermeable to inner mitochondrial membrane. So it transfers its electron to electron shuttle. So we have two types of shuttle system. One is malate aspartate shuttle system. Another is glycerol phosphate shuttle system. One is malate aspartate shuttle system. Another is glycerol phosphate shuttle system. So malate aspartate shuttle system is in liver, heart and kidney. Electrons will transfer from NADH2 of cytoplasm to NAD inside the mitochondria. Means it is in liver, heart and kidney. So glycolysis may we have NADH2. It is passing its electron to NAD in the mitochondria because itself it is impermeable. This is more efficient. Glycerol phosphate may you imagine. NADH2 of glycolysis. It is passing its electron to FAD na. And now you know if it is passing its electron to NAD, so it will follow this route 1, 3 or 4 wala. If it will pass this electron to this, it is going to follow this route. So obviously this route will pump 10 protons. This will pump 6 protons. So if this is going to be the case, at the end you will get total 32 ATP. But if this is going to be the case, so from glycolysis, from the NADH2 of glycolysis will give you less ATP. So it is in muscle cell and nerve cell where NADH2 are transferring their electron to FADH2. So net gain of ATP is less than malate aspartate. So here you have 32, here you have 30. You can even count that. Baki beta, you have RQ value. Another important topic, volume of CO2 evolved to the oxygen absorbed. So you know that for carbohydrates it is 1. This is the reaction. You people can clearly see 6 CO2 evolved to the 6 oxygen used. The answer is 1. So if RQ value is 1, because it tells us about the type of respiratory substrate. So if RQ value is 1, it is clearly telling us that the respiratory substrate is carbohydrate. Then beta, it can be generally less than 1 also for proteins, for lipids. Just see, for fats it is going to be 0.7. So look at this reaction, stearic acid. This is the formula. 26 oxygen are used, but 18 CO2. 18 by 26, 0.7. Less than unity, less than 1. This is for tripalmitic acid, palmitin acid, given in NCERT, 145 oxygen, 102 CO2. The answer is 0.7. Again, 0.7 is there. For proteins, it is less than unity. It is going to be 0.9 beta. Right, it is going to be 0.9. And when will it be 0? In succulent plants. Right, beta, because they have insufficient oxygen. RQ is 0 when oxygen is insufficient. I am not saying it is absent. I am saying it is insufficient, which is not capable enough, right? So, which cannot oxidize glucose completely, but rather partially. So, see, 3O2, you do not have the value. You do not have, matlab it is like you don't have CO2. 0 by 1 is going to be 0. 0 by 3 is going to be 0. So, RQ0 indicates that oxygen is insufficient. You cannot say it is absent, it is insufficient. Organic acids mein it will go more than 1 beta. Jaise malic acid, you can see 4 CO2, 3 oxygen used, 1.33. So oxalic acid ke liye it is going to be 4. Baki, in the absence of oxygen, RQ value is infinite because oxygen is absent. So 1 by 0, है ना, या 2 by 0. So 0, RQ is 0 when oxygen is insufficient. It is infinite when beta oxygen is absent. That is it. Baki beta, in the NCERT, you discuss this particular portion. You talk about that, right? Balance sheet I have already explained, right? So there are certain assumptions for the balance sheet. So this is what we assume. It's not like that in your body. Why do we say at the end that respiration is amphibolic in nature? Beta, because it is not like that ki glycolysis, it has made pyruvate. Pyruvate will definitely go to the link reaction. It depends on my body's need. If at that point of time, pyruvate is required somewhere else, so my body is going to utilize it there. My body will be like, I'll utilize it there. No need to give it to me. It is like that. Okay, so we assume, there are certain assumptions that there is a sequential, right? These are certain assumptions, beta, that there is a sequential orderly pathway functioning with one more substrate forming the next. So we assume that, right, ki first glycolysis, then link reaction, TCA, ETS is there. That is what we assume. And we also assume that the NADH, which is synthesized in glycolysis, it is transferred into the mitochondria, and that is also undergoing oxidative phosphorylation. Right, beta? So none of the intermediate in the pathway are utilized. Matlab, we are saying ki jaise in the sequence we have studied, everything is going as per that. Right? Nothing will be changed here and there. So this is what we are talking. So only glucose is being respired. No other alternative substrates are entering in the pathway at any of the intermediary stage. Is that clear? Is that clear? Is that clear? So this kind of assumptions are not valid in the living system because everything works simultaneously. They are not taking place one after another. Right? So whenever whatever is required, that substrate will be withdrawn. It will be used later. So this is what, let's say here they are saying like it is useful to do this exercise to appreciate the beauty and efficiency of the living system. But here they are saying that this is a net gain of 38 ATP. So this is beta incorrect. This is incorrect. Now I told you the logic behind as well. Okay, so this is incorrect. Do rectify this part. So baki beta, it is amphibolic. Why do we say it is amphibolic? Amphibolic means catabolism and anabolism is going on. So this is important. Question can come from this part. So you can see fats made up of fatty acids and glycerol. So when required, body can use these fatty acids to make acetyl coenzyme. That acetyl coenzyme can enter in Krebs cycle. Glycerol, it can ultimately form glyceraldehyde 3-phosphate or dihydroxyacetone 3-phosphate. See, that can also enter, make pyruvic acid, can enter in the Krebs cycle. Same for carbohydrates, simple glycolysis, your amino acid, they can either form pyruvic acid, then they can enter in the process. So when required, I can just break these fats, fatty acids and glycerol, we can add that here, right? We can add that here, right, in the processes, and when required, we can withdraw this. Just when there is a requirement of fat synthesis, I can take acetyl coenzyme, it will make fatty acid, glycerol, and then the fats will be there. So this is all about the topic, beta. Let's solve few questions and then let's go ahead. So ye dekho, recent PYQ. So let's get started. Everybody, let's get started. Then our last chapter is there, which will hardly take your 1.5 hour, right? I'm not going to write anything there. We will simply discuss it. We will simply finish it. So you read the question so that we can start the process then. Okay? Quick. Quick, everyone. So see, if you know the RQ value, you can very simply answer it. And if you read NCERT directly, so you can, right? See, it's simple. Chalo, you solve it and then I'll answer. Do it quickly. Yes? See, beta, 145. So CO2 evolved is, CO2 is 102, 145. So you know that you are talking about the fats, the value will be 0.7. So it will be in between 0.5, right? It will be in between 0.5 and 0.95. है ना? So B is the correct answer. I don't know why are you making this. You can even simply do the calculation. Complex 2, right? Again, 2025 PYQ beta. Complex 2 is also known as. So you get such simple questions from this topic, right? You people get such simple questions from this topic. Hanji, quick. Complex 2. We know it very well. Very simple it is. So it is succinate dehydrogenase. So B, let's stop the poll so you can see the result. Chaliye, next question. Match column 1 with column 2. Quick. Let's see the question first, then let's start. Match column 1 with column 2. Quick, everyone. Match column 1 with column 2. Uh-oh. I'm starting the poll. So you answer it. Quick. Quick, everyone. So oxidative decarboxylation, glycolysis. So you know that it is the EMP pathway. B will go with 4. Okay. So then oxidative decarboxylation is occur. Oxidative phosphorylation, that is after the electron transport chain. This. So D with 1, A with 2, C with 3, D with 1. This is what we have, A. Next part. How many times decarboxylation occurred during each TCA cycle? So the question is saying each TCA cycle. So make sure you remember the reactions basically, beta. So then it is going to be very easy for you. Okay. How many times decarboxylation occurs during each TCA cycle? So it is going to be it will occur twice, right? It will occur twice, right? 2 times in each cycle it will be there. So what is the result? 1. Nobody answered it. How many times decarboxylation occurs? Beta, during each TCA cycle it is going to be twice. Chaliye, fatty acids are connected to respiratory pathway through. How the fatty acids are connected to respiratory pathway? Quick. How fatty acids are connected to respiratory pathway? So fatty acids are connected with respiratory pathway through fatty acids and glycerol. Fatty acids and glycerol. So you know that it is going to be acetyl coenzyme A. Is that clear? Acetyl coenzyme A. So 2 chapters are done and dusted. Let's start the last chapter, PGD, which we have even discussed during our previous NEET attempt also, but let's discuss it here today as well. So then it will be very easy for you. So beta, if you guys will follow this particular plant physiology one shot, there is nothing which you have to discuss or do extra. Everything will be covered here, okay? Everything will be covered here. So let's start. So any doubt so far? Any doubt so far? Chali, let's go. So this is what we are going to discuss, plant growth and development, a very simple chapter. So what you have to understand here, the differentiation, de-differentiation, points like that, and then the topic is going to be easy. So before starting any point here, let's discuss few things, right? Let's discuss few things here. Dekho. First of all, you know that we all develop from a single cell, which we call as zygote, isn't it? We all develop from a single cell, which we call as what? Which we call as zygote. Yes or no? We all develop from a single cell, which we call as what? Which we call as zygote. Now, this zygote is going to grow into a complete organism. So this property is known as totipotency. First, I'll explain you the basics and then we will start. That property is known as totipotency. So totipotency means it is the ability of a single cell. What's that? It is the ability of a single cell to grow into a complete individual. To grow into a complete individual. Is that clear? So it is the ability of a single cell to grow into a complete individual. So that is what totipotency is. First point clear? That is what totipotency is, a very important property. Just in tissue culture also, we take a part of a plant, which we call as explant, right? That part, we will make sure that it has to gain its ability to divide again so that we can develop a complete part from that. We'll come to that. So firstly, totipotency. Now, you know that, just say, let's take the example of humans. We are developed from a single cell, which we are calling as zygote. We all are developed from a single cell, which we are calling as zygote. This is what I told you. Now that single cell will divide. It will form 2 blastomeres. It will form 4 blastomeres. It will form 8 blastomeres, 16 blastomeres, and so on. And so on. Yes or no? And so on. Yes or no? Yes or no? Yes. Just a minute, beta. Just allow me 1 minute, please. Done, bache? So this is what we have, right? Totipotency. Now, what's the next part that we have to keep in our mind here? See, now the single cell is growing, right? The single cell is growing and it is going to form a multicellular structure, right? Multicellular, proper organ system and all like this. Now, what you have to understand here, you have to understand here a very simple thing. There are 3 things that we need to know here. One is differentiation, right? And if this topic is clear, everything is going to be crystal clear for you. De-differentiation, differentiation, de-differentiation, and then there will be re-differentiation. These are the 3 things which you have to complete. Differentiation, de-differentiation, and re-differentiation. That is what we need to know. Just say initially cells, they were just dividing. They were just dividing. Even in the case of humans also, they were just dividing. Later their fate, right? Later what is going to happen? Their specification will occur. right? Their fate will be fixed. Just for an example, in plants, if I have to give you the example, you have the meristematic cells, you have the meristematic tissue, you have the meristematic tissue. You know that it is just dividing. And once this meristematic tissue, bacha, some cells from this meristematic tissue, they become permanent. So what is the point of this permanent tissue? Permanent tissue is the one which is having a fixed function, fixed role, that this is what I have to do. I am parenchyma, my job is this. I am xylem, my job is this. So this type of tissue, they are specified. Their fate is clear. So this is what differentiation is. This is what differentiation is. So differentiation means when you, let's suppose you're actively dividing cell, when it will become specified, when its specification will be done, when its fate will be fixed. Ki now you are not just actively dividing, you have to do a particular job. This is what differentiation is. Then comes what? Then comes beta de-differentiation. Then comes what? De-differentiation. Just say, let's suppose this is a tissue. This is a permanent tissue. Okay? Now this tissue again regain ability to divide. This tissue regain ability to divide. It start dividing again. It regain ability to divide. So this is the de-differentiation. De-differentiation. Ki again it will start dividing. Right? Now it is start dividing again. It is dividing again, beta. Right? It is dividing again. So if it is dividing again, right? If it is dividing again, then again its fate will get fixed. So this is what re-differentiation, that again it will get differentiated. So this is what we have to discuss in this particular chapter. So like you will start from promeristem. Just say your promeristem, it forms primary meristem, primary meristematic tissue. Right, that primary meristematic tissue will help in the growth of the plant, the primary growth of the plant. Then beta, when its differentiation occur, then what will happen? Then there is the formation of, after differentiation, what will you see? There is the formation of primary permanent tissue. There is the formation of primary permanent tissue, and after that there is there will be, let's suppose now this primary permanent tissue, if it will regain the ability to divide, then it forms secondary meristem. So this is what we discuss in anatomy as well, bacha. So secondary meristem is the result of de-differentiation, right? It is the result of de-differentiation, bache. And after this, secondary meristem will again become secondary permanent tissue. It will again become secondary permanent tissue. So this is re-differentiation. So first of all, this is something that you have to remember, and if you know this from the anatomy part, so when we discuss the secondary meristem, beta, when we discuss the secondary meristem, so that is required for increasing the girth. That helps in increasing girth or the diameter. That helps in increasing girth or the diameter, beta. Right? In dicot stems, we see that mainly. So that is secondary meristem, result of the de-differentiation. Now, in this particular chapter, what are they saying? Plant growth and development. What exactly is the development? Development is the sum of two processes, which is growth and differentiation. When it comes to the growth, beta, growth is an irreversible permanent change. What is growth? Growth is an irreversible permanent change. Irreversible it is, permanent change. And if you remember, it is basically a characteristic feature of living beings. है ना? Fundamental feature या characteristic feature, but it is not a defining feature. So why do we say that it is not a defining feature? Because in the case of non-living beings also, you will see that in their case also accumulation, right, will be there. The deposition will be there. External growth is there. So defining features are those which are present only in the case of living beings, yeah, which are not shared with non-living ones, and without any exception they are present in living beings. So this is what growth is. Now, when we talk about the growth, you know it very well, and you should know it, that growth is the result of what? If beta metabolism will result in growth, and in that metabolism your anabolism should be more than catabolism. The anabolism should be more than catabolism. Build-up should be more than breakup. Like if you want to save money, then your expenses should be less, your savings should be more. It is like that. And you already know what is the meaning of differentiation here. So all plant organs, they are made up of zygote. They will develop different structure, the functional characteristics, the attributes. This is cell differentiation, right? So cell differentiation means it's a biological process where a less specialized cell will become more specialized, bache. So here what is happening, all the cells are having same DNA, but they express different genes on the basis of their location and the signal they receive. Like in the case of plant, if the cells they are at periphery, they are going to make the epidermis. It's like that. So location also matters the most, and here this differentiation is both internal and external factors are responsible for that. So positional cues, like where that cell is, which gene is it expressing, the hormones are also going to be there. Now next part is, you know that in the case of plant, seed will be there, then seedling, then plantlet, then mature plant. Simple sequence: seed, seedling, plantlet, then finally the mature plant will be there. So we know that the trees, they grow continuously in height as well as in girth. Not all are growing in girth. In monocots, you will not see that there is secondary growth, but height will also increase. That is the primary growth. When girth or diameter is increasing, it is the secondary growth. In leaves and fruits and flowers, you will not find it, but otherwise we know that this is going to be there. Why? Because plants are having the meristematic tissue, but we have limited size. Even we have limited growth, and even these leaves, flowers and fruits, they are also having the limited size. They appear and fall periodically, time to time. This is what we know. Now, you know that in a plant, first vegetative phase is there, in which, it will build its root, stem, leaves, so that it can prepare itself for reproduction, because reproduction will require energy. So in that particular vegetative phase, when roots are well developed, they will absorb the water, right? The leaves are there, so the photosynthesis should be there, the storage of food should be there, and that is why after the vegetative phase, there is the flowering phase, because flower is the site of sexual reproduction, beta. Here, the energy is required. So vegetative phase will make sure, that it should, vegetative phase means it will prepare itself. It is going to prepare itself for what? It will prepare itself for the flowering phase, because that is basically the phase in which what will be there? Energy, right? Basically, you know that energetically expensive process. So growth, I told you already, one of the most fundamental and conspicuous, clearly visible characteristic, right? So it is irreversible, permanent increase in size of an organ, in its parts, or even of an individual cell. So we know that metabolism, chemical reactions are required for it, and here it occurs at the level of energy. But always remember, for a growth to occur, right, the anabolism, that is build up, should be more than catabolism. Anabolism should be more than catabolism. That's what we have to keep in our mind. Is that clear? So plant growth is generally indeterminate. In the plants, beta, open form of growth is there, and here you will see that meristematic cell, they keep on dividing, and the cells that they form, they don't have their fate fixed. Matlab, it's like, let's suppose there is a couple, they have a child, and they are like, this child is going to be a doctor for sure. Mother, that child is not having a choice. That child will grow. Everything is there, but that is fixed, that you have to be a doctor. Now, there is another couple who is saying, okay, our child can be anything, right? Doctor, engineer, cricketer, singer, whatsoever is there, right? So, plant growth is indeterminate. This is what I just said. So, what does it mean? That plants, they retain the capacity for unlimited growth. Why? Because they have the meristematic tissue. Why? Because they have the meristematic tissue. So this ability of plant is due to the presence of meristem. We know it very well. So meristematic cells, they will divide, they will self-perpetuate. They are increasing their number, they are dividing. They are making more cells like them, they are dividing. This is what we need to know. Now, new cells are continuously added by meristem. We know it very well. So here, the new cells which are added. So any cell is having potential to divide and to form new tissue or organ. This is the open form of growth. So when I'm saying open form of growth, means the new cells that are formed, they can, right? The new cells that are formed, they can form, right? Any, like, meristematic cell is dividing, it is forming the new cells there, and that particular cells, right, that particular cells can form new tissues and organ. So this growth is indeterminate, means the growth is continuous, right? What is the meaning of indeterminate growth? That it is continuous. And what is the meaning of open form of growth? That the new cells that are formed that can divide and can form any part. This is the meaning. Just say apical meristem of root and shoot. Cells, they keep dividing, beta. They are making new tissues. So why do we call it as open? Because new structures can arise continuously from these undifferentiated cells. So cells are undifferentiated, they can form new cells. So plant growth is indeterminate, means it is continuous. What is the meaning of closed growth? Growth where the future fate of cell is already fixed, right? Just I told you that this kid is going to be a doctor. That is it. So specific group of cells, they will make specific structure only. So this is closed form of growth. So in lower plants you will see that, right? In lower plants, you will see that, and growth is more deterministic and restricted there. Like new cells will form, but their fate is going to be fixed, that this is what you have to form ultimately. Is that clear? So meristem is closed in organization. It is having fixed initials producing fixed tissue. They'll be having fixed initials. They'll be like, this is what we have and this is going to make this only. Matlab, like rigidity. Rigidity is there. Now, differentiation, told you already, the process by which cells will derive from root apical meristem, shoot apical meristem, and even cambium, they mature to perform specific function. So if you will ask me differentiation in simple words, specification, fate will be fixed. Fate will be fixed, and as per that the structural changes will be there. As per that the changes will be there in their cell wall, the changes will be there in their protoplasm. This is what we are going to have. Just say for an example, if I talk about the xylem. So xylem will have what? Xylem will have tracheids. Xylem is going to have vessels, right? Xylem is going to have tracheids, vessels. You will have xylem fibers there, and then you are having the xylem parenchyma, which is living, rest all are dead. Now tracheids and vessels, they are the treachery element. Treachery element means they are the one that are going to they are the one that are basically going to transport the water. Okay? So because they have to move the water and ions, so here you will see the cell wall is going to be quite rigid, like thick, and the protoplasm will not be there. Like it is, these are the dead cells. Why? Because they have to, right? They have to carry the water. So the changes are as per the function. The changes are as per the function. So if differentiation will be there, if fate is going to be fixed, so the changes will be there, they will be as per function. So treachery element which are having xylem, vessels and tracheids, cell will lose protoplasm. They will develop thick, elastic, lignocellulosic secondary cell wall. So basically structure will be related to the function. Baki de-differentiation, direct PYQ beta, it is the de-differentiation due to which secondary meristem will form. So phenomena where living differentiated cells, right, they will regain the power of division under certain conditions. Okay? So जैसे interfascicular cambium, cork cambium, they both are the result of secondary meristem. So these two you will understand if you have studied the anatomy. Just in anatomy, when we see the stem structure, just you remember in the dicot stem, the vascular bundles, they are in the form of a ring. So in between these two, these are the medullary rays. Here parenchyma cells, they will become, you know, they will again gain the ability to divide. They will form this interfascicular cambium here. So cork cambium, which is also known as phallogen, it also form like this, right, due to the de-differentiation. So they are the example of secondary meristem. Done beta. Baki redifferentiation hai. Again beta, these de-differentiated meristematic cell, they will again become mature. Just it's like you have studied, you have completed your masters, like you have completed your degree, now you are the MBBS doctor, right? You have started doing a job or something. Now you're like, no, I'm not satisfied. I want to be a UPSC aspirant. I want to crack UPSC. Now you started studying again. Means you are de-differentiated because you started studying again. Now after studying, if you will become IAS officer, again you acquired a job. Again, like I'll be like you are re-differentiated. That's how you can understand that. Is that clear? So secondary xylem, that thick woods in the dicot stems, that secondary, this thick wood, secondary xylem. So they all are the result of redifferentiation. So that's what you have to remember. Okay, beta. So growth can be open, closed. Same way differentiation can also be open or the closed. Open differentiation means, जैसे cells near root apical meristem, they can become root cap cell. Cells push to periphery, they will mature into epidermis. So location is also defining, but so here you can see growth जैसे plant में beta, it can be in continuous or fixed. So differentiation can also be open. So open differentiation means cells are from the same meristem. Same meristematic cells are coming from same meristem, but they can grow into different structure on the basis of their position. So positional cue is the thing that we are discussing here, okay? Okay, so cells are coming out from same meristem, but the structure they are forming, they are different. So this is the diagram, shoot apical meristem, root apical meristem, but always remember root apical meristem is subterminal, right, little away from center. You can remember it from the morphology diagram. Just here you will see root cap, and then after that you will have meristematic zone. So that's what you can see. So beta, this is how they are showing. So they will help in the primary growth. And when you are talking about vascular cambium, which is the example of secondary meristem, so here you will see there will be increase in girth, right? Girth will increase, the diameter will increase. That's what you have to remember. So your dicot stem, your gymnosperms, they are going to show the secondary growth, right? Primary growth means, right, from primary meristem, the elongation will be there. Secondary mein there will be the increase in the girth. So lateral meristem, which is also known as cylindrical meristem, secondary meristem. So cock cambium and vascular cambium is the example. They appear later in life beta, and they are responsible for increasing the girth. So secondary growth I have already explained. Baki beta growth is measurable, right? You can see, let's say in a single cell, protoplasmic growth, protoplasm increase, you might not be able to see. So how can we understand that? Let me make it very simple for you. Just say growth. If growth is at cellular level, right? So why? Because there is increase in amount of protoplasm. So it is difficult to measure directly. So we can measure the, say, some quantity which is more or less proportional to it, right? Just say you cannot measure something directly. So more or less proportional quantity you people can measure. Then beta, other parameters are there. We can go for fresh weight, dry weight, length, area, volume, and cell number as well. This is what we can do. So growth is expressed as increase in cell number. So this example is important beta. One single maize root apical meristem, one single maize root apical meristem can give rise to more than 17,500 new cells per hour. This data is important. Very good, Mr. Ames. This data is important. So one single root apical, one single maize root apical meristem cell is giving 17,500 new cells in 1 hour. That's what you have to remember. Now, increase in size of, that is as per the cell number. Size of cell may, just say watermelon may increase in size by up to 350,000 times. Okay, so in the case of pollen tube, when you will discuss, you will see the length will increase in the surface area in case of leaf. So that is how we can measure the growth in different ways. Okay, that is how we can measure the growth in different ways. Done? Done? Is that clear? Is that clear? Is that clear? Done? So phases of growth are also there, right? And beta, do not miss the description part as well. So just say we have the zones here, right? We have the zones here. So zones of elongation is there. So by parallel line method, right? By parallel line technique, we can just detect the zones of elongation by parallel line technique. So this is given here. Do remember it. It is important, and do solve the questions from NCERT exemplar as well. So zone A, B, C and D, it immediately behind the apex have elongated most. Now, let's understand that the period of growth, what is it? Just we have, if you remember, even in the case of morphology, just we discuss na, just morphology mein when you talk about the root, right? When we discuss this regions of root, जैसे beta in root, this is going to be your base, this is going to be your apex, this is going to be your base, this is going to be your apex. So this is going to be root cap, right? This is going to be meristematic zone, this is going to be meristematic zone, then this is going to be zone of elongation, and then bache, there will be, finally, there will be maturation zone, because here cells are mature, they have the fixed function. So that's why this is also the root hair zone, right? With the help of this part, let's discuss this portion, okay? Let's discuss this portion with the help of this part. What is happening? Meristematic zone. What do you see there? You are going to find here actively dividing cells. So beta, if a cell is actively dividing, right, actively dividing, so what will you see? You will see cells are smaller in size. They are smaller in size. Because they are smaller in size, they are definitely active. In this case, you will see they have primary cellulosic cell wall. And this primary cellulosic cell wall is there. This primary cellulosic cell wall is going to be there, right? And here it is going to be thin as well as flexible. It is going to be thin as well as flexible. This is primary, right? The living cell wall. Now, when you look at it, you will see there will be conspicuous nucleus. The nucleus is going to be conspicuous, like it is prominent nucleus. Dense cytoplasm will be there. Prominent nucleus, dense cytoplasm will be there, and here it is going to be compact. It is going to be compact, and you will see there will be abundant plasmodesmatal connections. There will be abundant plasmodesmatal connections are there. So many gap junctions which are linking it. So now these cells are actively dividing. Their metabolism is high. They don't have stored food. And moreover, students, here you people are going to see, because from these properties they do ask the question. They do ask the question. So here either vacuole is absent, either vacuole absent, if present will be of very small size. If present will be of very small size. That's what you need to remember. Okay, either it is absent or if it is present, it is going to be of very small size. These are the meristematic cell. Slowly in the zone of elongation, you will see increased vacuolation will be there. Right, vacuolation will be there. So ultimately this is the part, right? So the changes in cell wall will also start. So this is the part which will help in increase in length. This is the part that will help in increase in length. That's what you need to know. This is the part which will help in increase in length, and finally the maturation is there. So this is what we have here when we talk about the phases of growth. So that's what is basically written in NCERT also. Jैसे constantly dividing cells at the root apex and the shoot apex. They are showing meristematic region. Here cells are rich in protoplasm, dense cytoplasm, rich in protoplasm, large conspicuous or prominent nuclei. Cell walls are primary in nature. They are thin, cellulosic beta with abundant plasmodesmatal connections. I told you they are compactly arranged as well, right? They are compactly arranged as well. Now next to it, just away from the tip, you have elongation zone where vacuole formation will be there. So cell enlargement, new cell deposition will be there. This is the characteristic. So it helps in increasing the length as well. Then further away from the apex, right? Further away from the apex, what else do you have? Right, it is more proximal to the phase of elongation. It is more proximal to the phase of elongation that lies the portion of axis which is undergoing the phase of maturation. So cells of this zone, they attain their maximal size in terms of wall thickening and protoplasmic modification. So modifications will be there. Modifications will be there, right? Modifications will be there. Done beta? So in anatomy part we also understand that. Chaliye, next part is related to the growth. Dekhiye, two types of growth is there, right? Basically here we will talk about the growth rate, not growth rate. So growth per unit time, it is the growth rate. Growth per unit time, it is the growth rate. So we have two things here. One is very simple, arithmetic growth. Beta, let's suppose you have one cell, it is dividing into two. Now, this particular cell is saying कि I will not divide further, I will just grow. है ना? I will just grow, will do a job, I am not going to divide further. So this cell is like, it's okay, I will divide. I will divide. So after its division, the two cells you will get. So out of that again the same story. One is saying कि I will not divide, I will just undergo the maturation. Another one will like, chalo, I will divide. So after that what will happen? Now this cell will again divide. So this is the arithmetic growth. One, then two cells. So out of these two, only one is dividing, one is just simply growing. So that's why you have three cells here. Here also, one is dividing, one is simply growing, four cells. So this is the arithmetic growth rate, 1, 2, 3, 4, 5, like this. Now, in the case of geometric, Now, in the case of geometric, what you people will see, one cell dividing into two. Just let's suppose your zygote is growing. These two cells, they will form four. Then four cells, they are going to form 8 cells, like that. So that is how it is going to grow. That is how it is going to grow. Is that clear? That is how it is going to grow. Is that clear? Done, beta? Done. So zygote will divide. Zygote will divide. Geometric phase mein all the cells they divide. So in the arithmetic phase, some cells will be capable of division, but some cells they will lose the ability to divide. So diagram based questions can be there. Now let's discuss this particular graph and everything. So this is what the arithmetic growth is, beta. So x-axis is showing you the time, y-axis is showing you the height. So you will never ever draw it from the origin. You are talking about the growth rate. Matlab some growth is there. Now you are checking in per unit time what is the rate of growth. So you will not draw it from the origin. Origin means 0. So that is how it is going to be. So this is what you will see when it comes to the arithmetic growth. Constant linear growth. This is for length against time, right? Length against time. Is that clear? Is that clear? So growth rate, it shows an increase that may be arithmetic or may be geometric. So arithmetic mein what is happening? Arithmetic mein what is happening? The simple thing that is happening here is falling mitotic cell division, only one daughter cell, told you already, will continue to divide, other will differentiate and mature. So always remember this trick, it is beta DM. First differentiation, then maturation. First differentiation, then maturation. What is there? First differentiation, then maturation. After that, what is happening? The simplest expression of this growth is in the form of root elongating at a constant rate. So we are plotting length against time. What are you getting? This is what you are getting. LT is equals to L naught plus RT. That's what you have. So LT means length at time t, length at time 0. So R is basically growth rate to the elongation per unit time. It is the growth rate to the elongation per unit time. This is growth rate elongation per unit time, beta. This is what you have to understand. This is simply the absolute growth. Ki growth rate, matlab in given time, what is the growth rate? So growth rate here is elongation per unit time, right? Growth rate, R is the growth rate here. What R is showing growth rate here? Elongation per unit time. Now let's talk about the geometric phase. So geometric growth, it is showing us this S-shaped curve, right? Sigmoid shaped it is. S-shaped, sigmoid shape. So here what you people will see, you will see lag phase, you will see log phase, which is also known as exponential phase, and then stationary. So what is happening here? Growth is there, but source, the limited food, right? Unlimited supply is not there. It is going to be limited. So initially growth is slow, then exponentially it will rise, and then beta, because the resources, food is also going to be, less, but limited. So there is a stationary phase. So x-axis is showing you the time, y-axis is showing you the size as well as the weight. So this is the geometric growth, beta. This is the geometric growth. So S phase, it is basically S-shaped or sigmoid curve is the characteristic. And you know that in living beings, it is going to be like that. Slow, then exponentially it will rise, and then beta, there will be the stationary phase. So in most systems, you know that initial growth is slow, which is the lag phase. And then it increases, which is the exponential phase. Right. So progeny cells, they are dividing, both the cells they are dividing, right? But limited nutrient supply is there. So growth will slow down beta after a time, and due to which what will happen, the stationary phase will be there. The stationary phase will be there. So if we plot this parameter, we will get a sigmoid growth. So in natural environment, the sigmoid growth is there. Just you can relate it with ecology. We discussed Verhulst Pearl logistic growth curve. There is one J-shaped growth curve. So J-shaped is theoretical. Verhulst Pearl logistic growth curve is basically what? That is basically S-shaped. Right, beta? So this is typical for all cells and all. So how do we write it like this? W1 is equals to W0 ERT. So in the paper, they have even asked what is E, right? That is again a PYQ, right? Base of natural logarithm. What is R? You know that growth rate. Just we also call it as elongation per unit time. But here you have final and the initial. And we know that final will always depend on the initial. Just say, let's suppose some people beta they start from zero, they build up everything by their own. Some people they are at certain level, then they start building up. Obviously in their case you will see more growth because they were already at a certain level. But some people, those who build everything from scratch, in their case growth will be there, but in comparison to another one who's there at this level, the difference is going to be there. Okay, so here R is the relative growth rate. R is what? R is the relative growth rate, and it is the measure of ability of the plant to produce new plant material, right? R is basically the relative growth rate. So it is the measure of ability of plant to produce new plant material. So it is the relative growth rate. So it is also the efficiency index. It will tell us, right, because you are comparing, you will see, right, here you will see the new plant material produced as well. So it is the efficiency index. So final size will depend on the initial size. So here we can see the comparison here also beta. Just say these are 2 leaves. Dekho, in one case it was at 5 centimeter. Last growth is 10. Matlab growth rate is there 5 centimeter square. This is this absolute growth rate. In a given time, right, the increase is this. Same way, here in this leaf, it is at 50 centimeter square. It will grow to 55 centimeter square. Again, the increase is 5 centimeter square. So if for this leaf, for A as well as for B, for A as well as for B, right, A as well as for B, the absolute growth is basically 5 centimeter square. Now let's go for the relative growth rate. So here, what are we saying? We are saying that the measurement and quantitative comparison is there, beta. So measurement and comparison of total growth per unit time is absolute. Just say for an example, you were studying. You told me, ma'am, I solved 10 questions. Now after 1 hour, you are like, ma'am, I have solved 100 questions. Okay, there is another student. I'm like, okay, how many questions you solved? Ma'am, I have solved in 1 hour, I have solved 200 questions. So now I'm not comparing you. Okay, one has solved this, another has solved this. Okay, done. Matlab, earlier in 1 hour, you solved 100 questions. In next 1 hour, you were like, ma'am, now I have solved 200. So I'm just comparing. Ki, acha, theek hai, student A has done this, B has done this. Absolute growth, when I'm saying ki measurement and comparison of total growth per unit time, ki in a particular time, what is that total growth? Now when I'm saying relative, so growth of given system per unit time on the common basis, जैसे either I can take per unit initial parameter, जैसे here if I will take, again here if I will take initial parameter, it is 5. Initial parameter per unit initial parameter is called relative growth rate. जैसे growth here is 10, but initially what was there? 5. Initially what was there? 5. Yes or no? Yes or no? Initially what was there? 5, then 10. But that one will be from 50 to 55. So that's how you check the growth rate, relative growth rate. First you will write the absolute growth, right? That is the growth of given system per unit time. First you will write the absolute growth rate, that is growth of given system per unit time. Because it is relative growth, so you will also consider the initial parameter. Then you are going to multiply it with 100. You are going to multiply it with 100. So in first case, right, leaf A, 5, initial was 5, multiply by 100, answer will be 100. In the case of leaf 2, initial was 50. Sorry, initial absolute growth rate was 5, initial was 50, multiply by 100. So you know that it is going to be 10. So that is how we check it. Do read about it, quick. Do read about it, guys, quick. Done? Okay. So it is like that. Done? So that is what we have, okay? So baki beta growth ke liye water, oxygen, nutrients are required. Baki this table, it's a very important one, right? This is a very, very important one. They can ask you the question from this part. So development, you know that sum total of, right? It is basically the term that will include all the changes that organism will go through. So development is growth plus differentiation, I told you. So you can see, right? You can see this is what they are showing. You have meristematic cell, the plasmatic growth will be there. Matlab its cytoplasm, its protoplasmic growth will be there. Its protoplasm is going to increase. If it is increasing, obviously bacha expansion will be there. Expansion will be there. Protoplasmic growth, expansion is there, and then there will be DM. That's what you have to remember. DM, differentiation, maturation, differentiation, maturation. Finally, you have mature cell, and that will undergo senescence, that is death, aging. So when there is a plasmatic growth, because we know that nuclear cytoplasmic ratio, when it increases, you know that a cell also divide into two. Cell also divide into two, and that cell, it grow, it shows expansion, differentiation, then maturation, and finally mature cell will undergo senescence, which is basically the aging, right? So this complete cycle from germination of seed to senescence is basically aging. Now next topic that we have to understand is plasticity. Okay, so after this plasticity wala part, we have PGRs, the last topic, and then the class will be over. So I think in next 40 minutes we can say bye bye to each other, but please pay attention. So plants, not just plants, but in animals also you see that. So they follow different pathway in response to environment or phases of life. They form different pathway in response to environment or the phases of life to form different kinds of structure. Just this ability is plasticity. What is plasticity? That plants, even this is something that we see in the animals also, beta. They show, right, different pathway in response to environment or even their phases of life. Just for an example, if you talk about coriander, the young leaves will have different shape, the mature leaves will have different. For the another way, just there is one more example of a plant, buttercup. Underwater, its leaves are dissected. That is the requirement there. Above water, that lamina is complete. So these are the kind of examples. This is plasticity. So plasticity means that in the lifespan, or you can say that in response to the change in environment, the different types of structures that we see. So plasticity is having example, that is what we call as heterophily. Which we call as heterophily. Is that clear? Which we call as heterophily. Is that clear? So heterophily in cotton, coriander, larkspur is there. So development mein beta extrinsic, intrinsic, both factors are there. So heterophily is a type of plasticity, right? So plasticity's type is heterophily. Ki bhai, it is showing different types of leaves in its lifespan. So ability of same, the genotype is same here beta, but it is giving me different phenotypes in response to environmental change. So this is another definition that we can have, that genotype is same, but in response to environment change, it is giving me different phenotype. So just cotton, coriander, larkspur, they are the example. Buttercup, which is also known as ranunculus, it is also the example. Maize and coriander seedling, their juvenile and adult leaves, they are different. Just ye dekho. Here they have given you the example in heterospory. So larkspur is given and buttercup. So buttercup is your ranunculus. So in this case, you can clearly see terrestrial habitat is having different leaf shape. Water habitat is having dissected leaf shape. What sort of leaf shape is there? Dissected leaf shape. And here what will you see? Juvenile and adult. So in larkspur, as per their lifespan, as per their habitat, they are showing the different leaves. So heterophily is the example of plasticity. Do remember that. Do remember that. Now beta, PGRs, that is plant growth regulators. Beta, the word is PGRs. They are the growth regulators, right? They will regulate the growth. So some are the promoters. They are going to promote the growth. Some are the inhibitors. They are going to inhibit the growth. Just say for an example, when you talk about the auxin, you talk about the gibberellin, you talk about the cytokinin. They all are going to be the promoters. And then ethylene is also there. Abscisic acid is also there. That is ABA. They are basically the inhibitors. Now, bache, you will understand that this ethylene is placed in both the categories, right? It is placed in both the categories. It is placed in both the categories, but majorly it is an inhibitor. That's what you need to know. Majorly it is an inhibitor. Is that clear? Majorly it is an inhibitor. Is that clear? So promoters, the one which are going to promote the growth, inhibitors, one which are going to inhibit the growth, right? So this is what you have to know here. Ethylene can be in both the category. Done, bache? Sure? Sure? Chaliye, let's move ahead. So PGRs, so this is what you need to know, bache. When you talk about the auxin, right? Auxin, so it is basically indole compounds. Indole 3-acetic acid, IAA, right? So auxin beta, so for auxin, your amino acid, for its synthesis, amino acid, tryptophan is required. Tryptophan is required. Then becha, adenine derivative is what? This is your N6-furfuryl aminopurine, my favorite, cytokinin. Adenine derivative is your cytokinin, which is mainly playing the role in cell division. So it is adenine derivative N6-furfuryl aminopurine, my favorite, right? N6-furfuryl aminopurine. Then comes beta your derivatives of carotenoids, that is your abscisic acid, which is the stress hormone as well. Then terpenes for gibberellic acid, and then the gaseous hormone is ethylene, and for that beta the amino acid methionine is required. So sulfur containing amino acid methionine here. So tryptophan is required for the auxin synthesis. This information is important. This information is important. So they are phytohormones as well, right, bache? So discovery of plant growth regulators, it is basically accidental. All the 5 plant growth regulators, accidental discoveries. For that we use the word serendipity, mane by chance discovery. Jaise some scientists, they were studying the phototropism, they discovered auxin, right? Some were studying the triple response thing, yeah, they were studying why seedlings, they were growing, I would say, horizontally and all, they got to know about ethylene. So these are the different ways. Matlab, scientists, they were studying something else, but they discovered something else. So this is serendipity, right? Accidental discovery is there. So beta, वैसे in the case of auxin, if I go as per NCERT, right, then it will be just completed in 1 or 2 lines, right? But here I have added many points regarding the discoveries, which I believe are not that much required for you. So let's, what do you want? NCERT or little extra? NCERT or little extra? What do you want? NCERT or little extra? NCERT right now is important, ma'am. We have re-NEET in few days. So let's stick to the NCERT. Don't worry, we will. So, ye dekho, discovery. So here I will just highlight the important pointers, but let me tell you what happened was ki Darwin and his son, they were observing the phototropism, right? They found that because phototropism, bending towards the light, so they found that there is something which is, some substances there which is basically causing this bending, because when they removed the tip, bending was not there. So this is like how they related it. But then some other scientists, they found ki are yaar, this movable thing that Darwin and his son was saying is a chemical compound, and then finally, right, went isolated that chemical compound which we call as auxin. So yes, in the notes itself I have added the extra pointers regarding the discoveries also, but you stick to the NCERT part. So as I said, all the discoveries are serendipity. So how it started? It started with the observation of Charles Darwin and his son Francis Darwin. So they were studying the colutile, the tip of canary grass, responded to uni, means single side, say illumination is there. So like this is the light source, this tip was growing towards that. So they were studying the phototropism. So they conducted many experiments, right? They were studying that canary grass. So question can be, the Darwin and his son, what were they studying? They were studying the canary grass. So they were studying the phototropism. They were studying the bending towards the light. So after a series of experiment, beta, it was concluded that the tip of colutile was a site of transmittable influence that caused the bending of entire colutile. Means they observed that, right? They removed the tip also. They conducted many experiments, and then they came to the conclusion that something is there in the tip which is going downward, which is causing this bending. which is causing this bending. And Boyson, the another scientist not given in NCERT, he found that that transmittable substance that Darwin and his son was saying is a chemical compound, and then finally it was isolated by F.W. Wendt. So he isolated it from the tip of oat seedling, right? That is Avena, scientific name. From oat seedling, they isolated that chemical that we call as auxin, So detailed discoveries are also mentioned in the notes. You can go for that. Then gibberellic acid. So it is very interesting. Jaise Irichi Kurosawa in 1926. Jaise what was happening in Japan. In Japan, what was happening? Ki rice seedlings, they were growing enormously. And it's not like if they are growing, ma'am, it's a good thing. What's so bad in that? Then they were falling down. Excessively, foolishly they were growing and just falling down. So E. Kurosawa in 1926, Erichi Kurosawa in 1926, he reported the symptoms of disease in rice seedling, right? Rice seedling when they were treated with sterile so what he did, see, first of all, just say in the Japan, this disease was there. So Becan, and I don't know the exact pronunciation here. So it is the foolish seedling disease of the rice, which was caused by a fungal pathogen, Gibberella fusarium. At that time, it was Gibberella fusarium. Now its name is Fusarium something, okay. So E. Kurosawa, he reported that the symptoms of the disease in rice seedling when they were treated with sterile filtrate of the fungus, the active substance were later identified as gibberellic acid. Basically, what happened was, rice seedling, they were growing enormously. So it was found that there is a fungal pathogen that was causing that. But later what they observed, even when they treated the rice seedling with the, they were growing the fungus in a, they grew that fungus in basically, so they grew that fungus in a culture, right? From that culture, they extracted, right? They just used that filtrate from that. The filtrate was not having the fungus, just filtrate, but still, right, the appearance of symptoms were there. So they came to this conclusion ki fungi, it is a fungi that is releasing a particular compound that is causing that, right? That the fungi is releasing a particular compound that is a chemical that is causing that. Just here, let me show you this. Ye dekho. Just early observation in Japan mein, कि Japanese farmers, they noticed that, they called it as foolish seedling. And in 1927, Irichi Kurosawa, Japanese scientist, he discovered that the fungus is causing that. This fungus is now Fusarium moniliforme. So he observed that the filtrates, that is the culture medium from the fungus, even without fungus, is also inducing this abnormal elongation. So he was like, ki the fungus is releasing some growth promoting substance. And initially, people were thinking it was just in fungus only, but later scientists, they isolated it from fungal culture, right? And with further research, we got to know it is not just in fungus, but in the higher plants as well. So this is the discovery of gibberellin. Right, beta? So first identified gibberellin is GA3, gibberellic acid, and now to beta there are so many gibberellic acid. Baki beta, next is for cytokinin. So when you people talk about the cytokinin, cell division. So what is happening here? Scientists, they were studying factors that promoted cell division. Okay? It was known that auxin alone could stimulate. Means it was already, by that time they were well aware that auxin can stimulate the cell elongation, but not sustained cell division. This auxin can elongate a cell, but it cannot sustain the cell division. So researchers were like, some other growth promoting substance will be there. So Skoog's experiment with tobacco callus. First of all, let's discuss what callus is. Callus is basically undifferentiated cell mass. What is the meaning of callus? Callus is basically undifferentiated cell mass. Now let me elaborate that. Just say for an example, in tissue culturing, the part that we take for tissue culture, we call it as explant. And now, due to the hormones, plant growth regulators, this explant, it will regain its ability to divide. It will form this cell mass, which we are calling as callus. So in tobacco callus, again, it is also serendipity. So Fox, Skoog and his team, they worked on tobacco pith tissue culture. So they were like, auxin can support some growth, but for continuous cell division, another factors they are required. So they extracted these factors from vascular tissue, coconut milk, yeast, right? Cell division was enhanced. So they were like, some division factor is there. Later, beta, Miller and Skoog, they isolated the first cytokinin, but it is not the natural one, from autoclaved herring sperm DNA. This is a fish. So from its fish DNA, from an autoclaved fish DNA, they have isolated the substance. They called it as kinetin, but it is not natural. So it is basically the modified adenine derivative. Adenine derivative, right? So baki beta, kinetin, as I told you, not natural, important. So it was artificially discovered, right? Baki natural was later identified, right? It was named as zeatin. So zeatin is the natural one. Kinetin is the synthetic one. Zeatin is obtained from your Z maize, that is your maize, right? That is your maize. Z maize is having that. Okay. So many natural cytokinins are there. So actually what happened here was ki it was a kind of, what happened was कि जैसे होता है ना, sometimes you're doing something, you are not getting the result. So out of frustration, that scientist, like a bottle was there in the lab. So he was like, he put that solution there. So he observed कि now the growth is there. Then he realized in that bottle DNA is present. So he tried to put the DNA to get the results, but he didn't. Later, he realized that that bottle was having old DNA. So maybe that DNA was the degraded one. That's how they came to this thing that it can be adenine derivative, or that's how they isolated that. Okay. Baki beta, ethylene. First of all, it's very interesting. In 19th century, what was there? People noticed that the street lamps where the burning coal gas was used, the trees surrounding, they were showing leaf abscission. Leaves were falling down and the stem, it was getting thickened. So they hinted that this is the gaseous substance, right, that is influencing the plant growth. Later, Cousin's experiment and Dimitri's experiment was there. But in simple words, what I would like to say here is, one second. So this part I would like to complete from this only. So later on, what happened? H.H. Cousins in 1910, they confirmed. So scientist name, they do ask in the paper. They do ask scientist name in the paper. So H.H. Cousin, they confirmed the release of volatile, matlab gaseous substance from ripened oranges, that is speeding up the ripening of stored unripened bananas. So later this volatile substance was isolated as ethylene. So for ethylene, always remember its bioassay is triple response. What is its bioassay? It is triple response. So bioassay means, bio means living. So basically here you are checking the, the concentration, the I would say the potency of a substance in a living being. Bioassay. So it is triple response, which we have to discuss. Then beta, during mid 1960s, 3 independent researchers, they reported the purification, chemical characterization of 3 different types of inhibitor. So inhibitor B, abscission 2 and dormin. Right. Inhibitor B, abscission 2 and dormin. Later all 3 were proved chemically identical. So this was abscisic acid. Next section is regarding the discoveries beta. So physiological effects. So here also I would like to discuss it from NCERT first. Not extra here, but just a minute. So that is going to be our last topic. So triple response also I'll discuss, but discovery is over. Chaliye, it's okay. Let's discuss it directly from here. So beta, before starting the auxin, so as I told you already, in the case of auxin, you have to remember they are indole 3 acetic indole compound. So we have indole 3 acetic acid. Indole 3 acetic acid, very common one. And about the auxin, there are certain other pointers which you have to keep in your mind, right? There are some other pointers which you have to keep in your mind. So in the case of auxin, always remember beta, it shows the polar transport, right? Let me discuss these points again. So auxin, I told you tryptophan is required. So here in this case, you people will also see that it shows what? It shows the polar transport. What will it show? It will show the polar transport. So what is the meaning of polar transport? Means the directional transport is there, right? The directional transport is there. So basically auxin show polar transport because it moves in a specific direction, okay? Right? So it moves in a specific direction. That's the point. Moves in a specific direction, but not randomly. So it moves in a specific direction, not randomly diffuse. So this is the meaning of being polar in transport here. Is that clear? So जैसे stem में it moves from shoot apex to downward. Moves from shoot apex to downward. So this is going to be basipetal. This is going to be basipetal movement. Are you getting my point? Basipetal movement. So in stem, this is how it is going to move. In stem, this is how it is going to move. Or roots mein beta acropetal. Move upward in some regions, but in some regions. So this is how it is. Okay, so it shows the polar transport. And now we have, right, when we talk about the auxin, another thing that we have to understand is that we have natural auxin also, we have synthetic one. This is also important. What do we have? We have natural as well as synthetic auxin. So do you have any idea here? Do you have any idea here of examples? Natural and synthetic auxins? Yes, everyone. Do you have any idea of it? There's a natural and synthetic auxin. So in the case of natural auxins, you will see, matlab naturally you are going to find it in the case of plants. So you have the example of indole 3 acetic acid, and IBA is also there. So what is this IBA? It is indole 3 butyric acid. It is indole 3 butyric acid. That is what you have. It is indole 3 butyric acid. That is what you have. Now in synthetic auxin, you have NAA. That is methylene acetic acid. You are also having 2,4-D. That is 2,4-dichlorophenoxyacetic acid. And 2,4,5-T. That is 2,4,5-T means trichloroacetic acid. Trichloroacetic acid. That is what you have. Okay? So this one is VD side, right? And here, one thing that you have to remember, IBA can be both natural as well as synthetic. Natural as well as synthetic, right? Natural as well as synthetic. So when it is the natural one, matlab it is also synthesized by the plants, but in the horticulture, it is also synthesized artificial and it is used. So this is what you have to remember, okay? So cell elongation, apical dominance, all that are points. Baki 2,4-D, 2,4-T, they are also there. So IBA can be used as both. It can be synthetic rooting hormone as well. Right? Done, beta? Sure? Sure? Done, beta? So this is what we have here. So IBA can be both. So it is used for rooting, right? So it is also used synthetically. So it is basically used for rooting. I'll discuss that too. So these are some pointers, extra pointers which we have to discuss. So when it comes to the characteristic function, rest you can just read the NCERT. But yeah, bacha, apical dominance is there. So what is the meaning of apical dominance? It's simple. See, tip is going to make the auxin. And you know that it will be having what? Right? The tip is going to make the auxin, and you know that it is going to have what? It is having the polar transport. It will move downward. So because of that, it will get collected there. So lateral buds, they will not be able to grow. Lateral buds, they do not grow. So this is apical dominance. Right? This is apical dominance because auxin will form here. It will go downward. Right? It will get collected there, so it will not allow the lateral buds to grow. So this is what we have as the apical dominance. So what is the, see, no doubt cytokinin can overcome the apical dominance, but in general, you know that decapitation, when we remove the tip, right, decapitation will help in increasing the lateral buds, so in their growth. So this is what, you know, For hedge making, this is what is used for tea plantation. You can just remove that tip, then lateral buds will grow. So phenomena in which apical bud dominates the growth of lateral bud, right? So in the presence of apical bud, lateral buds will not grow. So shoot tips, when we remove them, then lateral buds will grow. So for in tea plantation, you can just remove it and you can grow them. And in hedge making also, hedge in the parks, you might have seen that they will grow. So that's what you can keep in your mind. Baki cell division and enlargement is there, but mainly for cell division you have cytokinin, and auxin also controls the xylem differentiation. This is important. Baki beta, it can cause, jaise there has stem cuttings, it is responsible for rooting, and IBA to specially used for that, okay, for rooting, for the root growth in the stem. Baki bache, IAA, NAA, they all prevent premature abscission. Means in the case of young, right, in the case of young plant organs, they should not fall down. But yeah, they also promote the abscission of older mature leaves. Baki beta, auxin promote flowering in pineapple and lychee. You will not find it anywhere, but in the case of pineapple, you should definitely write it. Okay, you should definitely write it. Is that clear? Is that clear? Is that clear? Any doubts? And it's a beta, please do solve the PYQs on the basis of that, okay? So this is what you have to remember in the case of auxin, okay? Baki beta, it also induce, done? Chaliye. So baki beta, it also induce the parnithocarpy, that is partheno, wow, what am I saying? Parthenocarpy ko parnithocarpy. Beta, I need a cup of tea. So parthenocarpy here, it means it will result in seedless fruit. Why? Because bache, the ovary here will get transformed into fruit without fertilization. So seed is the product of fertilization. If fertilization is not there, seed will not form. So by giving the hormones, we are directly changing that particular ovary into the fruit. So in NCERT also, this example is given. Examples you have to follow as per NCERT. So do not make mistake here. In tomato case, they have given. Baki dichlorophenoxyacetic acid, I told you, it is used to kill the dicot weeds. This is again important PYQ. It doesn't affect the monocot plants. Baki bache, this is extra, that malic hydrazide, NAA, right, they keeps lateral bud of potato tubers dormant. So potato tubers can be stored. So this is extra, but rest everything is as per NCERT. So this is the diagram also they have given. Okay, then beta gibberellin, it's just like growth hormone, G for growth hormone. So gibberellic acid. So ABA is its antagonist. You know na, ABA, stress hormone, its antagonist, means their function is in opposite direction. So this main function here is beta bolting. What is this bolting, ma'am? This is the main function here, bolting. Jaise, do you know about the rosette plants? There are some plants in which internode length is so short, right? The leaves, they will be at the node. The leaves, if they will be at the nodes, internode length is so short, they look like this kind of, leafy. So rosette plants. So in the rosette plants, just prior to flowering, right, their internode length elongate. That is what we call as bolting. That is what we call as bolting. So this bolting is a characteristic feature of gibberellic acid. So it means that internode length will elongate beta. So gibberellin induce internode elongation just prior to flowering in rosette plant like beet and cabbage. So this is the bolting. So this is the characteristic function here. Am I clear? So this is the characteristic function here. Is that clear? Done? Sure? Sure, bache. Chaliye, let's move to the next part. So elongation of genetically dwarfed plants. So when gibberellins are applied to dwarf maize, pisum, even they are genetically, right? Even they are genetically small, but still they can do what? you have here. So in genetically dwarf plant also it can help in increase in length. Baki beta gibberellin, it induces the synthesis of hydrolyzing enzymes like alpha amylase, right? Alpha amylase, lipases and proteases that will help in seed germination. How will it help? It will help in beta seed germination by promoting the breaking of endosperm. So in seed germination also it is helping. It is increasing the length of grape stalks. So it will elongate and improve apple fruits as well, right? Their shape as well. So that is also its role. Okay, so it also speed up the malting process. So this is totally mammary based. So malting process is also it is going to speed up. So that is why it is useful in brewing industry. It increases the height of sugar cane, right? So because of that, what will happen there? Yield will increase by about 20 tons. Baki beta, it is going to delay the senescence, that is aging, because it is helping in the growth. But the main effect of delaying of senescence is of cytokinin. Okay. Okay, so spraying juvenile conifers with GA can speed up the maturity period, thus leading to early seed production. Is that clear? Is that clear? Done? Chaliye. So beta gibberellin na, this is extra function that I am telling you. So gibberellin, this can also basically replace or mimic vernalization. Do you know what is vernalization? What is vernalization? Vernalization is low temperature treatment. Low temperature treatment. जैसे बेटा in some plants, जैसे biennials and all, they need the low temperature for their flowering. मतलब they have to pass their lifespan through a phase where the temperature is low so that they can do the flowering. So this is vernalization, low temperature treatment. So in some plants, for their flowering, we have to give them low temperature so that they can flower. So we can, if gibberellin is directly given to that plants, so it mimics or it can replace that vernalization. It mimics that effect. मतलब giving gibberellin is just like कि flowering will be induced in such plants, right? Flowering will be induced in such plants. This is also something that you can write down in NCERT. Baki. In the case of cytokinins, right, they have many effects, cell division. So here you will talk about Richmond-Lang effect. In the case of cytokinins, you will talk about Richmond-Lang effect. So what is that? It means to delay senescence. To delay senescence. This is the meaning, right? Because this cytokinin, you know that kinetin is what? Kinetin is synthetic, and I told you already beta, it is synthetic, and I told you that zeatin is going to be what? Zeatin is your natural. Zeatin is your natural. So cell division is this hormone will do. So obviously aging will also be delayed, and moreover beta, this is very important which you should know. In tissue culturing, how will it used? जैसे low cytokinin है but auxin is high. Cytokinin low, auxin high. Root differentiation. So auxin, more auxin will help in root differentiation, but if there is high cytokinin but auxin is low, then it will result in shoot differentiation. So this is an important part. This is morphogenesis, root and shoot development. Baki yeah, cytokinin is also used to overcome apical dominance. Richmond-Lang effect, that is, it is going to delay senescence. It promotes the lateral shoot growth and even the adventitious shoot formation. Is that clear, beta? Is that clear? So nutrient mobilization is done. New leaves will also form due to that. Baki beta, ethylene. Ethylene mein the triple response, it is also its bias. The triple response, it is also its bias. What happen is, let's suppose there is a seed. If the seed is growing, the seed got the hurdle. So here what is happening? This seedling will start growing like this. Matlab, rather than growing vertically, it will start growing horizontally, due to which the swelling of its axis will be there, and then it develop as apical hook. So this is what triple response. Whenever you hear the word triple response, mark ethylene without any doubt. That basically there will be the horizontal growth of the seedlings, the stem thickening will be there, and apical hook will form. And ethylene, which is a gaseous hormone, we know it very well beta. Ethylene, which is a gaseous hormone, we know it very well that this ethylene's role is to, right? This ethylene's role is to what? Is to what? Fruit ripening. Is that clear? Is that clear? So that is what you have to understand here. So ethylene, simple gaseous hormone, beta, right? So the part which are undergoing senescence, the parts which are undergoing senescence, undergoing aging, they will have more ethylene formation. Baki banana, apple, tomato. So some slides, I think they are missing from this part. Baki beta, in the case of ethylene, there's a triple response is something very important. Apical hook formation. Firstly, horizontal growth of seedling, swelling of axis, apical hook formation, very important. So they are basically in that part which is undergoing aging, senescence. So yes, beta. So there you will see. Baki, they will help in ripening of the fruit as well. So flowering may synchronizing fruiting in pineapple. So pineapple part you also read in auxin. Auxin will do what? Promote flowering in pineapple. So that's how you can relate. Okay, so promote flowering in pineapple, and what this ethylene is doing, it is flowering and synchronizing fruiting in pineapple. So flowering in mango is also done by this ethylene. Baki, it is also causing thinning of coconut, cherry and walnut. Baki beta, banana, apple, nowadays ethylene is used at commercial level. Beta, ethylene is what? Anybody in the class who knows? You cannot say that ki it is basically the ethylene, right? So in general, ethylene mein you have to know fruit ripening thing, you have to know triple response. Then I will tell you about the climactic fruit. But now I will tell you about the ethylene first. Right? Let me talk about the ethylene. So here slides are little, these pointers are little jumbled, right? So we will discuss that. Beta, ethylene is the source of ethylene. It is not ethylene, but yeah, it's the source of ethylene. It is used commercially. What is it? It is SEPA. It is 2-chloroethyl phosphonic acid. What is this ethylene? Not given in NCERT, but this is important. So it is basically 2-chloroethyl phosphonic acid. It is used commercially as a source of ethylene. So it enhances the respiration rate during ripening of the fruits. It will enhance that, and such fruits whose ripening rate increases, respiratory rate increases, they are the climactic fruit. Just your apple, your banana, they are climactic fruit. They are having their respiration rate is high. Just in the case of apple, you might have seen when you buy the apple, they are covered with a kind of wax paper. Why? Because that wax paper is not just to beautify it. The wax paper is covering it because to make sure that oxygen should not penetrate more. Because if it will get more oxygen, the ripening will be faster. The respiration will increase. So ripening will be faster. So this is the meaning of climactic fruit here. So that is what you have to remember here, beta. So this is given in your NCERT exemplar. Is that clear? Done? Done. So ethylene functions are important. Apical hook formation is important. So climactic fruit, you have to remember that continue to ripen even after being harvested. Take care. What are climactic fruits? What are climactic fruits? So climactic fruits are the one which even continue to, their respiration rate is high. They continue to ripen even post harvesting. They continue ripening post harvesting. Right? That's why in the case of apple and all, I'm telling you, they cover it with the wax paper. They cover it with the wax paper because they have to make sure, right? They have to make sure that oxygen should not pass, and you know, the respiratory rate should not increase. Baki beta, it promotes root growth, formation of root hair, increase the absorption surface. Baki femaleness in cucumber, that more female flower means more seeds, that will be done by this. So it breaks seed and bud dormancy. So it is its promoter function. Okay, so it initiate. So these examples which are given in NCERT, they are literally very important. So it helps in germination in peanut seeds, sprouting of potato tubers. So it also promote, that is again a PYQ, internode or petiole elongation in deep water rice plants. So it helps leaves and upper parts to remain above water. So that is what we have in case of ethylene. So when it is ethylene, do remember what? Do remember the triple response, very important. Baki bacha, ABA, which is your stress hormone, antagonist of gibberellic. This is also the anti-transparent hormone. So it's not a bad hormone. It's not like that, okay, it is a bad hormone, right? It is the hormone which is making sure that plants should survive in the unfavorable condition. That plant should, right, a plant should survive in the unfavorable condition. If let's suppose a seed is not getting the favorable conditions for its growth, so it will be like, seed bro, you should be in the dormant stage, you should not grow, right? You should be in the dormant stage because now the conditions are not favorable for you. Just say, let's suppose I know your parents are angry, I'm telling you, beta, do not go to your parents, you are angry, don't do this, otherwise they are going to beat you. Now I'm trying to protect you. Now if you will not listen, you will go, you will come up with 2 or 3 slaps. So that is what I'm trying to tell you. This is the role of ABA. So ABA is like, it induces senescence, aging and abscission, means of leaves and fruit by increasing the activity of cellulase and pectinase. It induces the seed and it increases the bud seed dormancy. It will inhibit seed germination, right? Why? Because conditions are not favorable. So that is why we say that it play a major role in seed development, maturation and dormancy. So by inducing dormancy, it will induce the dormancy in that, so it will help that seed to withstand desiccation, to withstand or to survive in unfavorable condition. Baki beta, when plant is under water stress, it will close the stomata. Why? Because then the gaseous exchange will not be there. Loss of water, basically it is closing the stomata so that loss of water in the form of vapor should not be there. So it is antagonist of GA, it works opposite to GA. That is it. This is all about the hormones. So here you will also find a comparison table which you people can later use for the quick revision. Okay? So this is all about the topic today, and here you can see even the NCERT last part is saying jaise hormones hai beta, many hormones, they work together, right? All that things are there, genomic control and everything I have discussed, right? So they work together, right? They work together also. So here they have used even the word vernalization. S is there, by mistake I mentioned Z, rectify that. So this is it. This is all about the plant physiology. We have discussed everything. So you can just simply read the NCERT part, right? You can just simply read the NCERT part for your quick revision. Is that clear? Is that clear? So let me check if there is something that I have to share. So regarding uphill and downhill, I have already told you, as per NCERT, when we go, what is the explanation? In general, what is the explanation? So here a correction is required. So this part I'll correct and then I'll share the notes. Right, beta? Then I'll share the notes with you all. So this is all about today's session, right? So complete plant physiology is there, right? Complete plant physiology is there. Now you can just have, chalo, let's solve this particular question, and then we will end our class. So I'm going to start the poll. You can have a look. Formation of interfascicular cambium from fully developed parenchyma is an example of. Quick. So you know that interfascicular cambium. So it is 2026 PYQ beta. So you know that basically what is going to happen. It is basically, how will it form? It is basically when there is when these medullary cells, when these medullary rays, when they will again start dividing. So obviously we know that it is because of the so interfascicular cambium. So it is because of the D-differentiation. Is that clear? It is because of the D-differentiation. Nahi beta, fully developed parenchyma is what? Fully developed parenchyma is the permanent tissue. From that permanent tissue, you are making interfascicular cambium, which is the example of secondary meristem. So this is because of the D-differentiation. Okay, it is because of the D-differentiation. Chaliye, next. Spraying sugar cane crop with which of the following plant growth regulator is going to increase the length of the stem? Quick. Is going to increase the length of the stem? Quick. Done? So, sugarcane. So, we will talk about gibberellic acid, isn't it? So, let's see. Very good. So, question will come from PGRs. So, which of the following phytohormone will promote the nutrient mobilization and delay in the leaf senescence? So, you know that it is going to be your cytokinin, isn't it? Chale, now answer this one. Answer this one. Quick. Answer this one. It's a very simple question. It's a very simple question. Quickly answer it. Here we go. Answer it quickly. So it's a very simple one, isn't it? It's a very simple question, isn't it? So beta 2,4-D, we know that herbicide, this is going to do what? Brewing industry, kinetin, nutrient mobilization, ABA will go there. So A will go with 3. You got your answer. It is going to be third one. It is going to be third one. Is that clear? Is that clear? Chaliye, one more question. You see, this is easy. Heterophilous development in response to environment is an example of which phenomena? Plasticity. So that is it. I hope you enjoyed the lecture, beta. So this is all about this, and do let me know that which unit you want me to start next. Okay, so if you like the class, right, if you like the class, then do let me know in the comment section. Notes will be provided to you in my personal Telegram as well as in our WhatsApp channel as well. So take care, guys. All the best. Thank you so much. Do let me know in the comment section which unit you want me to start next. Take care. Bye bye. Okay, quick.